Friends, for the second technical session of today. The theme of the present session is Ayurveda through the lens of material science, data informatics and technology. Today, we will explore how material science, data informatics and technology, these three fields are intersecting with Ayurveda, a traditional system of medicine that has been practiced in India for thousands of years. Material science can help us to understand the properties of natural materials used in Ayurveda and how they interact with the body. Through the use of advanced analytical techniques, we can better understand the chemical and physical properties of these materials, which can inform how they are prepared and used in Ayurvedic remedies. Data informatics can help us to better understand the outcomes of Ayurvedic treatments and how they can be optimized for individual patients. By collecting and analyzing large amount of data related to safety and efficacy of Ayurvedic treatments, we can develop evidence-based approaches that are tailored to individual patients' need. I extend my heartfelt gratitude to our esteemed speakers who bring a wealth of knowledge and expertise to this session. I also express my appreciation to all of you for joining us today, as your presence reflects a shared commitment to exploring. Thank you, and let us now embark on this enlightenment journey together. In this session, we have 7 renowned speakers from different parts of the country, and to chair the session, we are delighted to have Dr. Anupam sir and Dr. Kunal Bhattacharya sir here. Please give a regarding our virtual session chair, Dr. Anupam Srivastava sir. Sir is presently working in Patanjali Research Institute as a Chief Knowledge Officer. He is associated with various medicinal plant survey projects in different parts of the India and various scientific publications of Patanjali Research Institute. Previously, he has also worked in Maharshi Ayurveda, New Delhi, as a senior research scientist and factory manager for 14 years and was introduced in the field of Ayurveda research. He has published various research papers in the field of environment science Ayurveda, medicinal plants and plant taxonomy, and has more than 40 years of research experience in various fields of botany and medicinal plants. We welcome you, sir. Please give a huge round of applause. Regarding our second conference chair, Kunal Bhattacharya sir. Sir is working as a principal scientist and head of department in Nano Ayur Biosciences at Drug Discovery and Development Division, Patanjali Research Institute. He had received his PhD from University of Duisburg-Essen, Germany, and has done his postdoctoral research work at Dublin Institute of Technology, Ireland. Dr. Bhattacharya's current research work involves the development of Ayurveda-based herbal and mineral nanomedicine with the purpose of enhancing their therapeutic values. He has also experience in performing reverse pharmacology of Ayurveda-based medicine. We welcome you, sir. Please give a huge round of applause. He is coming. Okay, we will move further. I request our conference chair to please introduce our first speaker.
Home. Our first speaker, Dr. Saket Ram.
This presentation is online.
He is online with an extensive research in the field of Ayush informatics Dr. Saket Ram is very well known for his work on the Namaste portal and distillation of Sanhitas. He has worked with National Institute of Indian Medicinal Heritage for more than a decade. He currently works as an officer on special duty in the Ayush Grid, Ministry of Ayush, Government of India. I welcome Dr. Saket Ramji. Please, Dr. Saket ji.
Namaste to all. Am I audible?
Yes. Please continue.
Respected Swami ji, Acharya ji, and Chair Anupam sir, and Co-Chair Kunal Bhattacharya ji, and dear participants, students, and the team members of Patanjali. To begin with, my apologies. I could not make in person due to official commitments. Very sorry for that. But anyway, thanks to the organizers for letting me present the work attempted by Ministry in the digital health platform domain. So I'll just present a case. It's not a very specific one-point solution, but we are going to showcase the efforts of the Ministry to develop the digital ecosystem to achieve the objectives as stated by our compare today. That was a very nice introduction. So let me share my screen. Yeah, hope my screen is visible. Can somebody confirm?
Yes, please continue.
Yes, sir. Let me just go into presentation mode. So this is going to be a very brief introduction to what Ministry of Ayush is attempting to create a digital ecosystem for Ayush domain in general, and of course with respect to Ayurveda in specific. So now technology, it's a very broad term. It's application of scientific knowledge to the practical aim of human life, or as it is sometimes phrased, to the change and the manipulation of human environment. So this is not a new phenomena. So the day we have decided to go up on feet, and the humans have taken a completely different trajectory in terms of evolution. See, like our counterparts like apes and other animals. So humans are a little bit distinct because we have decided to walk on 2 feet, and we have developed tools. We know how to utilize the mechanical tools. We have a great ability of empathy to learn from each other very quickly. So humans have a different kind of evolutionary advantage, which brings in a lot of technology and ability to use different types of technologies. And here we are going to be very specifically talking about the digital health platform, wherein we are going to envisage multiple components of the requirements, which will enable us to harness the digital health revolution to the advantage of Ayush systems. So as we have heard that there are different components, we get information from different platforms, and we need to streamline it someplace so that we can optimize the information. Mostly it is happening in verticals. Most of the institutes are doing their own work, and a lot of journals are publishing good work. There's a lot of public health initiatives are going on. Many state governments are doing good work. Central government is also putting in efforts. A lot of private organizations are doing very good work in India and abroad. But we need to have a platform where we can have exchange of this information so that our efforts are very clearly demonstrated. At the same time, we'll be able to learn from each other. So basically, Ayush Grid is a platform to enable this digital health ecosystem for the Ayush in general. So what's the definition of digital health? Digital health means the field of knowledge and practice associated with the development and use of digital technologies to improve health. Digital health expands this concept of e-health to include digital consumers with a wider range of smart devices and connected equipment. It also encompasses other uses of digital technologies for health, such as Internet of Things, artificial intelligence, big data, and robotics. So this is a very generic definition. And this is a networked world. We know we are all connected through internet, and we all use extensively the mobile mobiles nowadays. It's highly connected, especially in India. It is all the more very relevant because we are one of the few countries where we have very good access to internet compared to many other developed countries also. So though it sounds a little strange, but it is a fact. And this comes with a lot of advantages, and we have to use for the benefit of health care delivery. So that's all we are going to talk about now. So in 2030 Agenda of Sustainable Development 1 highlights that the spread of information and communication technology and global interconnectedness has great potential to accelerate human progress, to bridge the digital divide and to develop knowledge societies. So this is a UN charter which says that there is a lot of potential for harnessing the digital technology, and we are of course doing it, and we are trying to do it better in a very concerted manner. So what is a digital transformation? So it is an extensive thing. It comes in various forms, and we have to utilize whatever is available in the form of lot of data is available. We have to make meaningful conclusions out of that. So we have different tools, and as we have a lot of wearable devices nowadays, so we get a lot of real-time data also from the people who are using it. So that comes with a lot of advantage, especially in case of Ayurveda. I would like to tell you, because most of our audience are from Ayurvedic background. So earlier it was very difficult to conceive a project or say handle an Ayurveda based project because there are so many variables involved. Till date we know the kind of experimentation or say clinical trial setups where it is highly controlled. They are working on few very much set patterns or say set parameters. It was very difficult to envisage multiple parameters to be conceived because of the computational difficulty fundamentally. But today, thanks to the advancements in hardware and well as also the kind of computational techniques we have, we really have a supercomputing ability, and we can do these things on the fly now. So in number of parameters also we are able to handle. So that's how the big data analytics come into picture. So now it is not a limitation, but it is an opportunity. So we can virtually see a person's 24-hour and 365-day data and collect it from multiple people from population and see that what's happening to them. So this is, in a way, is a good opportunity for Ayurveda-based research. So now we are also talking about siteless trials. You see, this was completely unconceivable few years back. Not just few years, just before COVID itself, it was not thinkable. So trial means it's always a very controlled setup. Physically persons will come and do, but the COVID has changed all these dynamics. So now even online method also we can conduct siteless trials also, where person is in their home or say in a particular setup wherever they are comfortable, and the trial can be still carried out in a remote way. So these are the new opportunities which the digital technology provides us, and we have standards, we have teleconsultation standards, many standards are in place. Some of the standards are being developed, so that comes to a great advantage for all of us. So what is of course this is just a network of a display of how the Ministry of Ayush works. It is a government body, Government of India's ministry, working with all government and non-government agencies to streamline Ayush sector and also ensure that it is being utilized for the public good in a very efficient manner. So what is Ayush Grid? Ayush Grid is a unit of ministry especially conceived to harness the digital device advantage. So basically it works as a connecting unit to enable the usage of ICT, information computer technology, in health services, education, research, drug administration, medicinal plans, and of course on a larger plane it assists ministry to achieve its oversight. So the aim, the vision of Ayush Grid is to provide efficient, holistic, affordable and quality services to all through a secure and interoperable digital ecosystem. So we do have a digital ecosystem in place now. The Ministry of Health also has come up with a great solution called Ayushman Bharat Digital Mission. So Ayush Grid is a part of that, which of course has a more focus on Ayush, especially Ayurveda and other related indigenous systems. So its mission is to create organic and dynamic information and communication technology powered network interconnecting all streams of Ayush in their key functional areas, as I've already mentioned, in healthcare delivery, capacity building, research and development, drug regulation and education. So this will be beneficial for all stakeholders of Ayush and helpful for effective governance. Strategies for development will be in sync with the national and international policies and healthcare needs. So these are the broad goals: functional, technical and operational. I'm not going to get into that. It's understood. This is also self-explanatory. Of course, there are few applications developed already, and there are many other applications developed by the autonomous bodies of the ministry, like research councils and other things. So we'll get to a few examples a little later. So fundamentally, I want to emphasize on one very important point, that the whole system is being like getting transformed and more focuses on the citizen now. Earlier it was institutional centered. Basically, a person, if somebody falls sick, it was the person was looked at like which hospital will take care of him or which primary health care will take care of him. But now it's not the case. The citizen has the ability to choose, store his information or her information and share as they want. So now the focus is completely on the citizen, and every system is there to assist him. So our whatever we are going to design, whether it's in the public sector or private sector, hereafter our focus should be citizen. And I have seen wherever the citizen has been given more importance, those examples are highly successful. Of course, the example of Patanjali itself is a great like use case here, where they provide such a great facility to the citizens, and that's how it has become such a big enterprise, and it has set as an example. So is the case with the government agencies and other aspects. Wherever we are trying to create any facility, whether it is digital or non-digital, the focus should be the citizen. And of course, the Ayush Grid's main aim is to bring in all the existing digital health solutions with respect to Ayurveda, Siddha, Unani, etc., to the benefit of the citizen. So the fundamental prerequisite of achieving all these things, what all I talked about, like whether it is databased analytics or say artificial intelligence, whatever we want, successful drug trials, whatever has to happen and reach the patient, it starts with very fundamental thing, the block called patient record. So whether it is in a clinical trial or in a hospital setup or say general OPD setup, if these records are well maintained and we are able to capture the data in real time, so that will speak for itself, real life evidence, and also that can be subjected to further clinical trials, and we'll be able to have very good evidence, quality evidence, real time evidence. So for this, we need to start with very fundamentals. So basically, research does not mean that it happens only in the laboratories. Good recording practices are the key for any kind of great research and ultimate public utility and translation of the same. So this fundamental thing we all have to understand. My request to students sitting there and all my colleagues, especially the medical doctors, that whenever you do any kind of patient consultation, maintain your records very well and try to capture them electronically in some other format. That's a very fundamental block where we have to start with. And to enable this, we should have certain coding systems, some standardized terminologies and all, wherein I was fortunate to contribute with all of my colleagues from different councils and other agencies. The first outcome is the Namaste portal, National Ayush Morbidity and Standardized Electronic Portal, launched by Honorable Prime Minister during 2017. And then we are trying here, of course, many of you may be knowing it or may not be knowing it. I request you to please go and visit this Namaste portal. It is a humble attempt to provide some, to some extent, standardized Ayurveda, Unani, Siddha terminologies, and also morbidity codes. As I said, our fundamental block starts with the coding and the counting of the, at least the visit of the patients. I mean, that's a fundamental thing which will give us top 10 diseases for which patients who are visiting us and all, such kind of a data we can capture. And it did not stop there. As you can see, WHO was happy with this work and they have taken it up, and now the Ayurveda terminology is released by WHO. We have the documents online. You can search standardized terminologies and WHO, you'll get it. And now another good attempt is going on to add the morbidity codes into the WHO ICD TM2 module. So that's in the alpha draft stage. Maybe by next mid-year, we'll have Ayurveda, Siddha, Unani codes as part of the ICD-11. That's a big paradigm shift, wherein we are going to be coded along with the rest of the medical codes. So Namaste portal is basically kind of a backbone for Ayush Grid because it works at the core level. I mean, the fundamental level where we are creating to create, we are creating to set up an ecosystem on which we can build the futuristic tools. Like now we have come up with the morbidity codes, now we are preparing the procedure codes, we'll have the drug codes. So all this will help us streamline the data very efficiently. And these are the few outcomes of the Namaste portal. You see, one humble effort leads to many outcomes. So I already have mentioned. So we have an AHMIS where we are promoting the double coding. The NABH could publish glossary of Ayurveda terminologies, and many other works are designed based on this idea. And also this gave us, when we were working on the Namaste portal, we understood that how to actually organize our information so that we can utilize the same information very effectively in the digital ecosystem. So of course, this is the navigation of Namaste portal. It's very simple website, namstp.ayush.gov.in. You're welcome to visit the site and examine, and please let us know your feedback. So I'm not going to stay on these slides for a while because the time is very short. I know other speakers are waiting. So these are the outcomes which we have already discussed. So I'm skipping all these things. You please visit the website. These are all self-explanatory. I request my colleagues there to please help the delegates to visit this website. So these are the use cases. This coding system is useful in retrospective and prospective data collection both. So we have some examples on the Namaste portal. You can see yourself. So again, this is a reiteration of what I have already spoke. And now we are able to actually use these codes as part of the scientific publication also. So this particular article has used few of the Namaste portal codes. You can see here. You can see, for example, cervical spondylosis, lumbar spondylosis. So they have used the Ayurveda and Unani codes for counting and also highlighting them as part of the discussion. So we can now capture this data from research as well as from the real life hospital setting also. So these are some of the publications based on the Namaste portal. So I'm just flashing them. And we are also using it as part of the publication of the book also. For example, Sahasra Yogam book. Sahasra Yogam, it's an English translation from CCRA's earlier Hindi translation, where we have catalogued the yogas against different indications. So there we have used the Namaste codes. So you can see a few examples here, one page here. For example, for Abhishanda, which is HF1, which a simple English translation is given for convenience. It doesn't equate the same disease, but for convenience. And we have the yogas here. So along with the page number and other things. So these are the few attempts. So now it's very easy. So to map the indications to the coded diagnostic. This is one example. Yeah, these are the use cases which I already have discussed. So I'm not going to repeat that. It is self-explanatory. Please visit the website and let us know the feedback, and please start utilizing this is the completely open-ended information. You can even download the Excel sheets also. So please make the use of them and please give us the feedback. So this is the terminology document of Ayurveda, which is already in the public domain by WHO. This work was built on the Namaste portal's backing. And you must be knowing about traditional knowledge digital library. This is a huge initiative, almost happening for almost now 15-16 years by CSIR and then Department of Ayush now along with the Ministry of Ayush. So they have catalogued around 1.5 lakh formulations. Of course, now the data is available only for patent offices, but I'm told that very soon they are going to have a limited access through proper channel. So if that happens, we'll have another big repository for understanding Ayurveda and allied systems very efficiently. So these are the huge digital repositories where we could come so far. And we have something called Ayush Hospital Information Management System, wherein around 126 hospitals are using it now. So we are using dual coding, like we'll be coding each instance through ICD-10 as well as also through the Ayush morbidity codes, which I have just spoke about earlier. So there are some other portals like Ayush Information Hub and all. Of course, I'll be sharing these with Ashwini so that you also get these links by the time you leave Patanjali. So these are the few low-hanging things which we have published so far from the ministry. So these are all free tools. Anybody can come and use these tools. So Ayush Research Portal is there. Ayush Clinical Case Repository Portal. Now we are going to migrate it as Ayur Cell by Amrita Vidyalaya. So where you can have the case discussions. So before they publish as case series, and also ultimately some trial happens. So wherever there is a clinical success story, so they will be available on this Ayush Cell or Ayush Clinical Research Case Repository Portal. So this is how we start gathering generating the data. Of course, we have very good applications for yoga break and all. So again, I must humbly acknowledge the contribution of Patanjali, especially Baba Ramdev ji, in propagating yoga across the world, and how it took shape into the form of Yoga Day and all. So as a humble service towards that ministry is also like sort of escalating it in terms of creating different tools from the government side. So these are the few applications, and other things are also happening now. We are also going to be on the PM Gati Shakti portal, that is National Master Plan, wherein we are going to map with all the specific geolocations, all the hospitals, all the infrastructure from the Ayush, from public and private sector on one portal, so that we can understand how best we can use the resources from other ministries and other agencies, state government and other things, like maybe power, maybe the connectivity, last mile connectivity, all that we can see on this GIS portal now. These are the few other student-centric portals. For example, Ayush Next portal is meant for students to understand what's happening in the ministry. So these are the few other portals. I want to just share the information for your benefit. There's an Ayush NGO portal, basically for all the schemes, central sector schemes and other schemes. If somebody wants to apply, they have to come through this portal now. It's a very transparent portal. So they can apply and also see how the sanction procedure is happening, and all that is streamlined through this particular portal. There is a National Ayush Mission portal for the public health initiatives of the Ministry of Ayush. Of course, many people, I hope you must have seen these samhitas, Charaka Samhitas and e-books. They are available on National Institute of Indian Medical Heritage website. So if you are already using, very happy. Otherwise, if you are not using, you can go and visit that. You can use the very good Sanskrit Mulapatha of Charaka Samhita, Susu Samhita, etc. is available. And I am also going to cite some good examples from the other players who have come up with very good online solutions. For example, see this Charaka Samhita online edition. It's a very good translation by a group of people. Very good translation. I recommend people to visit this site and use this information. It's completely free and it's available online. There are very good number of articles are also available on this. So this is for the students' benefit. Now, with the tools like ChatGPT and all, so they can learn quickly from this kind of information, and this kind of huge data available in electronic format also helps us use the machine learning techniques and process our data. We can try different things from this kind of resources. Of course, this already we have discussed, and Ministry also has worked with WHO and released a web application, mobile application called WHO M Yoga. This is available online for Android as well as for the Apple phone. So this basically talks about like yoga lessons based on the International Yoga Protocol. So I recommend you guys to try this. It's a very good portal. And there are also other private sector people, young fellow from Kerala. They have come up with a very good app called Dravya app. I'm not promoting anything per se, but whichever I found useful for the students, I'm trying to flash here so that they can straight away use. This is also a very good portal where we have information about the single ingredients like plant, mineral and animal origin drugs and related information. Very good application. And now a lot of things are happening using the AI technology. Of course, which is in the very early stages. Ministry is trying to work with WHO and International Telecommunication Union to come up with the basic ground rules, what should say, benchmarks for usage of AI in traditional medicine in general, and also for specifically for Ayurveda, Siddha and other systems of medicine. So let's see how it's going to pan out. But ultimately, there are already few players who are working in this domain, and let us see how it's going to pan out. It is just happening now. So there are good side and bad side of it. So we should be able to see the best part and should be able to use this technology for our benefit. There are many other things like there are BLQ and B Mobile initiative by WHO, and many things are happening. So whatever is there to conclude, I would like to say, so we are having almost 75 years of public health initiatives in the global domain, and Ayurveda Siddha are a part of it. Ayush is a part of it. We know this already I have told. We are coming up with AI benchmarks. So what happens? Now this is the time to invest in the digital health. That is very important. Otherwise we'll be left behind. But we also have an advantage of joining late, but at a very appropriate time. So there's no question of comparison now. So we are a part of system agnostic healthcare ecosystem. So ecosystem means it covers everything. It is not specific to Ayurveda or modern medicine or whatever. So we are a part of a big picture now. So wherein we have to find our space and create our own ecosystem, right? Proper investment intellectually, materially, and many ways. So these are the few slides I borrowed from WHO to showcase the importance of it. So finally, I would like to say, because most of the audience here, I assume, are young people, that there are no digital quick fixes for young people's health. Digital solutions work best when they are part of the existing health system. So we should be mindful of it. So we can't create a virtual system and imagine that that works better. No, it doesn't happen like that. All these are basically assisting tools. They work best only in a real-time setup. So they can make things better, but they can't replace the proper people and proper setup. So with that one last remark, I also would like to say that the people should be involved in that, especially the younger people, and also all stakeholders have to be involved whenever we design a digital health solution. So that is a very important message I would like to emphasize on. Even in ministry or other places, I have seen many times wherever the stakeholder involvement is missing, that product fails. We launch many websites, many things happen, but very few have become very successful. The reason is very simple. So there is no proper consultation of all stakeholders. In such undemocratic method of doing results in this kind of disasters. So we should be mindful of it, and we should be able to consult everybody and get the best. So there are no digital quick fixes. Again, I'm trying to say we have to plan and invest wisely to support the health system needs. So thank you for this great opportunity. I know I have not been any specific here, but I wanted you to get sensitized that we are in the process of building an ecosystem, and Ayurveda is here to enable, and views of all stakeholders are important for us. Don't look at this as one public department, say government department. Basically, it is an ecosystem creating department where we are here to listen from all users and try to bring in people and networks so that we'll be able to build a great digital health ecosystem for Ayush. Thank you all. Namaste. Thanks.
Thank you, Dr. Saket ji. Dr. Saket ji talked about Ayush Grid, digital health platform, AI for e-health, digital therapeutics, digital trial, and different applications of digital technology for Ayush. And we thank him again for this informative talk. Thank you, Dr. Saket ji. Any questions from audience? Yes, please.
Hello, Namaste sir. It was really pleasure to see the Charak Samhita portal, online portal to cite for. Sir, is there a portal being made for different sutras of the Samhitas and Vedas, which also consist of so many important treasure knowledge to be cited? But because usually we find it in the textbooks, Sanskrit textbooks, which are actually a little bit harder to, convert them in as a reference in the English journals. Yeah, thank you, sir.
Yeah, thank you. Great question. Right now, Chikitsa Sutras, we are working on them. In the Namaste portal itself, you will get this information. And yeah, definitely I'll try to find out from the Vedic sources. We have a project going on with Tilak Mahavidyalaya, where they are collecting the information from all the 4 Vedas. So I think once they complete this project, of course, only 1 year has been completed. But as and when we have an access, we will try to publish on Namaste portal. So you will get that information also. Thank you for the question.
That's very nice. Any more questions? So one thing that I would like to ask you, sir, about the digital Ayush grid. And the question is that there is a lot of public data that will be filled out in those grids. So there are 2 perceptions about that. One is the how much of this data is accessible for future researchers who want to look at the, want to do some statistical analysis or online research. That's one question. Second is the, because it's a digital footprint, so what is the safety of the public data that will be uploaded on that website?
That it is not one single platform where we are going to pull all the data and put in one website or say one database. It is not the case. Basically, the job of Ayush Grid is to bring different types of resources available to network between them. It is basically a federated thing. So the data will be there where it is generated. I mean, whichever is agnostic, whichever is shareable, that will be shared in common standard database, metadatabase formats in a very neutral platform. The government need not own it. So we are thinking of creating a platform where different stakeholders can come together and create this data archiving system. So it is not idea of the government to create a single system so that everything is controlled by them or say somebody is owning it. It is not the case at all. That's not the idea of government at all. It is, for example, if Patanjali and like for example now JIM, it's a good initiative. I can see if this kind of an ecosystem can come and we can have our own, for example, for gene pools we have, for gene-based data, for biochemistry, for many databases are there which are completely open access and they are run in a very nice community spirit. So for Ayush-based data, we do not have much in that aspect. Of course, few government initiatives are there, but still that ecosystem is yet to develop. So where there we come into picture. Our job is to, that's the reason I am here to speak to you. I mean, that's one of the reasons. So now we have the opportunity to build our databases where we are having the ownership also. Of course, the security also will be done as per the international guidelines and things. So there is something called federated architecture. There is something called distributed privacy systems. So they are a little bit futuristic, but we are going to use distributed privacy where we need not really take out the information from the source. If Patanjali data is having certain important things with respect to patients, now we're going to touch that. Whatever is shareable, whatever can be shared within the scientific communities, only that will be tapped in, and we're going to use that. So again, I'm reiterating that Ayush's job is not to centralize anything and sit on that. No, that's not how it's going to work. Its basic job is to connect, ensure that this kind of digital tools are available. So that's a job.
Okay. Thank you very much. So,
Thank you, Dr. Sakeji. Thank you very much. Our next speaker, Dr. Vijay Kumar Thakur ji is from Edinburgh. He is the head of the Bio Refining and Advanced Materials Research Center at SRUC Edinburgh, UK, and is a principal supervisor in the School of Biological Sciences at the University of Edinburgh, UK. His expertise includes the synthesis and processing of sustainable and functional polymer materials. He has been a co-PI on several projects and published 400 journal articles and 55 books on many more. He has 2 patents and is also member of the editorial board of several reputed journals. I welcome Dr. Vijay Kumar Thakur ji. Please continue your lecture. Dr. Thakur.
Good afternoon and namaskaram to everyone. Thank you, sir, for the nice introduction. I would like to begin by expressing my gratitude to Shraddhe Baba Ramdev ji and Acharya ji for making this knowledge of Ayurveda and Ayurvedic medicine popular all over the world. Also the organizing committee for extending me the invitation to present some of the work we are doing at SRUC. So a little bit about SRUC. SRUC is a premier higher education institute that sits within the University of Edinburgh. We are primarily focusing on agriculture, food and veterinary related research in the UK. Recently we have started a vet medical school that is third in Scotland in the last 200 years. And we have 6 different campuses across UK, also 25 consultancy offices. catering the needs of the farmers. We work very closely with the farmer community across Sri Lanka, and amid this pandemic, we were able to generate more than 50 million research funding from different industries related to agriculture. Just talking about one of the previous speaker talk about the technology, how it is going to change the world. So recently Samsung has come out with a small picture envisioning how the world will change in next 100 years. So it has been shown that we will have cities with mega skyscraper because of the change in the technology. Also, we will have underwater metropolises with breathable atmosphere. This is really amazing. Then drones will be used for personal transportation, and we have seen currently these are being used for more of destructive purposes, especially in war zones. But in the near future, they will be used for mankind's health. And then most importantly, medical pods will also diagnose and treat illness from the comfort of the home. But one most important thing that is going parallelly with the advancement in materials is thrust for sustainability. In everyday life, we hear about sustainability, sustainable materials, or net zero emission that most of the country want to achieve by 2050. So what really sustainability is? According to World Commission on Environment and Development, anything that meets the needs of today's generation without compromising the needs of tomorrow's generation comes under the category of sustainability. And luckily, anything green we see around us comes under the category of sustainability. And luckily in Indian culture, everything in one way or another has been linked to some of holy way. I will talk to this in later on. So why there is so much need of combining technology with sustainability, with health, the answer lies in front of us. By 2050, we will have significant population that won't have access to the food or water, energy, or even our atmosphere will be really polluted. As we know, in winter, Delhi becomes highly polluted. Also, currently, our oceans, they contain more microplastic than feces. So it is high time that we look back several hundred years back, where our old generation, they have been using or learning from nature, and somehow they have connected it with one way or another in more of a pious way. And if I talk from more of like scientific perspective, couple of years back, there was an article in a famous magazine, Science, where they throw light on the importance of fasting, how fasting can improve our health. There is a lot of biology as well as another science behind it. I won't go into more detail, but just to give you a flavor that what most of the advanced nations currently are trying to explore has already been linked in Indian culture, whether it is Ayurveda or another medicine, but it is there. We just need to find it to make it more of a acceptable thing in a scientific way all over the world. And at SRUC, we are primarily focusing on making use of bioprocessing and biorefining to develop new sustainable materials and manufacturing for the future, so we can empower the local community as well as we can find better ways to use different materials. So we have been making use of different types of natural materials coming across the way. Also, we have been making use of different waste streams that are generally considered waste, developing more of biofuel, bioplastic that are a traditional alternative of synthetic chemicals and so on, and the materials derived thereof. In terms of sustainable materials, we have been working a lot on the materials generally we see around us, cellulose for more of biomedical application, gum, pectin, chitin, one of the most important material used in biomedical field. I will talk about it shortly. Alginate and starch, that is obviously a essential part of our food, and we are mainly interested in enhancing their properties at nanoscale. So we are mainly focusing on their engineering both from biomimetic as well as synthetic way. Interestingly, all the natural materials we see around us, they have really very good surface characteristic that can be really engineered. One of the area is hydrogels, where we have been using them for a lot of biomedical applications. We have been learning from the nature as well, using advanced manufacturing technology such as 3D printing and 4D printing of these material, so that we can directly target a particular molecule or material depending upon the need of the patient. And I will talk a little bit more about turmeric again. Coming back to our old culture, turmeric is an essential part of Southeast Asia's food. It's in one way or another way, we every day use turmeric. But turmeric has really huge scientific potential. It has a compound, or you can say a molecule within it called curcumin, that has enormous biomedical properties, antibiotic, anti-inflammatory, and so on. So we have been extensively working on exploring the use of curcumin for biomedical application, for cancer therapy and dressings as well. So we did develop different types of hydrogels. Here you can see how these curcumin molecules impact more of like these species, but we also want to try it in near future once we have the approval of these molecules. So we have been first extracting this curcumin from these different types of species and then trying to interact, see how these molecules behave when we embed them in a particular hydrogel system. Again, whether we are talking a metathesis or another type of health related issues. And one of the interesting thing to look into this material is it has immense properties. You can use it to inhibit bacterial growth. You can use it for wound caring with the proper combination of molecule. For example, we have been using dextran, again, that is a biopolymer. We are trying to embed them together. Then chitin nanoparticle, again, another important biomedical material that has a huge potential for anti-cancerous applications. Here in this article, we have summarized the use of curcumin for biomedical applications. If you are, I am cautious about time, so if you are more interested in looking into use of curcumin or say turmeric or in Indian term haldi, you are most welcome to go through this article. We have made it open access and you can see the huge potential of turmeric for biomedical application. Similarly, we have embedded it with another molecule, cellulose. As I mentioned, it is a biomolecule, and we prepare a dressing material for burning application. In US alone, lots of people die because of burning. They really don't die because of burning, but when the dressings are used by medical doctors, the pain is so much that the patient goes into such a trauma, so they are not able to bear it. So we are trying to develop more of self-disintegrable dressings that you don't need to remove. You put some water on a material made up of curcumin and another biopolymer, so that you avoid that painful process when you are using it for burning application. Similarly, we have been looking into bacterial cellulose. One of the problem we have come across with the traditional cellulosic nanomaterial, we have developed this, sometimes it contains impurities, so these materials are having some biocompatible issues as well. So we are developing lot of cell factories to develop new bacterial cellulose, that is the most purest form of cellulose. Only issue currently is we are not able to produce it at a large scale, and it's not only our group, but different groups across the world, they are not able to produce it at a large scale, and we are working closely with another colleagues how we can scale up, but because if we can scale it up, it will solve some of the biomedical problems. Similarly, we are making use of biopolymer that we have been traditionally used in Indian culture or in another civilization as well. One of them is cartilage. As you know, when people start getting old, they have started pains in knees and so on, and the cartilage damage is the prime reason behind it. So we are developing new material that are having similar properties that can address the cartilage issues. So we have been playing a lot with hyaluronic acid and cellulose nano fiber films. Especially, we are trying to develop new cellulose nanocrystal so that we maintain the mechanical strength of the molecules as a whole. And another area we are looking into, again, these biomolecules, we are trying to make them used for osseointegration applications, especially for hip replacement and other application. What happens when there is a replacement of hips or knees? Most of the time, after 5 years or 10 years, the body starts rejecting these outer implants because we cannot really replicate the characteristics of human body. And one of the problem is incompatibility between the implants as well as the human body. So we have been developing new type of bio-based coating where it gives more of like significant biocompatibility. So we did develop different types of coatings based upon the molecules, natural molecules we see around us. One of them has been on cellulose ester synthesized by the resveratrol. This is an interesting molecule with lot of antimicrobial properties. It has shown good antimicrobial as well in vitro characteristics. Similarly, we learned from silk. So we made use of silk-derived sericin molecule to immobilize again on cellulose ester membrane for biomedical application. Currently, trials are going on in Eastern European country together with medical doctors who are seeing how long these material can sustain in the body or how effective they are without the coatings. Similarly, we are looking into another molecule, chitin, as we have seen earlier. So chitin is another important molecule. Some of us might not be aware, but most of the stitches that doctor uses during biomedical application, they are made up of mainly chitin fiber because it has really very good promising antimicrobial properties. So we have been developing different types of material from chitin and chitosan, especially nanogel and nanofibers for both biomedical and another applications. So we are primarily interested in use of chitosan based molecule, especially at nanoscale for cancer therapeutic application because two molecules, PT and DTX, they are really very less soluble in solvent as well as they are not really biocompatible. So even if you give these doses to patient, they are not really able to digest them or they really did not have a significant impact on the recovery of the health. So we have been trying to develop more of nano-based molecule from chitosan nanoparticle and then trying to incorporate them together with PT and DTX for better progress, and preliminary study has shown very good results. And here you can see we have been looking into different types of material because we want to get a molecule with optimum properties. Similarly, we are looking its application for cancer imaging and forensic. That is also another interesting area. We are working with colleagues with electronics with the University of Cambridge on see how these biomolecules can be really engineered for these cancer imaging applications. Then here is one another different application of chitosan nanoparticle where it can provide a protection against degradation in the stomach, and it enhances the absorption of PTX and DTX because of these nanomaterial interaction with these molecules. Also, amid this pandemic, we have started looking into embedment of different antimicrobial materials, say TiO2 nanoparticle that has been used for a number of solar-related applications, but we have been looking them for more of bio-based coating. So we have been making use of these biopolymer, chitosan and cellulose along with starch for antimicrobial application because most of the time surfaces are also a big carrier for these different type of viruses or another type of pandemic diseases. So we have been developing different types of masks, textile cloth, especially we are looking into smart textile where we are embedding these nanomaterial, and also we are embedding new molecules where whenever any virus or another bacterial molecules come in contact with these, they will give you a signal, and you can know that you have been come in touch with different types of these viruses. We have been also looking into use of natural oils as a antimicrobial. So some of them have been tung oil as well as another oils that have been traditionally used in Indian culture for a lot of antimicrobial applications. In addition, we are also looking into different biomolecules, say fluoroquinolone, and also we are primarily focusing on making the use of green chemistry, because we always try to make minimum use of these hazardous chemical, as well as we keep in mind the this net zero emission target. Otherwise, what will happen if we really don't control the way we do the synthesis? It is envisioned that in near future the coastal countries will no longer exist, because the temperature will keep increasing if we keep focusing on more of petrochemical based resources. Similarly, we are developing new smart technologies, especially for health monitoring. We are making use of both sustainable materials, especially cellulose-derived nanomaterials, for more of like cell-powered sensors, where we can get idea about what's going on within the body, or we can have some sort of sensor where we can keep them in the pocket, or if something happens to say older people at home, we can get a signal on our phone that something is wrong going within the body. So this is something at very early stage, but we have been able to develop some sort of smart material at lab scale, where they are showing really self-powered properties for biomedical application. Also, this work was an extension of the work funded by BAE System, one of the largest manufacturer of weapons in the world. They wanted to come by 2050 by a self-powered helicopter, and learning from that, we also tried to this in biomedical field, and interestingly, the results are very promising. Also, again, coming back to biomedical, we are exploring the use of advanced engineering technology to develop new scaffold that can be used for biomedical application, or even they can be used for filtration application. Here we have made use of a bio-based polymer called polybutyl succinate. Again, it can be derived from bio-based sources. Graphene nanoplatelet synthesized by a technique developed at University of Cambridge, then nanofibrillary cellulose from the plants we see around us, and we were able to develop very nice structure that have again shown very good promising result for biomedical applications. Hemp is another important area we are looking into it. Hemp is considered generally more of a synonym of marijuana, but it is significantly different, and it has a really huge health benefits. So I think it's the only plant all over the world whose every part can be used for biomedical applications. Here, the hemp seed, they have really huge value for biomedical application. Even the leftover part that is considered as a waste has a huge potential for another application. For example, we have been able to develop new nanocellulose foam from hemp waste straw. That is again a waste, and farmers in UK, they have problem of disposing it. And we were able to replace traditional synthetic foam, or you can see the cushions we use in home. They are mainly made up of polystyrene or another synthetic polymer that are obviously health hazardous and ultimately contribute to enhancement in net zero emission issues. So we were able to develop a new technology where we replaced the total polystyrene or polyurethane foams with these hemp-based foams, and we are currently working with a couple of industries in UK to bring this product into market. And just to summarize, we have been working different types of biomolecules and biomaterials, and we are always making use of green chemistry principles to develop different sustainable materials. Obviously, I cannot cover all these in this short time. So if you are more interested in looking into the potential of different biopolymers that has been an indispensable part of our culture, you are more than welcome to look into this article that we have published couple of years back in Chemical Reviews, where we have summarized lots of applications starting from biomedical to electronics to packaging. That is also another issue, as well as automotive and optical applications. Similarly, we are making use of 4D printing. We have been using it for more of biomedical application. We recently funded this project. It was funded by Government of India as well as UK government on 4D printing of graphene with the vegetable oil. We made use of soybean, but that is obviously again a food ingredient, but we were able to convert it into biopolymer, and then we printed some interesting molecules. And interestingly, I want to talk about this dairy that is a foremost living source of farmers in India as well as around the world. So we have been recently funded by UK government to establish a digital dairy chain in UK. So we are the only institute to be funded by government of UK, and one of my colleague Richard, he is leading this grant. Idea is to how we can empower the life of poor farmers and people, and also make it more of a advanced technology, similar to another technologies that are being used for a number of applications. So we have been working closely to develop different types of material. One such example is we have made use of eugenol. Eugenol, I think you might not aware, but eugenol is something made from an oil called eugenol, and it is nothing but a oil that we call clove, or I'm not sure exact term, but we call it in Hindi long. So it has really very good antimicrobial properties. We engineered it, and we were able to develop new antimicrobial coating for leather or another sort of materials. Similarly, we are valorizing different bioproducts from dairy industry for food grade application as well as for energy applications. We are developing different oligosaccharide molecules for food applications. And recently we have been able to transform this cow manure that is obviously considered as a waste, and if you talk about manure, then people will always think, oh, it's not something novel. But one of our PhD student, together with a postdoc, we have been able to develop new type of sustainable material, both bioplastic as well as nanomaterial from cow manure. And I think this initiative can also be taken in India to empower the poor farmer as well as contribute to a greener earth. Then, to summarize, for a sustainable world, we must always use what we have not used yet, and we should make sure of food security to everyone, and we should also unlock the potential of different types of materials found in the earth or oceans as well. And last but not the least, there are many people who have contributed to the journey so far, so it won't be fair to name few and leave others. So thanks to everyone who have contributed to the journey so far, and also the government as well as industries who have generously supported our work. And thank you very much for your presence hearing. I will be happy to answer your question. And if you have any question, you can also drop an email to the email addresses shown here. And thank you very much again, and namaskaram.
Thank you, Dr. Thakur. To summarize the talk was on the application of nanocellulose in the formation of hydrogels with antibacterial, anti-wound for wound healing, and for taking care of the knee joint as a filler, as well as the use of cellulose acetate as a biodegradable material with biomedical applications. Also, it involved the use of chitin and chitosan as a nanogel for cancer theranostics, bioimaging, antimicrobial properties, and smart material with wearable technologies. Other than that, the talk was also on the bioprinting and application of hemp and wood-based nanocellulose foam for filling up as a filler for pillows and other applications. So now the talk is open for discussion. Any questions?
First of all, thank you, sir, for such an informative talk. My question is, will we be able to overcome the limitation of curcumin? It's hydrophobic in nature. My question is, curcumin is hydrophobic in nature. So when we are preparing hydrogels of that, will we be able to overcome this limitation?
Yes, this is very interesting question, and to address this, we are trying to transform curcumin into nanomolecules. So we are currently developing more of nanoparticles because its solubility in water is a big challenge, as you have mentioned. But whenever we embed it in hydrogel, it somehow we get over it, but still we cannot completely address this issue. So we believe that transformation into nanomolecules may address this problem, but still we are working on this.
Any other question? So thank you very much, Dr. Thakur, for the interesting talk.
Thank you, Dr. Thakur. Our third speaker, Dr. Manali Joshi. She is talking about computational pipeline to enable network pharmacology. Dr. Manali Joshi is an assistant professor, Bioinformatics Center, SP Pune University, with a master's in biotechnology from IIT Bombay and PhD in computational biochemistry. Dr. Joshi is instrumental in the research of molecular modeling and simulations. Her research interests include computational approaches to drug design and understanding the structure, function, inhibition paradigm of proteins. Dr. Manali.
Hello. Thank you so much for the kind introduction, and before I begin, I must say a big thank you to the organizers for this excellent opportunity for giving a talk. So today I'll be talking about a specific technique which is known as network pharmacology and how we are trying to automate this. मेरा टॉक मैं थोड़ा हिंदी और थोड़ा इंग्लिश में देना चाहूँगी. इफ़ आई मेक एनी मिस्टेक्स इन हिंदी जस्ट पार्डन मी, लेकिन ये आप लोगों के लिए है बिकॉज दिस टेक्निक इज समथिंग दैट विल कंप्लीट द एविडेंस बेस ऑफ आयुर्वेद एज वी हैव बीन हियरिंग फ्रॉम डे वन. So I'll begin by talking a little bit about my university and my center. So Pune University is in Maharashtra state, and very proud to say that in world ranking we are in 540 to 550 rank. Times Higher Education has given us a rank of 2, and we are also NAC accredited at A+. Our department, which is the Bioinformatics Center, was founded in 1987 by Professor Ashok Kolaskar, who is a stalwart in bioinformatics, one of the pioneers of bioinformatics in our country. We have a Center of Excellence from DBT from 2002 onwards. We also had a mighty Center of Excellence, which is Ministry of Communication and Information Technology from 2007 to 13. So at our center, we basically run 2 programs, which is MSc and PhD in bioinformatics. And please do visit our website and read more about us. तो हमारे सेंटर में हम ज़्यादातर क्या करते हैं कि वी डेवलप कंप्यूटेशनल टूल्स टू ट्राई एंड अंडरस्टैंड बायोलॉजिकल प्रॉब्लम्स. मतलब हम किसी भी बायोलॉजिकल प्रॉब्लम के लिए कंप्यूटर्स कैसे अच्छे से इस्तेमाल कर सकते हैं, या तो हम उसका डेटाबेस बनाएं या कुछ एल्गोरिदम बनाएं जिससे हम उसका एनालिसिस ज़्यादा अच्छी तरह से कर सकें. सो टुवर्ड्स दैट टुडे, लाइक आई सेड, आई विल टॉक अबाउट अ मेथड कॉल्ड नेटवर्क फार्माकोलॉजी. So before I begin about this method, I'll just like to give you a brief introduction about modern medicines and why we say that there is so much evidence. और आयुर्वेद में क्या evidence हमको और चाहिए, हम ये कैसे complete कर सकते हैं. So if we just take an example of a small molecule that is given on the slide, can anyone recognize what that is? आप कोई पहचान सकते हैं कौन सा small molecule है? वो छोटी सी दवाई की जो तस्वीर है उसमें लिखा है, शायद आप नहीं देख सकते, ये एस्पिरिन है. हम सब ने एस्पिरिन कभी ना कभी तो लिया ही होगा, सुना भी होगा, राइट? नाउ अब मॉडर्न मेडिसिन का ऐसे है कि एस्पिरिन हियर, एस्पिरिन इन चेन्नई, एस्पिरिन टेकन एनीवेयर will have the exact same chemical in that exact same amount. Aspirin 100 milligram is this same molecule whether you take it here or anywhere else. Okay, so this is what is very different about modern medicine. We also know that we can use aspirin for pain, fever, prevention of clots as a blood thinner. And why we can use that? एक लाल रंग की लाइन दिखाई है मैंने. तो दो प्रोटीन्स हैं हमारे बॉडी में. I am sure you all know what are proteins. So proteins are very important macromolecules जो बहुत सारे functions करते हैं. कभी वो enzyme की तरह होते हैं जो reactions करते हैं, कभी वो receptor की तरह होते हैं जो cell signaling में important होते हैं. So proteins are very very important and majority of the FDA approved drugs actually target proteins. Okay, तो हम जानते हैं कि एस्पिरिन क्या करता है, वो साइक्लोऑक्सीजनेज 1 और 2 नाम के प्रोटीन को जाके इनहिबिट करता है. एंड बिकॉज ऑफ दैट इनहिबिशन वी गेट रिलीफ फ्रॉम पेन एंड इन्फ्लेमेशन. हम ये भी जानते हैं कि दिस पर्टिकुलर स्मॉल मॉलिक्यूल इज रैपिडली एब्जॉर्ब्ड इन स्टमक एंड द स्मॉल इंटेस्टाइन. उसका प्राइमरी मेटाबोलाइट क्या है, मतलब हमने अगर एस्पिरिन लिया तो केमिकल जो लिवर में तैयार होता है, कहीं भी हो सकता है, वो क्या है वो भी हम जानते हैं. इस मॉलिक्यूल का बॉडी में हाफ लाइफ क्या है. सो फार्माकोकाइनेटिक्स ये एक जो स्ट्रीम आप सुन चुके होंगे, शायद पढ़ भी रहे होंगे, जिसका मतलब कि हमारे शरीर में इस केमिकल का क्या हो रहा है. Okay, so for this molecule, for instance, we know that the half life is 13 to 19 minutes. We also know the toxic level of this molecule, which is 1 gram per kg. Now, इतना ही काफी नहीं है. Now, actually to get FDA approval, okay, इतना काफी है कि ये इफेक्टिव है, इसका डोज हमको पता है, ये शरीर में क्या करता है. लेकिन एक्चुअली ये कॉक्स 1 और 2 में जाके कैसे इनहिबिशन कर रहा है, ये भी तो हमको जानना जरूरी है. बिकॉज द वे साइंस हैज इमर्ज, वी वांट टू नो हाउ थिंग्स वर्क, राइट. वी आर ऑल क्यूरियस, ये कैसे काम कर रहा है, हमको ये पता करना है. तो दिस वॉज अ नोबेल प्राइज विनिंग डिस्कवरी इन 1984, जो मैंने राइट साइड में एक तस्वीर दिखाई है. इट्स लाइक अ प्रोटीन, और ये प्रोटीन है कॉक्स 1 जो प्लेटलेट्स में पाया जाता है. सो लेफ्ट साइड में जो दिखाया है वहां पे उसका नेचुरल जो सबस्ट्रेट है एराकिडोनिक एसिड, वो जाके उस कैविटी में बाइंड करता है. नाउ व्हेन यू गिव एस्पिरिन, ओके, एस्पिरिन विच इज दिस मॉलिक्यूल, उसका एसिटाइल ग्रुप जाके उस कैविटी में रिएक्ट करता है और वो ब्लॉक कर देता है उस कैविटी को. Okay, so no longer this molecule, this protein can function by its normal way. So it can no longer form prostaglandins, it can no longer lead to inflammation and let's say pain. So we know this very well, and this was a Nobel Prize winning discovery. So the beauty about modern medicine is that we know a lot of things, okay. But one nice thing about aspirin, okay, ये इसकी एक स्टोरी है जो बहुत अच्छी तरह से वेद प्रिया मैडम ने बताया कि उन्होंने एक हर्बल एनसाइक्लोपीडिया तैयार किया है जिसमें अलग अलग देशों की प्राचीन संस्कृति के में जो औषधि थी वो भी लिखा है. सो इन एंशंट इजिप्ट, ओके, पीपल यूज टू यूज जो उसका बार्क होता है विलो ट्री का, उसकी पाउडर यूज करते थे पेन के लिए. तो ये मैं स्टोरी आपको बता रही हूँ कि एस्पिरिन बना कैसे, ओके. तो शायद आप ये टाइमलाइन देख सकते हैं कि सो इट वाज 1934 बिफोर क्राइस्ट, सो ऑलमोस्ट 3000 ईयर्स प्लस पहले लोग जानते थे कि आई कैन यूज दिस बार्क फॉर पेन, बट नोबडी न्यू मोर अबाउट इट. Okay, so for many things in herbal medicine, we are currently at that stage. We know it works, but we don't know how it works, which is what we want to know. Okay, तो एक American scientist थे जो not scientist, he was an American who was into medicine. He took this information, went back, and in 1763, मतलब कितने सालों बाद, he found the primary ingredient called salicin. लेकिन प्योर फॉर्म में सैलिसिन में अगर ले, इफ यू टेक इट, इट हैड अ लॉट ऑफ गैस्ट्राइटिस इश्यूज. सो दैट वाज मॉडिफाइड इनटू सैलिसिलिक एसिड एंड लेटर एसिटिल सैलिसिलिक एसिड, जो एस्पिरिन के नाम से फिर अप्रूव हुआ और बहुत पॉपुलर हुआ. सो दिस इज द स्टोरी एंड दिस ड्रॉ अ लॉट ऑफ पैरेलल्स यू कैन ड्रॉ फ्रॉम आयुर्वेद. Because the origin of this particular wonder drug is actually the bark of a tree, but we have made some chemical modifications to make it work better and to relieve it from some of the side effects that the original molecule had. Okay, तो हम ये चाहते हैं कि आयुर्वेद में जो formulations है हम उसको भी ऐसे मतलब evidence निकाल सकते हैं क्या, how does it work? Okay. So as opposed to modern medicine which typically is one chemical, एक chemical है जैसे आपने देखा. Now, अगर आप हर्बल मेडिसिन देखें जो आयुर्वेद में बहुत बार प्रेस्क्राइब किया जाता है, वो एक मिक्सचर होता है. कभी कभी एक फ्रूट होता है, जैसे हरितकी राइट साइड में दिखाया है, टर्मिनालिया चेबुला, इट्स वन फ्रूट, और इट कैन बी कॉम्प्लेक्स लाइक अ मिक्सचर ऑफ थ्री विच इज त्रिफला, राइट. एंड नॉट ओनली दैट, अभी वो एक फ्रूट जैसे हरितकी, और आप लोग शायद मुझसे ज्यादा अच्छे से जानते हैं क्योंकि आयुर्वेद तो मैंने मतलब ऐसे सीखा नहीं है, बट यू कैन गिव इट विद डिफरेंट मीडियम्स, राइट. कभी हम दूध के साथ, कभी घी के साथ, कभी सॉल्ट के साथ, अलग अलग डिसीजेस के लिए दिया जाता है. बट हाउ डज इट वर्क, ये हमको नहीं पता. दिस इज द पजल, पार्ट ऑफ द पजल दैट इज मिसिंग, विच फ्रॉम डे वन वी हैव बीन सेइंग कि वी वांट मोर एविडेंस टू अंडरस्टैंड. अभी द कॉम्प्लिकेशन इन हर्बल मेडिसिन्स इज दैट इट्स नॉट वन मॉलिक्यूल. Okay, so even if you consider this one fruit of haritaki, which is Terminalia chebula, it has more than 70 phytoconstituents, and that is only that number of phytoconstituents जो हम देख सकते हैं through experiments. Experiments may have limitations, which I'll talk about in a bit. फिर एक को छोड़ो, when we talk about a formulation with 2 or 3 things, आप सोचो उसमें how many phytochemicals are there. So now that has increased our complexity a lot. That one molecule of aspirin which I had shown, which interacts with one protein, वो सोचो आप इस साइड, और इस साइड हमारे पास ऐसा कुछ है जिसमें बहुत सारे chemicals है. So the situation is much more complex. So we need more and more intelligent methods to try and understand how it works. It works, we know it does, but right now we don't have the methods or the tools to understand how it works. Okay, so let me walk you through this method called network pharmacology. So what is a network? I am sure you all understand network क्या है. आपको मोबाइल नेटवर्क भी पता है, यहाँ से टावर में आपका जाता है, टावर से किसी और को, ऐसे ये एक नेटवर्क है. तो हम एक नेटवर्क बनाएंगे. What is pharmacology? Pharmacology मतलब कोई भी केमिकल और उसका इफेक्ट ऑन द बॉडी. So what we are trying to do is, ये बहुत सारे इंग्रेडिएंट्स है, उसका हम नेटवर्क फार्माकोलॉजी से इस्तेमाल करेंगे टू अंडरस्टैंड द मैकेनिज्म. सो दिस वाज अ मेथड दैट द लैब ऑफ प्रोफेसर भूषण पटवर्धन, he had used for the first time and reported for the first time to understand how ingredients in Triphala might be working. Okay, so this was published in 2015. It's their data, there are collaborators. तो how do we make this network? Okay, तो इस नेटवर्क के 3 steps हैं. दिखते आसान है लेकिन है नहीं. There are a lot of limitations which also I'll discuss. So the first is identification of bioactives. तो ये जो आप नेटवर्क देख रहे हैं उसमें 3 हब है. I don't think I have a pointer here. So एक जो बड़ा सा सर्कल है उसके बीच में एक पॉइंट है that represents one let's say botanical. Yes?
You can use the mouse pointer.
Yeah, it's somehow I don't see it from the pad, but it's okay. I'll explain it with the figure. So वो that circle that you see उसके बीच में जो है that represents one botanical. Let's say for instance आंवला, और उसके बाजू जो आपको yes it's okay और उसके बाजू जो आपको dots दिख रहे हैं, those are basically the phytochemicals in that ingredient. Okay, so आपको you see 3 of those kind. One is a big circle with one center, and then you see 2 small parts on the upper left and lower right side. Okay, तो ये 3 जो है, ये 3 botanicals है जो त्रिफला में है, और उसके phytoconstituents वो छोटे छोटे spheres है. अभी कुछ ग्रे में है, कुछ ब्लैक में है. I'll explain. The ones that are in black are the ones that are known to work experimentally. Okay, with their targets. तो हमने क्या किया, पहले हमने 3 बोटानिकल्स को रखा, उसमें कौन कौन से इंग्रेडिएंट्स है वो फाइंड किए. कैसे फाइंड करेंगे, आई विल आल्सो एक्सप्लेन दैट. उसके बाद द नेक्स्ट स्टेप इज लिंकिंग बायो एक्टिव्स टू टारगेट्स. अभी हमको पता है कि इन आंवला देयर आर सो मेनी स्मॉल मॉलिक्यूल्स. नाउ आई वांट टू लिंक दो स्मॉल मॉलिक्यूल्स विद प्रोटीन्स. सो व्हाट यू सी इन द सेंटर, जो बीच में गोल दिख रहा है, जो डायमंड शेप्ड है, वो उसके टारगेट्स है. तो हम ऐसा एक नेटवर्क बना सकते हैं कि ये 3 इंग्रेडिएंट्स जो है उसमें कौन से केमिकल्स एंड हाउ दोस केमिकल्स मैप टू डिफरेंट टारगेट्स, एंड देन वी कैन लिंक टारगेट्स टू डिसीजेस यूजिंग कंप्यूटेशनल टूल्स. नाउ ऑन द टॉप राइट साइड you can see a hand, which means currently this method is used quite a lot across India after this seminal paper. Lot of people are using this to try and understand, but it's not automated. हमको ये manually करना पड़ता है हर step. मतलब क्या? So let's go step by step, and I'll try to explain how at the Bioinformatics Center we are trying to enable this by automating this entire process. So just at a click of a button you will get this network. So let me explain how. So जैसे कि मैंने कहा, first step is identifying the bioactives in a formulation. तो हमारे पास एक formulation है, let's assume it's just a fruit for instance, one fruit, हरितकी. तो हम कैसे निकाल सकते हैं उसमें क्या bioactives है, वो what are the phytochemicals in that fruit? Three ways. Either you do experiments yourself, तो आप इसमें से कुछ experiments कीजिए जो बहुत commonly use करते हैं, HPLC, MS, NMR, उससे आप metabolites निकालिए. लेकिन उसका एक, now in every slide I'll have something in blue which are the limitations that one faces when doing that. One is that, and I think there was a question asked about cannabis by someone, that I have cannabis growing, can I just eat it? So very beautifully answered by the speaker is, a plant, what chemical metabolites it will have, secondary metabolites, depends on the condition in which it is growing. Unlike modern medicine where when you say aspirin, it's always aspirin that exact amount, we don't know what variables the plant faces when it is growing, what conditions it has. So I don't know if this standardization is available, if not it must be done. That haritaki grown, let's say here in Haridwar, or haritaki grown in South India versus anywhere else, uske fruit mein plus minus standard deviations में सारे phytochemicals कैसे मिलेंगे आपको? Because it will never be the same. You will never get an identical amount of, let's say, XYZ phytochemicals if you take different fruits. So this is the first thing that needs to be standardized. The second is, जब हम experiments कर रहे, all experiments have limitations. When I do HPLC or when I do something else, the type of solvent I'm using, I'll be limited by that. I will only be able to extract what is extractable in that solvent. So I may miss so many metabolites. Okay? तो ये हमको ध्यान में रखना पड़ेगा कि whatever I do will have its own limitations. I may actually miss उसमें जो important small molecule है, हो सकता I will miss that. The second is that I don't have the means to do the experiments myself. मैं देखूंगी कि किसने क्या publish किया and I'll review literature. This is also a popular approach because many people are interested in this, so many papers are published. So people do a literature review and find out, अच्छा 10 लोगों ने already MS किया है, तो I'll compile all the chemicals what they have reported. Again that is limited by what they have reported and you never know उन्होंने क्या miss किया है. Okay? Quickly I'll tell you bracket में क्या number है in a bit. The third thing is you rely on databases. So there are many databases that have actually compiled information from literature, from experiments, and one of the Indian let's say initiatives is a database called IMPAT, which is developed by Mathematical Institute Chennai. So it is a compendium of Indian medicinal plants and the small molecules that are there. So I'll show you a screenshot just to give you an idea. And there are several other databases also which may not be Indian but they do have Indian plant parts and their metabolites listed. जैसे knapsack है, natural product activity species source है, there are African natural products, Vietnamese herbal databases. There are many databases actually which do include Indian plants as well. Now I'll tell you what is there in the bracket. So one of my students was compiling a list of phytochemicals for haritaki fruit. कि हरितकी फ्रूट में क्या क्या है, what is the information out there. So literature review करके उसको सिर्फ 32 phytochemicals मिले, whatever papers. Of course she could be limited by what she searched. When she looked at different databases, अब आप देखिए हर database में अलग अलग number है, तो हर database में जाके finally जब she made a like a redundant list, उसको around कुछ 70 phytochemicals मिले. So this when you do manually is very cumbersome. Okay. Moreover, मेरा ये question है, हमेशा रहता है कि we are always focusing on small molecules. There are so many other things in plant parts, whether be it bark, be it fruit, it could be nanomaterial, it could be so many other things that we are missing in this methodology. Okay, that we have to remember. Another thing we have to remember, ठीक है, let's say I started with haritaki, I found some 70 phytochemicals. I made a list, but do we know the amounts and whether they are really absorbed and go into the blood? ये इंफॉर्मेशन सारे फाइटोकेमिकल्स के लिए नहीं है. ओके, सो दीज आर द लिमिटेशंस राइट नाउ, बट ऑफ कोर्स whatever we have accessible, उसके लिए हम क्या कर सकते हैं? अभी हमारे पास हरितकी फ्रूट है, हम उसका फाइटोकेमिकल लिस्ट निकाल सकते हैं. दैट्स द फर्स्ट स्टेप which right now you have to do manually, which I will show in a bit how we are automating. So this is just a screenshot of IMPACT, and you can see that I have typed Terminalia chabula there, and then you have different plant parts mentioned like bark, fruit. So you just have to go and download in appropriate format the phytochemicals you are interested in. So each database like this, you can go there and you can download the plant part and the ingredients of your interest. So now let's say I am using this example of haritaki fruit only. So now I know that in haritaki fruit there are some 70 phytochemicals. Now I want to know how it works, right? So I want to know ये जो chemicals है उसके कोई protein partners है क्या? Are there proteins in our body which these phytochemicals can interact with, and hence I can explain this is how it works. Okay, so there are several very useful databases out there, and I must mention one of them is called PubChem. तो आप जरूर PubChem ये database देखिए if you are not aware of it. So PubChem has 304 million bioactivities. It's a very nice resource compiled by NCBI USA. So what are these bioactivities? So a lot of let's say 2 decades ago, high throughput screening was a big thing. मतलब using machines or robotics to do assays. Okay, so people, and this was primarily of course for Western medicine. So if I have a target of interest, let's say some kinase or some protein, then what I'll do is I'll use a robotic setup to screen millions of molecules by the computer, क्योंकि हम हाथ से तो नहीं कर सकते. So all of this is compiled. So whatever people have done across the world, luckily it's open source, they have recorded. तो ये सारा compile किया हुआ है, plus छोटे scale पे जिसने जो assay किया है वो compile किया है. तो हमारे पास PubChem में क्या है? We have assays which link small molecules to different targets, not just in human but also in other organisms. So this is a very useful resource. अभी हमारे पास एक साइड में क्या था? केमिकल्स थे हरितकी के. अभी हमारे पास कौन से रिसोर्सेज है? वो रिसोर्सेज जिसमें ऑलरेडी एसेज अवेलेबल है. सो नाउ इट्स अ मैटर ऑफ फाइंडिंग आउट व्हाट केमिकल डू आई हैव, हैज द एसे ऑलरेडी बीन डन, सो डू आई नो द टारगेट टू इट. दिस इज डायरेक्ट एविडेंस. किसी ने एसे में दिखाया है कि ये केमिकल इस प्रोटीन पे काम कर रहा है. सो इट्स नॉट हाइपोथेटिकल. Okay. Similarly, there are some other useful databases like BindingDB, जिसमें भी 6 लाख से ज़्यादा binding data available है. BioGrid है, PDP है, which is more like a structural data, which gives you even more confidence as to the association. And of course, there's drug bank. So interestingly, many of the phytochemicals like ellagic acid or whatever you can find in these small molecules, some of them are undergoing clinical trials. So some of the targets you may actually find in a database which actually has FDA approved drugs. So these are all known associations, मतलब direct evidence जो assay से किया है किसी ने. Okay? लेकिन इसमें फिर भी एक लिमिटेशन है कि जैसे मैंने बताया 70 अगर मेरे पास फाइटोकेमिकल्स है, क्या ऑल ऑफ देम डू दे गो टू द ब्लड एट इनफ अमाउंट्स टू इनहिबिट और एक्टिवेट दैट प्रोटीन? दैट वी डोंट नो येट. ओके, सो विच इंग्रेडिएंट इज अब्सॉर्ब्ड एट विच कॉन्सेंट्रेशन वी डोंट नो. So this is just one screenshot from PubChem, and this is for chebulagic acid, which is one of the phytochemicals you can find in haritaki fruit. So you can see वहाँ पे लिखा आता है कि ये जो chebulagic acid है, it is active against and this is SARS coronavirus 2 protein, and it also gives binding constant. So this is direct evidence that somebody by using assays have shown that this compound works for this target. Okay, so the databases I have listed here are all those that have direct experimental evidence. Apart from that, you can predict. मैं computational tool से predict कर सकती हूँ कि अगर मेरे पास ये chemical है उसका target क्या हो सकता है. So as of now, I personally don't recommend including this type of method in the network pharmacology, because then it becomes a bit hypothetical, because we don't know. These are predicted associations, but people can use this. So we have phytochemicals that we identified from the fruit. We have some targets from direct assays from these databases. Now I want to know ये targets कौन से disease में associated है. For instance, we know that in breast cancer there is the BRCA gene product that is important, right? Similarly, we know in pain or fever, cyclooxygenase is important. तो ऐसे बहुत सारे डिसीज के लिए टारगेट्स ऑलरेडी हमको पता है. So the first database is actually, which is therapeutic target database, is a compilation of all approved targets which are used by FDA approved drugs. Apart from that, there is a very nice compilation called DisGeneNet, which actually makes literature association between genes and diseases. So how this works is that many times people are doing transcriptomic studies or gene expression, and if you find a disease and a gene constantly coming up in literature, it might have an association. Okay, so this is the type of association, and of course there's another compilation called GeneCard, but GeneCard is not amenable to coding by APIs, so right now I have put a star there, so we are not able to integrate that into our automation. Okay, so I hope आपको network pharmacology का थोड़ा बहुत in synopsis you have understood what we are trying to do. So if I know that Haritaki is working, let's say, for digestive conditions, I know that for sure you are prescribing it, let's say you are advised to prescribe, but how it works is not known. So we are trying to use this to fill in that gap. So Haritaki ki I found the phytochemicals, I found the targets with which those phytochemicals interact, and if these targets actually have a role in digestive diseases, this entire network I can make and hence show that this is how, you know, this Haritaki is working for digestive disorders. So in a nutshell, हम क्या कर रहे हैं कि we are trying to make a web server which we launch very soon, next couple of months. The user just right now has to upload phytoconstituents. So like I said, the first step जो मैंने हाथ दिखाया था, that still we have not able to automate it. That will come in the next version. So still you yourself have to go to literature or do your own experiments or go to databases and compile your phytochemicals in an STF file or some file. And you have to, ये आपको यहाँ upload करना पड़ेगा. तो जैसे आपको हरितकी और digestive disorders का link निकालना है, तो आपको क्या करना है, हरितकी fruit के phytochemicals निकालने हैं, either by experiments or databases or literature, upload it here. In disease name, you can put digestive disorders. We will have a listing of different types of disease and just say submit. आप submit करोगे तो your network will pop up like this. For instance, I have given you a toy network where मैंने सिर्फ ellagic acid को upload किया था and बोला था Parkinson disease. So it has shown me how ellagic acid can be useful for Parkinson disease with these targets, which are known experimental associations between ellagic acid. So it's not hypothetical. Okay, the only thing we are not aware of right now is at what concentration ellagic acid can go into the blood and how it can activate or inhibit these targets. And I'll say this will, I'll show you how this will come in subsequent versions. So shayad ye thoda technically complex hai, but I just want to show you that when we'll release it, the user just has to click and you will get an answer. But before you use such click, it's nice to know how it's working. What are the limitations of that tool? So हम क्या कर रहे हैं, and this is all an effort of a big team of students, all Python codes written here. So at the front end, when the user inputs an SDF file, we use a Python RDKit library to separate those files. So if it is, let's say 70 small molecules that you have uploaded in one file, we separate it into individual files, SMILES and INCHIKI, these are different small molecule formats. And then we are querying to 3 databases, PubChem, BindingDB and BioGrid. So we use the API services. Matlab these web databases have given us tools to query and access it for free. So we can, by code, what we do at backend, we take those input, query it to PubChem and retrieve the gene ID or gene symbol. So from PubChem, BindingDB and BioGrid, we get the gene symbols of the protein targets. We make a non-redundant list. Then we query from there to the DisGeneNet. Like I said, there are some databases that don't give a very nice API for querying. So, and also some are not available to download and so on. So we are a bit limited by resources. Then what we get is a listing of gene name, disease name and compound. तो ये जो मैंने network दिखाया है, this can be generated by a software known as Cytoscape. Okay? तो फिर हम क्या करते हैं? Again using a program, we it's a Python module for Cytoscape. So we take this as input and we generate that network. So what user will get will be a zip file. आप वो download करो. So not only will there be a Cytoscape network, but also directly an image, something like this. Okay, so this is how our interface is designed and what goes in the back end. So it's a way to help you to link your formulation to disease at the click of a button. Okay. So this is our future versions. I'll just discuss what are the limitations and how we are going to try to address them in subsequent versions. So like I said, right now you have to upload the phytochemicals yourself, which we will release, but little later we are also doing research whether directly the user can give name of botanical. So if I directly say Terminalia cebula or different types of botanicals here and disease name, then at the back end we'll use natural language processing to actually search through literature and download all the free text and go from document to structure. Okay, and there are some commercial tools to do this now. मतलब एक paper में अगर कुछ phytochemicals के नाम है, structure है, तो directly through code I can get it in the format I want. Or alternately also we will write codes to query different databases and we'll get a non-redundant list of phytochemicals. So one more step we will try to go back for you, where you just have to type name of botanical and disease name, click a button and you'll get a network. Okay, but of course, and I'll just come to the end, what will be the perfect version for this? मतलब when can we say कि हमारे पास सारा evidence है to connect botanicals to disease? Okay, so ideally it should be like this, where the user says botanical 1, 2, वहाँ type किया, कौन सा disease type किया. So I should, through all my automation, be able to say, oh, it has, let's say, 5 phytochemicals, and I should be able to tell that this phytochemical is absorbed and it will be in the blood at a concentration of this milligram per ml in plasma. Now, if I know that concentration in plasma, then for sure I can tell that at this concentration it can inhibit or activate which target and hence help alleviate which disease. So this is like the perfect version which one day perhaps we'll reach. Not all phytochemicals have such pharmacokinetic data as of now, but some do. So in the hope that in future a lot of people will work and we do have this available, the perfect version of course will be this. So this is the end. So this was what I wanted to convey to you about what is network pharmacology and what we are doing to automate this. So agar aapko interest hai, we will be releasing this in, let's say, couple of months. You can use it and give us feedback. Hopefully we are trying to establish, a connection, help you to accumulate evidence linking your botanical to disease. Ki how this is working, ye kaise kaam kar raha hai, to help you gather such evidence. So at the end, I'd like to thank our team at CCIH. Some of them are over here, who have been instrumental. So at the bioinformatics and my personal research, I do many other things. I look at structure function inhibition of proteins using routine molecular modeling dynamics tools. But they are instrumental in having us excited about Ayurveda and how it is a rich traditional medicine that we really must understand. And I think computation, like you have been hearing from different speakers, can definitely play a role in trying to gather the much needed evidence that is required. So from CCIH, I had specifically mentioned Dr. Swapnil Borse, Dr. Girish Tillu and Dr. Akash Saggam, who have collaborated with us a lot on putting this automation together, and like I said, we are yet to release it. Mrs. Smita Saxena, who is my colleague at the Bioinformatics Center, she looks into IT. And next are all the students, Pulkit, Bhupali, Sakshi, Aditya, Isha, who are bioinformatics students. Aditya was an engineering student from MIT at Pune. And they have actually worked on different aspects of this automation that was required in Python, developing the front end and so on. And of course, the Bioinformatics Center for Infrastructure Support. So I will stop here. Thank you so much. But before I stop here, once again, I'd like to say a big thank you to the organizers, Dr. Vedapriya ji. And because of this opportunity, I actually could be on the stage in person with Swami ji and Acharya ji, and I think that was an opportunity of a lifetime. I'll never forget that. So thank you very much, and open to questions if you have any.
So the podium is open for questions now.
Thank you, madam, for this talk. And heartiest congratulations for your portal first. Madam, I have a query. Like you have made two inputs, right? First was the phytochemical STF file, and second is the disease. So how many genes you are including for a particular disease?
There is no limit to that.
Because
So it depends on whatever will come at the back end. There is no limit to how many genes.
Because my question is to identify the gene responsible for the particular disease, it's a tedious task.
That's why it is automated.
See, when I'm talking, suppose, take an example of breast cancer. If I'm talking about breast cancer, see, you will be talking about estrogen receptor, progesterone receptors and all. But when I switch towards a triple negative breast cancer, none of these receptors will be
Which
involved.
is why I said we are not only querying the therapeutic target database, which has known FDA approved targets, but also DisGeneNet, which has disease gene connections from literature. So then we are sure not to miss many of the targets which have not been explored by modern drugs.
That's my request to you.
We have
Please.
already included that.
Okay, thank you, madam.
नमस्कार ताई, खूप अफलातून आयडी कल्पना
Thank
आहे
you.
तुमची. धन्यवाद. माझा प्रश्न असा आहे
Okay,
की
this is
Yeah,
becoming
I'll-
trilingual now. Either you can talk in Hindi.
Okay, I'll talk in English. Yes,
Yeah.
yes. So I really love to speak with you in
Sure,
Marathi.
sure, yeah.
Yo, so the question is, if we talk about any particular herbal treatment, जैसे मान लीजिए अगर उसको गर्म कर दिया जाए या फिर पानी में भिगो दिया जाए, उसका जो प्रकृति है वो बदल जाता है. So when we talk about whatever element and then particular disease, so how would that
This
be
is an
catered?
excellent question, and this is still one of the limitations which we don't understand. One is that when you give it with different mediums, how the properties of the small molecules change. So we need some very good chemists to help understand this better. Now in Ayurveda, I have come across this, give this with ghee, give this with milk. This is very important. But how does that change the property of the ingredients? We don't know. So actually we should have an excellent brainstorming of chemistry peoples who will help us understand this better. So as of now in this network pharmacology, we are not able to understand how medium affects the ingredients and how when they come together as a mixture, how the properties change. That we are not able to address yet. So Ayurveda is so complex. This is just one baby step towards understanding it.
And ma'am, even I feel the prakriti of the person also depends upon the potency of that particular element towards treating that particular disease. Don't you think so?
So I think, see, each person would have the same protein, right?
Okay.
That will not change.
Okay.
But depending on prakriti, you may be giving different things. That might change.
Okay.
Isn't it?
Yeah. Dhanyavad.
Thank you very much. Thank you. Sure, one question.
Dr. Joshi,
Yeah.
my congratulations for
Thank
an
you.
excellent presentation. And like you, I'm also not from Ayurveda. I'm basically from botany, but of course associated with medicinal plants for the last three to 4 decades. Now, I was keenly listening to your lecture. I won't say that I have 2 questions, but I have 2 queries to make,
Okay.
because I understand the limitations of each
Yeah.
one of us. One is when we were listening to your presentation in the backdrop of evidence-based therapies,
Yes.
because you are trying to provide us a solution to create evidences of therapy of this traditional system. Now, what do we understand by evidence? I want to, please educate me on this, because supposing there's a disease condition in a person, supposing I am personally suffering from gout for the last 20 years, and I am managing it through Giloy.
Okay.
I'm not saying it putting on record,
Okay.
this is my personal
Okay.
experience. And this is treating me, my gout very well for the last 20 years. I just don't do anything. Now for me, it is an evidence that Giloy is working in me. Now if we can get some evidence that Giloy extract is working to treat gout, which is controlling my uric acid level, that would be fine. No, what in Giloy is actually working, in which way, why does it matter?
It does matter.
Yes,
Okay.
let me, because it does matter, because we are trying to bring in more reductionist approach in our
Yes.
system. Because that is not core Ayurveda. Although I am not from Ayurveda, but what we have understood, it is not Ayurveda, because we are far, as you have rightly pointed out in your own presentation, there are so many limitations.
Yes.
There was a beautiful question asked about the
Yes.
chemistry. You said you don't have even a chemistry. I was surprised that without that feedback, how can you arrive at some conclusions with this one? This is my one query, number one. Second is that when we look at, and this is not only on the basis of your presentation, but what we have understood, generally when we assume the therapeutical value of any medicinal plant, we focus on secondary metabolites.
Yes.
Why is it that we are ignoring the primary metabolites? Because why I'm saying this, there is a background talk happening that, take example of ashwagandha. The modern science or modern literature is more focusing on the vithanolide content in ashwagandha, without providing much of a convincing evidence that the advantages, the benefits of ashwagandha is wholly and solely due to vithanolides. Even there is some literature coming up that it is the carbohydrate content which is also responsible, which is, even there are some industries who are valuing the carbohydrate content much more than the vithanolide content. So we still don't know, we are much far away from that. So why are we excluding the primary metabolites in this whole area of our research and trying to create an evidence?
Sure. So I'll answer both of your questions one by one. So like I said, even in Western medicine, to be FDA approved, you don't necessarily need to have the molecular mechanism worked out. Like I had said, when aspirin was approved, we didn't know that it actually goes, the acetyl group goes and chemically attaches, blocks it, but it's scientific curiosity. We need to know the complete story. It's a curiosity that one is driving us to understand this connection. That's answer to your first question, because even as a person, I'm sure you must be wondering ki I am taking Giloy, why is it curing me? Haven't you thought of that?
So
that's what it is. I mean, nothing else.
But that is not addressing my core concern to provide evidence to my.
Like I said, evidence can be at different levels, as you correctly said. Even to be FDA approved, like I said, I need not provide molecular mechanisms like this. As long as I prove in vitro it's working, on animals it's working, human trials is working, I need not know at target or protein level how it's working. That is still true for modern medicine as well. But the sheer scientific curiosity, how it works, that drives this whole science. And that's why for so many modern medicines also, we know the molecular mechanisms. Because we just want to know how it works. It's not satisfying not to know how it doesn't. So it's the same thing here. And I'm sure there are limitations and we need a lot more study because Ayurveda is much more complex than this reductionist approach. But that doesn't mean that we should never take steps. In fact, these people might remember, I always used to be on the opposite side, that how can we use computational tools, to understand this. But unless we take some steps, we'll never get there. And I'm totally convinced by that. So unless we need lot more chemists to model these mixtures or to understand also primary metabolites. Like I said, which is why I said we need to know every metabolite is there in what concentration. So what people are focusing on is going by vithanolide is there so much, that's why they're focusing on that. But that doesn't mean anything else in micro concentration may not have an effect. Definitely it could. There are so many other things. There are nanomaterials, like you said, carbohydrate content. So many other things can trigger. We don't know that yet. But at least this is one way of trying to understand or trying to explain using data that is already there, experimental data. Of course, we may be missing so many things, but we need more brainstorming and more science to understand this. That's my answer.
Exactly.
Oh, can I?
Ji, sir.
I know I'm going to take little time, but I think in the view of the context of the conference, this is very, very important. First of all, I want to compliment you for the nice presentation.
Thank you.
Number two, I'm standing here because I believe that the bioinformatics can provide major solutions to provide an integrated modality for the technology adaptation that we are talking. We have been talking about it for the last 3 days. So whatever I say should be taken in a very positive manner. Now let me give an example of Triphala, why it happened. There are very few people who are aware about it, but I was one of the earliest people who identified in 11 clinical areas 9,700 formulations. That was computed between 1982 to 87. And in that, Triphala was found to be present in 37% of the formulations. And that is from where we picked up this idea long back with Bhushan.
Okay.
That why did we find 37% of the formulations having Triphala and 33% of the formulations having Trigger-2. That is how the bioequivalence approach of papain was emerged, and this is how the studies took place. Somewhere, a very physical data of collecting information on formulations and the ingredients played a major role in doing that. This is a simple solution. We are talking about simple solutions. Now, if we are looking at these solutions, we borrowed the word, and many times we we end up doing that. Chinese were the first to use the word network pharmacology,
Yes.
Li and other people, if I remember correctly.
Yeah.
And then we followed it and we published as a theoretical paper in 2015 by Bush and the
Yes,
people.
yeah.
And then we are still struggling to find out where the Triphala stands.
Yes.
Very interesting. At the end of now so many years of work, have we been able to position Triphala in the right manner? Then, just now he gave an example of ashwagandha. In 1980, I was asked by Ranbaxi to collect data on ashwagandha. I was only young, 28-year-old botanist and Ayurvedist, and I collected data on about 10 pages and gave it to them. Just about a month back, the Ministry of Ayush has formed a separate group on ashwagandha, including those people from American Homeopathy under the Upton, Roy Upton. So we are still struggling about ashwagandha from 1980 to 2023. Now in that situation, if we want to use the bioinformatics in the right manner, the question is, are we going to make it? I liked your word. You said that Ayurveda is very complex, and you are so right. But you must understand that Ayurveda is most simple. I can define all these people in terms of Vata, Pitta, Kapha. I take simple things of principles of Panchamahabhut. Are we afraid of using the simplicity of Ayurveda to understand the complexity of biosciences? That requires a different respect and mutual understanding, sitting across the table and respecting the basics of classification of the Ayurvedic drugs, how they are taking place. If you are looking at the chemicals, Tamilala chabula and Tamilala barica have the minimum 5 phytoconstituents with compared to any other drugs. Are you aware about why? Because for many, many years, the chemists have avoided doing any work on that because they have found only tannins.
Okay.
But you must be knowing that our whole human body, when we die, is nothing else but tannin, tannin, and tannin. Now, if this similarity of tannin, of terminalia, and if the body, human body, when it is no more, that is also made up of tannin. If you cannot identify the biosimilarity of that, then I think there is something wrong with our approach. Okay. We need to look at the fundamentals of Ayurveda. Why Haritaki is called as Yatra Swagure Matra Nasti, Tatra Mata Haritaki. What is the meaning behind that? I think if we look on doing phytoactives only, we will never be successful. You did mention about chemotoxicity,
Yes.
but in my opinion, what you need to look at the biological bioactives which are given, and you have to look at the biological molecular pathways, and that is what will provide the super sandwich. Thank you.
Thank you.
हमें लगता है कि आयुर्वेद जो 5000 या उससे भी पुराना विज्ञान है, उसको समझने के लिए मॉडर्न विज्ञान को अभी बहुत समय लगेगा, और इसलिए जो आयुर्वेद में जैसे एक च्यवनप्राश होता है, उसमें 52 द्रव्य पड़ते हैं, और उन 52 द्रव्य में कम से कम हजारों फाइटोकेमिकल्स या एक्टिव इंग्रेडिएंट्स होंगे. तो हम अगर कहते हैं कि च्यवनप्राश खा के हमें च्यवन ऋषि रिजुविनेट कर गए, तो उसके लिए किसी एक एक्टिव इंग्रेडिएंट को रिस्पॉन्सिबल हम नहीं बता सकते हैं. इसलिए आयुर्वेद को समझने में हमें थोड़ा समय लगेगा. धन्यवाद मैडम, आपका धन्यवाद.
Thank you very much.
Next, I would like to invite Professor R.C. Dubey. He is a professor and former head of the Department of Botany and Microbiology, Gurukul Kangri University, Haridwar. His field of interest is in rhizosphere microbiology and phytopathology, having expert in biocontrol of soil-borne plant pathogens, mycorrhization, helping bacteria, rhizosphere, microbiology, microbial biotechnology, and botanical pesticides. He has published several research and review articles in national and international high-reputed journals and has produced around 27 PhDs. And Professor Dubey has been a member of the Research Degree Committee and Board of Studies of several universities. I welcome you, sir.
Good afternoon to everybody. I extend my heartfelt thanks to the organizer, Honorable Acharya ji, Dr. Ved Priya, and other members to deliver lecture. And thank you, Mr. Chairman, sitting on the dais, and honorable members, scientists, doctors sitting offside. I am not practitioner, neither medical practitioner, neither Ayurvedic, nor any. I am simply a teacher of botany and microbiology in Gurukul Kangra University. I have been given time to discuss with you a new concept of the microbiology, Vedic microbiology. People generally ask me, is microbiology described in Vedas? It is really a question. Because in Vedas, every hymns are written are in Sanskrit language. And microbiology term is English. The term originated from the other country, not India. But question is any ethos or any intuitions regarding the microbes and related diseases? I say yes. People put question who have not studied Vedas, but who know about the Vedas. They can say there is a mention of all these, but we have to search it out. Most difficult part of our country is that during our childhood, we have never been taught about the Vedas. Even to university level, higher education, no teaching to the science student about the Vedas and to this Vedic student about the science. So this is a gap, this is a lacuna on the part of Ministry of Education of India. Who will fill up this gap? We teachers are responsible to spread the message about the Vedic philosophy, Vedic thoughts, at least to primary level or secondary level or intermediate level. So keeping all these things in view, I suppose that I should give a message about the microbes that have been described in the Vedas and how our rishis discovered all those. This is a miracle. And really, as our chairman sitting on the dais, they said that the modern science is still far behind to realize about the concept of Ayurveda. This is fact. This is true. So there is a need to combine the modern science with the Vedic science, including Ayurveda. So this was the purpose to come to here and discuss all these. थोड़ा मैं ब्रीफ में बताना चाहता हूँ वेद कहते किसको हैं, क्या वेद लिखे गए हैं? वेद लिखे नहीं गए, लिखने के लिए पेन चाहिए, कलम चाहिए, स्याही चाहिए, कागज चाहिए, और उस पीरियड में न तो कलम था, न तो स्याही थी, न पेन था, नथिंग एल्स, कुछ तो नहीं था। तो वेद कहते किसे हैं? वेदास आर रिवील्ड ट्रुथ। मेडिटेशन करते समय, ध्यान करते समय जो हमारे उस तरह उस जमाने के ऋषियों को कान में आवाज में सुनाई दिया, वो रिवील्ड ट्रुथ है। सत्य के बारे में, सच्चाई के बारे में जो सुनाई दिया, आवाज आई वह, और यह हजारों वर्ष तक परंपरा चली रही। गुरु लोग अपने गुरुकुल स्टेब्लिश करते थे, शिष्यों को पढ़ाते थे, बताते थे, और शिष्य अपने गुरुकुल में ही रख करके गुरुओं के द्वारा दिए गए ज्ञान को याद करते थे, लिखते थे नहीं, याद करते थे। उसको बोलते हैं स्मृति। स्मृतियों में सब कुछ था। धीरे धीरे वह परंपरा खत्म होती गई, स्याही आ गई, कागज आ गया, भोजपत्र आया, अब तो ढेर सारे तरह के कागज आए हैं। अब कुछ नहीं, अब तो मोबाइल आ गया, उसी पे टाइप करते हैं। लिखना भी आदमी की आदत धीरे धीरे खत्म होती गई, जमाने के साथ में परिवर्तन होता गया। तो वेद कहते हैं जो रिवील्ड ट्रुथ है, मेडिटेशन के समय जो हमारे ऋषियों को प्राप्त हुआ, लिखा नहीं गया, वह है नॉलेज वैदिक। और शुरू में 4 ऋषि थे, अग्निदेव, वायुदेव, आदित्य, अंगिरा, जो परम पिता परमात्मा का ध्यान किया, और उनको क्या मिला? वेद। अग्निदेव को ऋग्वेद मिला, वायुदेव को यजुर्वेद, आदित्य देव को सामवेद, अंगिरा को अथर्ववेद। आगे इसका भी कई भागों में बांटा गया है। ऋग्वेद का उपवेद है आयुर्वेद, और यजुर्वेद का है धनुर्वेद, सामवेद का है गंधर्ववेद, अथर्ववेद का है अर्थवेद, और इसको ब्राह्मण ग्रंथ में बांटा गया है। खैर वो सब जाने दीजिए, ये लोग जो जानते हैं अच्छी बात है। और जितने भी वेद हैं सबके एक महावाक्य है। जैसे ऋग्वेद का प्रज्ञानम ब्रह्म, यजुर्वेद का अहम ब्रह्मास्मि, सामवेद का तत्वम असि, अथर्ववेद का अयम आत्मा ब्रह्म। ये सब सीक्रेट नॉलेज जानने के लिए ही ये सारे ये जो महावाक्य हैं ये बताए गए हैं। और आखिर वेदों की उत्पत्ति हुई कहाँ थी? वेदों की उत्पत्ति कहाँ हुई थी? जहां कहीं वेदों की उत्पत्ति हुई थी उसे कहते हैं आर्यावर्त। उस भूभाग में रहने वाले लोगों को कहते हैं यह भूभाग आर्यावर्त है। वह पाया गया है कि जब तिब्बत के एरिया में ये आर्यों का उद्भव हुआ और वहीं से स्प्रेड हुए ज्ञान का उन्होंने, क्योंकि बार बार वेद में हिमालय का वर्णन आता है, बार बार हर वेद में, चारों वेद में हिमालय का वर्णन आता है। इसलिए वेदों का प्रादुर्भाव मान लिया गया हिमालयी क्षेत्र में हुआ है, और यहाँ के रहने वाले लोग को आर्य बोलते थे, और उस स्थान को कहते हैं आर्यावर्त है। आर्य किसको कहा गया? निरुक्त में आया है आर्य ईश्वर पुत्र, जो ईश्वर का पुत्र है वह आर्य है। ऋग्वेद में आ गया अहम भूमि ददामि आर्यम, मैं यह भूमि आर्यों को देता हूं। कृण्वन्तो विश्व आर्यम, यहाँ के ऋषियों ने यह संकल्प लिया कि हम पूरे विश्व को आर्य बना देंगे। लेकिन हमें क्या पढ़ाया गया है? विदेशियों का इतिहास पढ़ा गया कि आर्य यहाँ नहीं, ये मध्य एशिया से आए हैं। मुझे भी यही पढ़ा गया, यहाँ पर नहीं थे आर्य, ये मध्य एशिया से आए और यहाँ लड़ाई झगड़ा हुआ और यहाँ इस्टेब्लिश कर दिए। ये द्रविडियन से लड़ाने के लिए ऐसा कहा गया, ये बिल्कुल गलत है। इसलिए मैं कहता हूं हमारा जो इतिहास मुझे भी पढ़ा गया, ये गलत पढ़ा गया है अपनी उत्पत्ति को लेकर के। दो व्यक्ति का मैं नाम लेता हूं, एक थे बसंत साठे और एक डॉक्टर नीरज राय, एक भूगर्भ वैज्ञानिक और एक जो मॉलिक्यूलर बायोलॉजिस्ट। इन लोगों ने खुदाई किया हीरागढ़ी एक जगह है हरियाणा में, वहां से कंकाल निकले, ढेर सारे नर कंकाल निकले। डीएनए फिंगरप्रिंटिंग किया, मॉडर्न बायोटेक्नोलॉजी का यूज करके डीएनए फिंगरप्रिंटिंग किया, और फिंगरप्रिंटिंग करने के बाद उन्होंने पाया, उन्होंने कहा अरे विदेशी नहीं, भारत के मूल निवासी थे, ये बाहर से नहीं आए, यहीं के मूल निवासी। इंडस वैली सेटलर्स हैड ए डिस्टिंक्ट जेनेटिक लिनिएज, इनका जेनेटिक लिनिएज किया। आर्यों ने भारत पर नहीं किया था आक्रमण, वे यहीं के मूल निवासी थे। ये अखबारों में जो निकला है, कुछ चीजें मैं आपको दिखा रहा हूं। ये जो नर कंकाल निकले, 50 नर कंकाल निकले, सबका पाउडर बनाकर उन्होंने डीएनए फिंगरप्रिंटिंग किया, और आज के डेट में अलग अलग जाके ब्लड सैंपल लेके मैच किया, मैच करने के बाद पाया यही आर्यों ने भारत में आक्रमण नहीं किया, वो यहीं के मूल निवासी। जो हमें इतिहास पढ़ाया जाता है वह गलत है। और एक थे डॉक्टर लालजी सिंह, मर गए, जिसको सन ऑफ द सॉइल उपाधि मिली गई, पद्मश्री के साथ सन ऑफ द सॉइल, जिन्होंने फर्स्ट डीएनए फिंगरप्रिंटिंग हिंदुस्तान में किया। और राजीव गांधी एसेसिनेशन जो हुआ था, इंदिरा गांधी एसेसिनेशन, उन सब में उनका रोल है। ऑस्ट्रेलिया रहे, इसके बाद यहाँ जाके डीएनए फिंगरप्रिंटिंग का केंद्र इन्होंने हैदराबाद में खोला। और उनका भी है जेनेटिक एविडेंस फॉर रिसेंट पॉपुलेशन मिक्सचर इंडिया। आज की जो पॉपुलेशन है रिसेंट इंडिया की, यह मिक्सचर है। यहाँ के लोग विदेश गए, विदेश के यहाँ गए, शादी ब्याह कर गया, सेटल गए, और मिक्सिंग है। अब थोड़ा मैं आ जाऊं माइक्रोबायोलॉजी के बारे में। निरुक्त कहता है क्रव्य मेदद क्रमते वी स्यात् शरण कर्मण कामते वी। क्रव्य जो न्यूट्रिएंट डिराइव करे, दूसरे जगह से न्यूट्रिएंट डिराइव करे, उसको कहते हैं क्रव्य। अब क्रम किसको कहते हैं? टू मूव, क्रमण करना, आगे आगे चलना। और काम टू विश, डिजायर। तो जो मूव करे वो क्या है? वो क्रीमी है, जो मूव करे एक जगह से करे। और हमारे यहाँ ऋषि कण्व हुए हैं। अथर्ववेद में इन्होंने बहुत मंत्रों का वर्णन किया है। ऋषि कण्व को मैं तो कहता हूं इतना ज्यादा वर्णन किया है, व्हाई नॉट ऋषि कण्व शुड बी रिगार्डेड एज फादर ऑफ माइक्रोबायोलॉजी। एंटोनी वान लीeuwenhoek को पढ़ाया जाता, क्यों पढ़ाया जाता? ऋषि कण्व को क्यों नहीं पढ़ाया जाता? इसलिए कि हमारी एजुकेशन व्यवस्था में जो भी टीचर बन के आते हैं उनको ज्ञान ही नहीं है, न टेक्स्ट बुक में लिखा है, न टीचर ने बताया। उनका कोई दोष नहीं, मैं किसी को दोष नहीं देता हूं, दोष देता हूं शिक्षा व्यवस्था की। इसलिए मेरे हिसाब से ऋषि कण्व मस्ट बी रिगार्डेड एज द फादर ऑफ माइक्रोबायोलॉजी। आगे मैं इनके ढेर सारे चीजों को, एप्लीकेशंस को बताऊंगा। मनुस्मृति में आता है कि आखिर जितने भी लाइफ फॉर्म हैं उनको कैसे बांटा गया। स्थावर कृमि कीटाश्च मत्स्यास्त्रपाशकच्छपा पशुश्च मृगाश्चैव जगन्या तापसी गति। जो स्थावर हैं वो प्लांट हैं, कृमि जो क्रमण करे वो कृमि है, कीटाश्च कीट जानते हैं आप लोग, इंसेक्ट, मत्स्य जो टोटोइस हैं, सर्पा स्नेक्स हैं, सकच्छपा पशुवश्च क्वाड्रूपेड, मृगाश्चैव जगन्ना तामसी गति। ये चार जो तीन गतियां हैं, इन सबको जगन्य गति में बांटा गया। कृमि का वर्णन आता है, मनुस्मृति ने भी इसका वर्णन किया है। और आगे क्लासिफिकेशन जो पद्म पुराण में आता है, संख्या भी बता दिया गया। जलजा 9 लक्षाणी, स्थावरा लक्ष विंशति, कृमयो रुद्र संख्य का, पक्षीणाम 10 लक्षणम, 30 लक्षाणी पशव, 4 लक्षाणु मानुषा। जो जलजा है, वाटर बॉर्न ऑर्गेनिज्म 9 लाख हैं। जो स्टेगनेंट है, स्थावर है, प्लांट हैं, वो कितने हैं? 20 लाख हैं। माइक्रो ऑर्गेनिज्म 11 लाख हैं। बर्ड्स ये 10 लाख हैं। क्वाड्रूपेड 4 पैर चलने वाले जानवर 3 लाख हैं। और ह्यूमन स्पीसीज 4 लाख है। हम लोग सोचते हैं कि ह्यूमन सेपियंस हम अकेले हैं, वनमानुष को क्यों भूल जाते हैं? वो भी उसी कठिन 4 लाख। अब खोजना पड़ेगा कौन कौन है। कहा गया 4 लक्षाणु मानुषा, ये 4 लाख मानुष की प्रजातियां हैं। कौन कौन है? माने मानुष माने मैमेल्स कह लीजिए, इसको मैमेल्स कैटेगरी में एक कर लीजिए। एक खोजना आज के मॉडर्न साइंस का एक काम है। लेकिन हमारे ऋषियों ने उस काल में, वैदिक काल में ये सारा वर्णन कर दिया। तो आज के जो वैज्ञानिक है वो ज्यादा एडवांस है, या पहले के ऋषि जो थे वो ज्यादा एडवांस हैं? ये भी एक सोचने की बात है। आगे माइक्रो ऑर्गेनिज्म को के लिए कहा गया सूक्ष्म योनि भूतानि तर्क गम्यानि कानचित। जो सूक्ष्म योनि के जीव प्राणी हैं, माइक्रो ऑर्गेनिज्म है, उनको आप देख नहीं सकते, लेकिन तर्क गम्यानि कानचित, आर्गुमेंट करके, आर्गू करके, असेस करके वहां तक आप पहुंच सकते हैं। किसी ने कोरोना वायरस को देखा नहीं, लेकिन लक्षणों के आधार पे कह दिया आपको कोरोना है, तुरंत ले जाकर के एक जगह सुला दिया कि नहीं आप 14 दिन तो यहीं रहेंगे, घर नहीं जाएंगे। आजकल यही हो रहा था 2 साल पहले। देखा किसी ने नहीं माइक्रो, तर्क गम्य आने माने उसके लक्षणों के आधार पे तर्क करके आप वहां तक पहुंच सकते हैं। पहला कौन सा व्यक्ति था जिन्होंने माइक्रो ऑर्गेनिज्म या ऑर्गेनिज्म को माइक्रो ऑर्गेनिज्म को देखा? और माइक्रोस्कोप भी था क्या? भगवत गीता में आता है, न तो जिस समय भगवान ने, कृष्ण ने अपना विराट रूप दिखाया और अर्जुन से कहा देखो सारा ब्रह्मांड मेरे मुंह में प्रवेश कर रहा है। अर्जुन ने कहा न तो माम सकसे द्रष्टुम न नेव चक्षुषा, आप क्या कहते हैं, मुझे तो कुछ नहीं दिखाई देता। तब कृष्ण भगवान ने कहा तुम मेरे उस दिव्य रूप को नग्न आंखों से नहीं देख सकते। न तो माम सकसे द्रष्टुम नैव चक्षुषा, नग्न आंखों से मेरे उस रूप को, इन सारी चीजों को नहीं देख सकते। उनके शरीर का स्पर्श कर दिया, उनका दिव्य चक्षु खुल गया। तब उन्होंने कहा दिव्यम ददामि ते चक्षु पश्च मे योग में श्रम, मैं तुम्हें दिव्य दृष्टि देखता हूं। अब देखो, जैसे ही स्पर्श किया, देखा ब्रह्मांड के सारा छोटा से छोटा, अणोरणीयान महतो महियान, छोटे से छोटे बड़े से बड़े जो ऑर्गेनिज्म थे सब मुंह में प्रवेश कर रहे हैं। ये पहला उदाहरण है कि जिन्होंने कुछ देखा, नॉन विजिबल एंटिटी को देखा, ये पहला उदाहरण है। अब जो वेदों में वर्णन है माइक्रोब के बारे में, नाम अलग अलग है। माइक्रोब तो अंग्रेजी का शब्द, लेकिन जो संस्कृत का शब्द है यातुधान, जो वायु के द्वारा फैलते हैं, एक जगह से दूसरे जगह जाते हैं, कहते हैं यातुधान। ककुमथ, ध्वनि उत्पन्न करने वाले, वाइब्रेशन करने वाले, क्योंकि बैक्टीरिया में फ्लैजला होता है, आप जानते होंगे जो लोग पढ़े हैं बायोलॉजी, सभी लोग पढ़े हैं। उसमें जिसमें फ्लैजला हो, वाइब्रेट करेगा, वह कंकुमथ है। प्रमुथान, अत्यधिक संक्रामक क्षमता वाले जीव को बोलते हैं प्रमुषान। रक्षग्रीव, जो गले में दर्द करता है, इन्फेक्शन करेगा, रक्षग्रीव। श्रीमह, विषाणुओं को उत्पन्न करने वाला जीव। सुनामा, स्त्रियों में रोग करने वाले जीव को सुनामा बोलते हैं। दुर्डामा, जो यूटेरस में डिजीज करता है, वह दुर्डामा है। दुर्गंधिन, जो बैड स्मेल पैदा करे, दुर्गंधिन है। लोहित, जो लाल रंग के माइक्रोब हैं, लाल रंग के माइक्रोब हैं, वो लोहित हैं। इस तरह से 111 नाम माइक्रोब का वेदों में आता है, 111 नाम, ये सब मैंने कैलकुलेट किया है। अब मैं आपसे पूछूं, अमीबा सबको पढ़ाया गया होगा, किसी का पेट खराब होता है, डॉक्टर कहता है आपको अमीबियासिस है, एंटामीबा हिस्टोलिटिका डिजीज कर दिया है। शब्द अमीबा ये क्या अंग्रेजी का है क्या? बिल्कुल नहीं, ये अमीबा शब्द संस्कृत का है। यस्ते गर्भम अमीबा दुर्डामा योनिमाशय, जो यूटेरस, जो फीटस में डिजीज करे वह क्या है? अमीबा है। फीटस में डिजीज काज करे वह अमीबा है, और जो यूटेरस में सेटल कर जाए, यूटेरस में सेटल कर जाए और वहां रहे, उसका नाम दुर्डामा। यह ऋग्वेद में आता है। तो यह संस्कृत का शब्द जो हम समझ रहे हैं अंग्रेजी का है, बिल्कुल नहीं, यह संस्कृत का है। यस्ते गर्भम अमीबा दुर्डामा योनिमाशय। और महर्षि चरक ने इन सारे माइक्रोबियल डिजीजेज को 4 भागों में बांटा है, सहज और वैकारिक। वैकारिक जो विकार करे, सहज माने जो नॉर्मल लिविंग माइक्रोब है, पुरुषज, कफज, शोणितज और मलज। ये जो फीकल मैटर में रहते हैं वो पुरुषज है, स्पूटम में, थूक में रहता है वो कफज है, कफ में होता भी है माइकोबैक्टीरियम, टीबी भी जो करता है। इसके अलावा ढेर सारे माइक्रोब होते हैं। ब्लड बार्न माइक्रोऑर्गेनिज्म शोणितज है, जो केवल ब्लड के ऊपर डिपेंड रहे, और मलज जो वेस्ट मटेरियल वार्ण है वह मलज है। महर्षि चरक ने इसका डिस्क्रिप्शन चरक संहिता में कर दिया है। और माइक्रोब कहाँ क्या? बटर और मिल्क में, मक्खन में, दूध में, छाछ में और फूड मटेरियल में माइक्रोब का वर्णन किया गया है अथर्ववेद में। क्षीरे मां मंथे यतमो ददम्भ कृष्ण पक्षे असने धाने च तदात्मना प्रजा पिशाचा वियातियंताम गदोमस्तु। पिशाच शब्द आया है, लेकिन आप तो कहेंगे कि नहीं, तो उसमें जो हनुमान चालीसा है उसमें भूत पिशाच निकट नहीं आवे, वो तो भूत वाला है जो पकड़ लेता है। बिल्कुल नहीं, जो दिखाई न दे और हवा के माध्यम से चले और कष्टकारी हो, कष्ट देने वाला हो, वह पिशाच है। क्या भूत पिशाच वो दिखाई देते हैं क्या? वो भी हवा के माध्यम से चलते हैं, दिखाई बिल्कुल नहीं देते, कष्ट देते हैं। वेद में पिशाच शब्द माइक्रोब के लिए आया है, वेद में पिशाच शब्द माइक्रोब के लिए आया है, यातुधान भी आया है। अब अथर्ववेद में हरिमा शब्द है, क्षुद्र रोग, इसको इन सबको कहा गया जाउंडिस। जाउंडिस स्मॉल पॉक्स मम्स है, जिसको जाउंडिस हो जाए, स्मॉल पॉक्स का वर्णन। अब आपको लगेगा कि जाउंडिस तो वायरल डिजीज है, क्या वायरस का डिस्क्रिप्शन ये हमारे वैदिक लिटरेचर में है क्या? क्या स्मॉल पॉक्स का है क्या? मम्स है क्या? मैं कहता हूँ बिल्कुल है, पर पढ़ना पड़ेगा, पढ़ाना पड़ेगा, खोजना पड़ेगा। यो हरिमा जायांग वेदो विशल्कप सर्वतेजक्ष में, यह इसमें हरिमा माने जाउंडिस, इसका वर्णन है, डिटेल में पढ़ने की जरूरत है। आगे है जाउंडिस का काज क्या, कारण क्या है? ये चरक संहिता में आता है, पांडु रोगा स्मृता पंच वात पित्त कफ श्रेय चतुर्दश सन्निपाते न पंचमो भक्षणामृतम। ये 5 इसके कारण बताए गए हैं जाउंडिस के। कौन कौन से है? वात से, पित्त से, कफ से, चतुर्थ सन्निपाते न, चौथा सन्निपात जिसको हो जाए, और पांचवा है भक्षणामृतम, जो मिट्टी खाता है। मिट्टी में माइक्रोब होता है, वायरस भी होगा और माइक्रोब भी होंगे, तो इसलिए वो मिट्टी खाना भी एक कारण है। तो बच्चों को मिट्टी कभी नहीं खाने देना चाहिए। लेकिन जब शादी ब्याह में हम लोग जाते हैं, सलाद खाते हैं, सलाद में जब छिलते उलते नहीं है, जहाँ से उसकी रूट्स निकलती है वहाँ मिट्टी लगी रहती है। हम खा के आते हैं सलाद घर में, रात के 12 बजे पेट गुड़ 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 करने लगेगा। तो हमेशा सलाद नहीं खाना चाहिए, खाइए धो धा करके बिल्कुल साफ सुथरा, क्योंकि मिट्टी लगी ही रहती है। पंचमो भक्षणामृतम, इसका ध्यान रखिएगा। मम्स, गंडमाला, अपचिता में इसको अपचिता बोलते हैं। अपचिताम लोहिनाम कृष्णा माते सुतम मृदेवन मूले ने सर्वा विद्यांसता, अथर्ववेद में आया है कि ये जो अपचित है, जो मम्स है, जो माइनस सेंस आरएनए रूबेला वायरस जिसे कहते हैं, मिक्सो वायरस, इसके द्वारा होता है। और इसका निदान कैसे होता है? मुरुदेव ने मूले से, मुरुदेव कहा गया है पलाश को, पलाश के जड़ को निकाल के क्रश करके एक्सट्रैक्ट बना के वही अगर उसी का लेपन करिए उसको ठीक हो जाएगा। हमारे अथर्ववेद में इसका वर्णन है, मम्स का माने वायरल डिजीज का। तो मैं कैसे कह दूं कि वायरोलॉजी का डिस्क्रिप्शन नहीं है? वायरोलॉजी का डिस्क्रिप्शन है। और वायरल डिजीज में और आता है स्मॉल पॉक्स, गलगंड ग्रह रोगा, ये जानते हैं द्रावणम निड़ाम, गलगंड इसको गले में हो जाता है, वो भी आता है। और स्मॉल पॉक्स, गलगंड बता दिया आपने, स्मॉल पॉक्स तो या सर्वगात्रेषु मसूर मात्रा मसूरिका पित्त कफात प्रतिष्ठा, ये जो स्मॉल पॉक्स है इसको वैदिक लिटरेचर में चरक संहिता मसूरिका कहा गया। मसूरिका जो मसूर के दाने की तरह हो जाता, जब ये सूख जाएगा तो इसका सिम्टम मसूर के दाने की तरह होता है, पित्त कफ। और ये जब बिगड़ जाता है तो इसी के कारण होता है। और पित्त कफ कैसे बिगड़ता है इसको तो हमारे आयुर्वेद के जो डॉक्टर्स हैं ये तो बताते हैं, इनका तो इलाज उसी पर बेस्ड है। वात पित्त कफ दवा बता देते हैं। और अथर्ववेद में आता है कि ये जो सूरज की किरणें हैं इनका भी सेवन करना चाहिए। क्यों? क्योंकि सूरज की किरणों का जब सेवन हम करेंगे तो हमें जो शरीर के ऊपर माइक्रो ऑर्गेनिज्म लगे हुए हैं वो मर जाएंगे। उद्यन आदित्य कृमेन हंतु, उगते हुए और अस्त होते हुए सूरज की किरणों का सेवन करना चाहिए, दिन में नहीं। दिन में तो कोई बैठ ही नहीं सकता। आखिर क्या कारण है? उगता हुआ सूरज और अस्त होता हुआ सूरज, उगता हुआ और अस्त होते हुए सूरज की किरणें लाल रंग की होती हैं। और लाल रंग की किरणों में क्या खास बात क्या है? लाल रंग की किरणों की खास बात यह है कि उसमें इंफ्रारेड होती है। और इंफ्रारेड किरणें जर्मीसाइडल हैं, जर्मीसाइडल इंफ्रारेड किरणें हैं। आगे और इसके लाल रंग की किरणों का सेवन करने से किसी को भी हार्ट डिजीज नहीं होती और पीलिया भी नहीं होगा, जाउंडिस भी नहीं होगा। आगे वाला मंत्र कहता है अनुसूर्य मुदयताम हृदय तो हरिमाचते गो रोहित से वर्णन। गो माने गाय नहीं, यहां होगा गो माने सूरज की लाल रंग की किरणें, रोहित से वर्णन लाल रंग की किरणें। अनुसूर्य उदयताम, जो सूर्य उदय होता है और अस्त होता है उसका सेवन करने से हृदय तो हरिमाचचे, हार्ट अटैक नहीं होगा और जाउंडिस नहीं होगा। आखिर खास बात क्या है? क्यों सूरज की किरणें सुबह शाम लाल रंग की दिखाई देती हैं? जैसे माना कि आप यहाँ पर खड़े हैं, सूरज यहाँ निकलता है, किरणों को यहाँ से यहाँ ट्रेवल करने में टाइम लगेगा। और जो डस्ट पार्टिकल होती है, ये डस्ट पार्टिकल इसके जो किरणें और लॉन्ग वेव लेंथ होती है, शॉर्ट वेव लेंथ होगी, उसको स्कैटर कर देंगी। ये लाल रंग का दिखाई इसलिए देता है क्योंकि यही लाल रंग की किरणें पहुंचती हैं, बाकी नहीं पहुंचती। और सूरज दोपहर के समय लाल रंग का यह नहीं दिखाई देता है। और इसका एक एग्जाम्पल और दो, ट्यूबरकुलोसिस का। ट्यूबरकुलोसिस के बारे में बहुत कुछ कहा गया है अथर्ववेद में, और इसका डिस्क्रिप्शन, क्योंकि ये भी माइक्रोबियल डिजीज है, बैक्टीरियल डिजीज है। माइक्रोबैक्टीरियम ट्यूबरकुलोसिस करता है। ये होता कहाँ है? पहले तो बताया गया ये लंग में होता है। आज के जो डॉक्टर्स हैं ये बताते हैं कि नहीं, लंग के अलावा और जगहों पे होता है। और जगहों पे कहाँ कहाँ होता है? 6 मंत्र हैं अथर्ववेद में, और सब डिस्क्राइब कर दिया उन्होंने कि इस जगह होता है। अक्षियाम ते नासिकायाम, कर्णामाम चुकावती, यक्षम शीर्षाण्याम मस्तिष्कम, जिह्वायाम विहामिते, आंख में, नाक में, कान में, ठुड्डी पे, शीश में, मस्तिष्क में, जिह्वा में, ये सब जगह होता है। और कुछ इंटरनेट से मुझे मिल गया पिक्चर, मैं आपको दिखाने के लिए भी ला दिया। मंत्र तो आप इसलिए पढ़ रहा हूं। और आगे है ग्रीवाभ्यस्त, गले में, उष्णी हास्याम की सांसों, अनुष्याद यक्षम दूषणभ्यम, और ढेर सारे अंगों में ट्यूबरकुलोसिस होता है। भुजाओं में, आर्टरीज में, बोन्स में, हड्डियों में, आंतरेभ्य, गुदाभ्य, वनिष्ठभ्य, आंतों में ट्यूबरकुलोसिस, एनस में ट्यूबरकुलोसिस, बाउल्स में ट्यूबरकुलोसिस, पेट में, मेजेंटिक नेवल और नाभि में ट्यूबरकुलोसिस ये होता है। इन सब का वर्णन किया गया है। जो 6 मंत्र हैं, मैं सारा मंत्र यहाँ नहीं पढ़ रहा हूँ, शार्ट में मैं बताना चाह रहा था आपको। आखिर ये ठीक कैसे होगा? ठीक होने के लिए भी इन्होंने कहा है हमारे ऋषियों ने, न ते यक्षमा अरुंधते तेन सुपतो अनुसुदे यम भेषजा गुलगुलो गुलगुलो सुरभि गंधो अस्नुते, कि जो गुगुल का सेवन करता है, जिसके नाक में गुगुल का धुआं चला जाए, न ते यक्षमा अरुंधते, उसमें उसको यक्षमा नहीं होगा, उसको ट्यूबरकुलोसिस बिल्कुल नहीं होगा। क्यों? क्योंकि गूगल में गुगुलोस्ट्रान नाम का एक केमिकल पाया जाता है, इसी का चमत्कार है। गुगुलोस्ट्रान नाम का केमिकल पाया जाता है। इस केमिकल की डिस्कवरी तो बहुत बाद में हुई, ऋषि ने हमारे बहुत पहले कह दिया। और दूसरी बीमारी लेप्रोसी, दूसरा बैक्टीरियम है माइकोबैक्टीरियम लेप्री, इसका भी वर्णन आता है अथर्ववेद में। कहा गया है ये तो देवस्त्राणम कुष्ठो हिमोतस्र, कि इसको ठीक करने वाली दवा हिमालय पे पाई जाती है, और हिमालय से भी कलेक्ट करने के लिए का एक समय होता है। मैंने बताया ना कि हमारे जो ऋषि महर्षि थे और हमारे वेदों का प्रादुर्भाव हिमालय क्षेत्र तिब्बत के आसपास से हुआ है, तो उन्होंने कहा कि हिमालय पे ये पाई जाती है। तृष्ण ते कुष्ठ नामानी नद्य मारो नद्या ऋषय, हे कुष्ठा, माने कुष्ठ रोग को दूर करने वाला प्लांट, आपका 3 नाम है, कुष्ठ मारो नद्या ऋषय, कुष्ठा, नद्यमार और नद्या ऋषि। अब बॉटनिस्ट की जरूरत है, वो खोजें कहां पर है। नद्य मारो नद्या ऋषि है, फ्लोइंग वाटर में, स्टेगनेंट वाटर में, और जहां पे पानी नहीं होगा सूखे जगहों पे, जहां डॉक्टर साहब ने कहा सेकेंडरी मेटाबोलाइट जहां पे होता है, क्योंकि ये सेकेंडरी मेटाबोलाइट है, ऐसे 3 जगहों पे होता है। इसका इकोटाइप भी बता दिया। इकोटाइप तो आज हम कहते हैं, लेकिन हमारे ऋषियों ने पहले कह दिया, नद्य मारो नद्या ऋषि। और एक नई टेक्नोलॉजी शुरू हुई वैदिक टाइम में अग्निहोत्र की, कि अग्निहोत्र करिए, अपने इन्वायरमेंट को शुद्ध करिए, अपने को शुद्ध करिए, बीमारी नहीं होगे। आयु यज्ञ कल्पताम, प्राणो यज्ञ ने कल्पताम, चक्षु यज्ञ ने कल्पताम। क्यों? यज्ञ करने से आयु, प्राण, चक्षु ये पुष्ट होगा, ये हेल्दी रहेगा। इसलिए यज्ञ भी एक थेरेपी है। यज्ञ थेरेपी एक नई थेरेपी डेवलप होने वाली है, यजना थेरेपी भी लोग कहते हैं, या हो गई है। इस पर भी रिसर्च चल रहा है, हमारी यूनिवर्सिटी में चल रहा है, हमने भी रिसर्च पेपर छापा है। अब दूसरा सवाल उठता है कि आखिर अग्निहोत्र कौन करेगा, किसके लिए? कुछ रिस्ट्रिक्शन भी है। अग्निहोत्र फॉर ऑल, इररेस्पेक्टिव ऑफ जेंडर, रेस, कास्ट, रीजन एंड रिलीजन। यह कभी नहीं करना चाहिए कि अग्निहोत्र केवल हिंदुओं का है। अग्निहोत्र कोई भी कर सकता है। इट इज फॉर ऑल जेंडर, ऑल रेसेज, ऑल कास्ट, ऑल रिलीजन, क्योंकि वैदिक काल में कोई रिलीजन, कोई रीजन, कोई कास्ट था ही नहीं, सारे आर्य थे। और यहाँ पे देखिए अलग अलग देशों का यह लिया है हमने, यहाँ पर लोग कैसे कैसे जगहों पे इकट्ठा होकर के अग्निहोत्र कर रहे हैं। ठीक है अग्निहोत्र नहीं करते, लेकिन इसको लोगों ने दूसरे ढंग से अडॉप्ट कर लिया। अगरबत्ती, अगरबत्ती में क्या है, सारे आर्टिफिशियल केमिकल डाल दिए गए, उसका भी धुआं मुंह में जाता है। धूप बत्ती, उसमें भी इंग्रेडिएंट डाला जाता है, उसका भी धुआं। आखिर धुएं का प्रयोग करते हैं कि नहीं करते घरों में, अगरबत्ती के रूप में या धूप बत्ती के रूप में? अग्निहोत्र जो नहीं कर पाते, अपना माचिस जलाया, वो जय जगदीश हरे कर लिया, हो गया उनका अग्निहोत्र। और जिसको मंत्र आता है, समय है, वो बैठ करके अच्छे ढंग से करेगा। आखिर सिग्निफिकेंस अग्निहोत्र क्या है? क्योंकि जैसे आग जलेगी, समिधा में डालेंगे, तो इस हवन कुंड में अलग अलग जगहों पे अलग अलग टेम्परेचर पाया जाएगा। यह टेम्परेचर होगा नीचे से लेकर ऊपर तक आओ, लगभग 300 to 400 डिग्री सेल्सियस, आगे ऊपर 1200 से 1300 डिग्री सेल्सियस होगा। अलग अलग डिग्री सेल्सियस टेम्परेचर पे जो केमिकल्स होते हैं उनका फ्यूमिगेशन होता है, और निकल करके क्या आएगा, फार्मिक एसिड, एसिटिक एसिड, पायरुविक एसिड, लारिक एसिड, ये निकल करके बाहर आता है। कहाँ, ये कहाँ पाया जाता है? जो अग्निहोत्र में सामग्री में केमिकल सामग्री डालते हैं, उसका केमिकल निकल आता है। और क्या होता है, अल्कोहल, एसीटोन, एल्डिहाइड, बोर्नियाल, ईस्टर, ईथर, ऑर्गेनिक एसिड, ये अलग अलग टेम्परेचर पे निकल के बाहर आएंगे। और इसी के बगल में आप बैठे रहते हैं स्वाहा स्वाहा करने के लिए, तो आपके नाक में तो जरूर जाएगा। थोड़ा थोड़ा अगर रोज जा रहा है, तो जो रेस्पिरेटरी ट्रैक्ट में अनवांटेड माइक्रो ऑर्गेनिज्म है, सर्टनली दे विल डाई, उनको मरना ही मरना है। और आगे भी यही नहीं, आगे इफेक्ट का अग्निहोत्र और क्या करेगा, पॉल्यूशन, अगर बगल पॉल्यूटेंट्स हैं तो पॉल्यूटेंट्स को दूर करता है। वाटर प्यूरिफिकेशन, बगल में आप पानी को रख दीजिए, वो भी प्यूरिफाई हो जाएगा, क्योंकि वहां भी केमिकल जाएगा। इफेक्ट ऑन माइक्रोब, माइक्रोब तो 194% माइक्रोब मर जाते हैं। हमारी यूनिवर्सिटी में भी रिसर्च हुआ है, और जगहों पे लोगों ने रिसर्च किया, 94% माइक्रो ऑर्गेनिज्म मर जाते हैं ड्यू टू राइज इन टेम्परेचर एज वेल एज रिलीज ऑफ द केमिकल कंपाउंड। इन्वायरमेंटल प्यूरिफिकेशन होगा, इफेक्ट ऑन ह्यूमन हेल्थ, और शरीर हृष्ट पुष्ट होता है, ऐसा लोगों ने किया गया। और यह फ्यूम की एनालिसिस किया गया, इसका GCMS के द्वारा अलग अलग केमिकल पाए गए, अलग अलग इसका ये केमिकल की एनालिसिस है। अब मैं यहाँ पर क्या पढ़ूँ, मुझे दिखाई नहीं दे रहा है, आप लोगों को दिखाई, जैसे बोर्नियाल हो गया, इथाइल अल्कोहल, इंडोन, ये बहुत लंबा चौड़ा है, जो GCMS एनालिसिस से किया गया, ढेर सारे केमिकल एनालिसिस किया गया। और उसको आगे मंत्र है ऋग्वेद में, मुंचिताम तो हविषा जीवनाय कम ज्ञात यक्षमात राजस, यक्षमा राजयक्षमा, अगर किसी को हो गया है तो अग्निहोत्र करने से दूर हो जाता है। इसका ब्रीफ मतलब ये हुआ। अभी कुछ वर्ष पहले मैंने अग्निहोत्र का जो फ्यूम निकलता है, इस फ्यूम पर एक रिसर्च करवाया, अपने स्टूडेंट को कहा कि GCMS एनालिसिस करो, ये इसके मटेरियल का जो केमिकल आते हैं, गुगलोस्ट्रान, इसका जो बैक्टीरिया होता है रेस्पिरेटरी ट्रैक्ट में जाता है, लंग में इन्फेक्शन करता है, इसका मॉलिक्यूलर डॉकिंग करवाया। मॉलिक्यूलर डॉकिंग, रिसेंट बायोटेक्नोलॉजिकल टूल यूज करके मॉलिक्यूलर डॉकिंग ऑफ सम एक्टिव इंग्रेडिएंट ऑफ ऑब्लेशन मटेरियल यूज्ड इन यज्ञ अगेंस्ट ट्यूबरकुलोसिस काजिंग माइकोबैक्टीरियम। आपको देख के आश्चर्य होगा कि माइकोबैक्टीरियम की प्रोटीन्स और गुगलोस्ट्रान का इंटरेक्शन हो रहा है, बिल्कुल अच्छे ढंग से कंबाइन कर लिए हैं। जितने भी हमने एक्सपेरिमेंट किए, सब जगह कंबाइन मिला। इसका मतलब यह मॉलिक्यूलर टूल से अगर एक कंबाइन इंटरेक्शन करके कॉम्बिनेशन मिल रहा है, तो नेचर में सर्टेनली अफेक्ट करता है, जैसा हमारे ऋषियों में मंत्रों में इसका उल्लेख किया है। अब कल्पना करिए माइक्रोब कहाँ कहाँ मिलेंगे आपके शरीर पे। हाथ में, हैंड्स पे 1000 सेल्स पर सेंटीमीटर स्क्वायर मिलेगा हाथ में, जहां कहते हैं भाग की रेखा। इस भाग की रेखा पे 1 मिलियन सेल्स पर सेंटीमीटर स्क्वायर मिलता है। स्काल्प में 1 मिलियन, नोज म्यूकस में 10 मिलियन, आर्मपिट में 10 मिलियन, सलाइवा में 10 मिलियन, फीकल मटेरियल में 10 मिलियन बैक्टीरियल सेल्स पर ग्राम या पर सेंटीमीटर अलग अलग मिलता है। ये हमने कुल पिक्चर यहाँ पे बना के दिखाया है। लेकिन आपको मैं एक अच्छे ढंग से बताऊं कि हमारे ऋषियों, अभी कोरोना आया तो ऐसा प्रचार किया गया कि कोई चीज छू के ना खाओ, 20-20 मिनट में हाथ धो, सैनिटाइजर यूज करो, ये सब कहा गया ना, टच न करो खाने वाली चीज को।
सॉरी सर, टाइम ओवर हो रहा
हाँ,
है।
टाइम ओवर हो गया, मुझे भी ध्यान है, बस मैं कंक्लूजन की तरफ पहुंच रहा हूँ। ये हमारे शास्त्रों में कहा गया है, लवणम व्यंजनम चैव घृतम तैलय तथा च लेहयम पेयम च विवितम हस्त दत्तम न भक्षयेत। नेकेड हाथ से न खाओ, हाथ धो के खाओ। ये हमारे ऋषियों ने बहुत पहले कह दिया। हम क्या फॉलो करते हैं? आगे कहा, हस्त पादे मुखे चैव पंचाद्रे भोजनम चरेत, हाथ मुंह धो करके तब खाना खाओ। न अप्रक्षालितम पाणी पादो भुंचित, बिना हाथ पैर धोए खाना न खाओ। ये भी चरक संहिता में है। और आगे कहा गया, शवम तत्पृष्ठम, आगे क्या, ये तो मैं आगे बताऊंगा। चिता धूम्र सेवने सर्वे वर्णा स्नानम व आचर्यो। जिस व्यक्ति, अब तो जानते हैं, जो लोग स्मशान घाट पे जाते हैं तो नहा के आते हैं, लेकिन अब ये ट्रेंड बदल गया, कुछ लोग नहीं नहाते, पानी छिड़क लेते हैं, ऐसे ही। और हमारे शास्त्रों में विष्णु स्मृति में कहा गया, चिता धूम्र सेवने, अगर किसी को चिता का धुआं लग गया है, स्नान करके घर में आओ। और इन सब इंफॉर्मेशन को मैंने एक किताब में छापा है, वैदिक माइक्रोबायोलॉजी के नाम से मैंने एक पुस्तक भी लिखा है, हिंदी में और अंग्रेजी में। हिंदी अंग्रेजी दोनों भाषाओं में 2 अलग अलग पब्लिशर ने मेरी वैदिक माइक्रोबायोलॉजी की छापा है। अगर जिसको इंटरेस्ट हो तो वो लोग मंगा सकते हैं। अमेजॉन बेच रहा है, ऑनलाइन भी देख लेंगे कौन कौन बेच रहा है, ये लोग बेच रहे हैं। बहुत बहुत धन्यवाद।
A huge round of applause for such an eminent speaker. Please give him a huge round of applause.
प्रोफेसर दुबे जी का बहुत बहुत धन्यवाद इस वैदिक ज्ञान को हम सब तक पहुंचाने के लिए। उनका जो पिछले 40-50 वर्षों का अनुभव था, उसका सारगर्भित भाषण उन्होंने हम सबके सामने प्रस्तुत किया। उनका बहुत बहुत धन्यवाद।
My request to our session chairs to present a memento to R.C. Dubey sir as a piece of memory from our institution. Please give a huge round of applause to R.C. Dubey sir. Please keep applauding for such a phenomenal personality. Thank you once again, sir. Now, my request to our session chairs to please introduce the next speaker of the event.
Our next speaker, Dr. Ramesh Vaishya ji. Sir is Director, Human Energy Research Center. He is in practice as a chartered accountant for over 50 years. His areas of specializations are international taxation and planning, international finance and cross-border investments. He has previously also served as director in charge of Coopers and Lebrand in New Delhi during 1992 to 95. He also remained to be a member of the Central Council of the Institute of Chartered Accountants of India for 9 years from 1979 to 88. Dr. Vash, please welcome.
ओम, हम सबका सौभाग्य है कि ऐसे रिनाउंड पर्सनैलिटी हमारे साथ हैं। साउंड हीलिंग थेरेपी के विषय में आपको बताने वाले हैं, और अगर आपने इस विधा को अच्छे से समझा तो आज आप लोगों को शायद इनकी तरफ से एक वेव मिल जाए। जैसे मैं एक बहुत बड़ी एक इनकी प्रशंसक हूँ इस चीज के लिए, क्योंकि एक वेव है री एनर्जाइज वेव जो इनके नेतृत्व में डेवलप की गई है, विच इज़ ए साउंड वेव। मैं उसको रोज सुनती हूँ और मैं ये मानती हूँ कि जो एनर्जेटिक हम रह पा रहे हैं या पूरे पूरे दिन और इतने कार्यों का निर्वहन कर पा रहे हैं, तो 2 ही काम शायद ज़्यादा करते हैं, 3 कहेंगे। एक पूज्य स्वामी जी आचार्य श्री का आशीर्वाद, दूसरा लोग आजकल कहते हैं कि मल्टीविटामिन्स लो तब जाकर आप एनर्जेटिक रहते हो, लेकिन मुझे लगता है मल्टीविटामिन्स ना भी लो, ये साउंड वेव आप सुन लो तो आप एनर्जेटिक रहोगे, इतना प्रभाव है इसका। अभी आप सबको पता है कि पतंजलि में बहुत रिसेंट कुछ डेवलपमेंट्स हुए हैं। अभी अरविंद राणा जी जो कि थर्ड आई मेडिटेशन करवाते हैं, वो भी साउंड चिकित्सा के द्वारा छोटे छोटे बच्चे, उनकी पीनियल ग्लैंड जो है वो एक्टिवेट हो गई है और वो देख पा रहे हैं, आंखों के ऊपर पट्टी बांध के वो देख पा रहे हैं। मुझे यकीन नहीं हुआ, बहुत इस चीज की लाइव भी किया गया, बिल्कुल यकीन नहीं हुआ। लेकिन जब मेरे खुद के बच्चे के ऊपर वो प्रयास किया गया तो, और अभी जोशी जी बैठे हैं, आदरणीय डॉक्टर राजेश मिश्रा जी बैठे हैं, उन सबके बच्चे आज इसी श्रेणी में हैं। और एडवांस जो थर्ड आई मेडिटेशन जो है उसके कोर्सेज का हम इसके लिए धन्यवाद करेंगे पूज्य आचार्य श्री का, जिन्होंने ये एक मौका बच्चों को दिया, क्योंकि बच्चों की पीनियल ग्लैंड एक्टिवेट हो सकती है, बड़ों की नहीं, क्योंकि बड़ों में कैल्सिफिकेशन होता है, और वो भी साउंड वेव थेरेपी का एक एग्जाम्पल है। क्योंकि आप सब जानते हैं कि पूज्य आचार्य जी हमेशा नई नई चीज़ों में रहते हैं और नए नए किसी प्रकार के अनुसंधान जो हैं वो करते रहते हैं। इसी श्रेणी में हमारे यहाँ से आदरणीय डॉक्टर उपासना अग्रवाल जी और आदरणीय डॉक्टर प्रशांत कटियार जी, और उनके साथ साथ पतंजलि आयुर्वेद हॉस्पिटल से डॉक्टर देविका जी, 3-4 लोगों की एक ऐसी टीम बनी है जो इसी विधा में रिसर्च करने जा रहे हैं। तो मैं आप सबके जोरदार करतल ध्वनि के बीच हम पुनः आभार व्यक्त करेंगे, अभिनंदन करेंगे, अभिवादन करेंगे आदरणीय डॉक्टर रमेश वैश्य जी का, जो एज तो मैंने सर से पूछी नहीं, लेकिन आप जानते हैं, देख ही पा रहे हैं, इतनी एज के बावजूद भी आप लोगों के बीच इतनी दूर से आए हैं और केवल साउंड वेव थेरेपी, जो इन्होंने एक पूरा का पूरा चमत्कारिक मैं चिकित्सा पद्धति कहूंगी, तो उसके विषय में आपसे आज अपने अनुभव साझा करेंगे। बहुत बहुत धन्यवाद आदरणीय रमेश सर, थैंक यू सो मच।
हम देख रहे हैं कि सर सवेरे से कल भी और आज भी पूरे टाइम यहाँ हॉल में बैठे रहते हैं। हमारे यंग बच्चे कई बार उठ उठ के बाहर आते जाते रहते हैं, लेकिन सर हर लेक्चर को बड़े गंभीरता से यहाँ बैठकर, और मैम भी साथ में बैठकर देख रही हैं, तो सर का बहुत स्वागत है। सर
परम पूज्य स्वामी जी, परम पूज्य आचार्य जी, आपका अभिवादन है। और आपने जो डॉक्टर देव प्रिया ने जो बात कही मेरे लिए, जो शब्द कहे, उसके लिए बहुत बहुत धन्यवाद है। मैं उस लायक तो नहीं हूँ, लेकिन अपने छोटी अकल से, छोटी बुद्धि से जो छोटा सा काम किया है मैंने, वो आपके सामने रखूँगा। और मुझे आपने इजाज़त दी है कि मैं अंग्रेज़ी में भी बोल सकता हूँ, तो ठीक है, क्योंकि इंटरनेशनल कॉन्फ्रेंस है तो ये बाहर भी जाएगा, तो आपकी सबकी अनुमति हो तो मैं अंग्रेजी का ही सहारा लूँगा। देखिए, there is so much of knowledge and information which has flown in this auditorium for the last 3 days. There is so much, such enlightened, such learned, and such experienced scientists and scholars are here, and I have been greatly privileged to be able to speak to such a distinguished audience. Lot has been said about the theory and concepts of Ayurveda and healthcare. Therefore, I don't want to spend any time talking about that. Instead of dealing with the theoretical and conceptual aspects, with your permission, I would like to place before you the practical application of those concepts and that knowledge for one specific purpose. And the one I want to highlight here is this application for the specific purpose of treatment of diabetes type 2. And this has been done by our humble applied research for the last few years on these concepts for the purpose of treating the various ailments in the human body. We have tried to cover a lot of subjects, insomnia, migraine, arthritis, But for the present, I would focus only on one application, that is for diabetes type 2. But before I do that, let me highlight two basic fundamental propositions. One, everything in the universe is in a state of vibration, very subtle vibration. Now, those vibrations are a manifestation of that supreme divine energy which is in circulation all over the cosmos, not only on this earth but all over. And that energy is manifested in various forms, even on earth, for instance, on the all living entities, human, animal, the plants, the forests, trees, the big waves in the ocean, the air which is flowing, light and heat in every form. It is the same ultimate energy that is flowing everywhere. Now, at the cosmic level, if an energy is in motion and it is flowing and getting manifested in a variety of forms, that creates a sound. If there is a vibration, sound is very obviously a natural consequence of that. That sound at the cosmic level has been described by our ancient scriptures and the seers as Brahmanad. The sound at the level of Brahman, the supreme divine force. Now, at our individual level, you may call Jivatma, for instance. We are a small part of the same divine power which we call Brahman. As it's been pointed out in the previous lecture, Vedas have recognized this Aham Brahmasmi. I am the same ultimate Brahman in a miniature form. Tat Tvam Asi, thou art that. So, the manifestation at our individual level, which we are calling Jivatma, is for all purposes a manifestation of the same supreme divine power called Brahman. And therefore, the vibrations which are present at that supreme cosmic level, they are also manifested at our individual level. In other words, the human body is also in a state of vibration. Every organ inside the human body is also vibrating at a certain frequency. They are very diverse frequencies, and depending upon the structure and the function of that organ, nature has designed what those vibrations are, and that is what is happening. Now, the manifestation of those vibrations at the individual level is something that I would like to deal with you. The principle that now I think is well recognized, that every organ in the human body, I'm confining only to the human body, every organ is driven by certain energy. Every organ is energy driven, all systems, not only our own. Every cell in the human body needs a certain energy level in order to function. What is that level? Again, dictated by the nature which designed that. Now, if the energy level falls below the optimum level as contemplated by nature, then the cell will not function, or its functioning will be seriously compromised. Therefore, it is very necessary that the energy level in the human body must be restored to its optimum level in order to let the organs and the cells and the systems function properly. Like in this hall, all the equipment and the systems here are designed to function at the energy level which we call 220 volts. If, for example, the voltage here falls from 220 to 110. None of this is going to work. The equipments will not work, the systems will not work, unless you restore the power back to 220 volts. Something similar is happening to human body also. What is at optimum level? That is a matter for inquiry. Now, if a cell cannot function because the energy level has diminished or declined due to various factors or pollution or environment or for whatever reason, how do we restore the working of the cell back to its optimum energy level? Now, I'll eventually come to the certain organs which are involved, but nature itself has devised and gifted us with a very efficient system of producing the energy inside the human body. If you consider that human body has more than 50 trillion cells, every cell has within it an inbuilt system called mitochondria, which is working for producing the energy and making it possible for the organs to work. Mitochondria absorbs the nutrients which are coming from the bloodstream, proteins and things like that, converts them, mitochondria, that very small mitochondria which is inside every cell. A cell may have one mitochondria or more mitochondria, that converts this nutrient into energy called adenosine triphosphate, ATP for short. Everybody knows here, all of you are student of science and this thing. So the ATP becomes the source for the power. ATP is then transmitted to each organ through the bloodstream, and ATP is the fuel, it is the source, it is the powerhouse for giving the necessary power to a particular organ or a system in order to allow it to function. Now, how do we bring ATP back? Instead of dealing with the cell, let me come straight to the organs which are involved in a problem of diabetes 1 or 2, doesn't matter. Let me just mention only 3 organs which are very evident. 1, the pancreas, 2, the liver, 3, the kidneys. Pancreas have the function of generating insulin apart from other functions, and you know that if the pancreas are not working at the proper capacity, then there is an insulin deficiency or insulin resistance and things like that. And that's why the medical science comes in and you start pumping synthetic artificial insulin into the body. So why would the pancreas not function at the normal natural level? Because their vibrational frequency is not what the nature designed. Research shows, it is a very well-known fact, that if you analyze the vibrational frequency of the organs, then a pancreas is supposed, understood, to be vibrating at 117 hertz. I'm just giving you an example. Liver and kidney around 356 hertz. The short point that I'm trying to convey to you, that each of these organs are working at a certain vibration frequency which is confirmed by tests, and it is no longer a matter of dispute. If a mitochondria is expected to function, vibrate at 117 hertz, and if it is not, we have a lot of equipment we have put together from foreign countries which is very efficient in allowing us to screen each organ in the human body. I will not go into the detail, but the hardware and the software we have got allows us to screen pancreas. If our screening shows, in the case of a diabetic patient, that pancreas in this particular case is not vibrating at 117, but let's say 87 hertz only. Now the obvious thing is, how do we bring 87 hertz back to 117? This is where the sound waves come in. And I'll because all of you are so knowledgeable and such learned people here, I don't have to go into the details. To bring back pancreas to its natural functioning frequency of 117, we use the sound waves. And this vibrational frequency, you see, in the last century, the famous great scientist Nikola Tesla, he summed it up beautifully. If you want to understand the universe, you must understand energy, vibration, and frequency. It's a beautiful something, and now this concept is being recognized. So our sound wave is the way to do it. Totally non-invasive, totally drug-free, no side effect. All we are trying to do is to bring the organ back to its natural level of frequency. How do we do that? Simple. We develop a sound wave at the vibrational frequency of 117, which is not difficult. Sound therapy and other things tell us. Once we have a wave of the frequency which is attuned to the pancreas, this is introduced in the human body through ear, through a headphone and all that. Once it goes inside the system, brain, by a process of entrainment, sends the corresponding signals within the human system, within the body, including the. In effect, what is happening is that this wave has created a vibrational field. Now, the field word is very important, because field theory, I think a lot of you would understand what is a field theory. It creates a vibrational field resonating at 117 hertz. Now, if we sustain this vibrational field, we calculated for 5 to 6 minutes. 5.6 is either, but we sustain it for 6 minutes by continuing the sound wave for that 6 minutes. What is happening? The vibrational field vibrating at 117 hertz is now there continuing. Pancreas, which were sort of sleeping, they wake up. I'm using a very simple language. And then gradually they will come back to 117. Now, this is not what exactly explains the process. What happens is, we should know, is a sympathetic resonance. A resonance in sympathetic with another source of resonance. So pancreas, by a process of sympathetic resonance, gradually come from 87 to 97 to 107 and to 117. Now, this sympathetic resonance is a very interesting phenomenon, and I'll just take a minute to explain. This is a quantum phenomenon. Classical physics did not know about it. They dealt with matter and forces acting on, but quantum physics has gone to the subatomic particle level and studied the behavior and responses of a particle at the subatomic level. And one of the known quantum physics is quantum interconnectivity. That means all particles in the universe, they are interconnected, inextricably interconnected at the quantum level, not at the classical level, not at the superficial level. The other concept which is also playing here is a field theory. If there are two particles in the field, a field, no matter what is the distance, one could be in Delhi, the other could be in New York, but if it is in the same field, then two particles at any point in space, they are interconnected at the quantum level. That's something you must check up, and I'm sure that this is very elementary knowledge about quantum physics. If quantum physics itself is not something you want to look at, it's a different story. So quantum physics predicates that if there are two particles in the same field, then they are inextricably interconnected, and therefore the behavior of the one particle is immediately responded by the other. The experiments, hundreds, thousands of experiments done in quantum physics showed that if there is one particle here, and the properties of particle amongst others are defined by the spin. So if you change the spin of one particle, another particle in the same field immediately responds, makes a corresponding change, so that both of them are spinning in reciprocity. And this interaction between the two particles, let me point out, is not because of a signal going from particle 1 to particle 2. If you study the mathematical equation which describes this interaction between two particles at two different points in space, that equation does not have a time factor at all. There is no t in it. So, because it is instantaneous, it is immediate, and it's a quantum reality. So, in a field there are two particles, they are inextricably interconnected, then one particle behaves in response to the other. Coming back to our issue, if a particle in my 117 hertz field, vibrational field that we have created by introducing the sound wave, if a particle in that field is resonating at 117 hertz, then another particle in the vibrational field of pancreas, which are only 87 hertz, that immediately responds by the principle of sympathetic resonance. No mystery. It is a hardcore quantum physics which is working at work. Now, if my sound wave has created a vibrational field, now you can ask a question. You are talking about a wave, suddenly you are talking about the particle. This principle of quantum physics, which is now well recognized by dozens or thousands of experiments, wave-particle duality. I mentioned one principle of quantum interconnectivity. The other principle, which is now universally recognized in quantum physics, is the wave-particle duality. What it means is this: a wave can behave as a wave, but depending upon the context, it can behave as a particle. There's so many experiments have been done, single slit, double slit experiment and all that. The wave, which is the sound wave, by the process of entrainment triggered by the brain, which is now the vibrational field within the human body, can behave as a particle. So what I am saying is, in simple terms, there is a sound particle, like light is a wave, but light particle we know, we work on that. Similarly, a sound particle in the field which is created by the sound wave inside the human system, that particle and the particle in the other vibrational field, which is the field of the pancreas, they get interconnected because they are in the same field, and the response is well understood. So, same thing happens about liver and about kidney. They are in the range of 356 hertz, but the principle remains the same. So we make a sound wave for 117 hertz to address pancreas. We also make another wave for 356 hertz to address liver, and so on and so forth. There is no limitation to what we can do in terms of sound waves. And let us say we have 6 different sound waves to take care of 6 different frequencies which we need to address. You can combine them, or in parallel, or you can put them in sequence. Sometimes we put them in sequence, one wave after the other, but they work. So all the waves which we are designed, they are based on the principle of quantum physics, well documented, well tested. Apart from the sound wave, to address the 3 organs that I mentioned for the sake of example, we have also looked at Ayurveda. We have great faith, and I personally believe so much in the herbal treatments of Ayurveda. So we looked at Ayurveda, and we found out what are the herbs which are known to be effective in the case of diabetes. There are a long list, but we picked up 4 for the sake of experiment. Berberine, Costus ignis, Vijayasar and Haritaki. Just 4 waves. And what we did, we wanted to find out what are the active ingredients. An active ingredient in one herb could be one or more than one. Like when we did the same exercise with ashwagandha, we found there are 10 active ingredients. Haritaki was mentioned in the one of the sessions here. We examined the active ingredients of Haritaki, and there are 7 different categories we found. There could be more. And there are 7 different components, 7 different active ingredients, each one vibrating at a different frequency, each one having its own qualitative result. So we could have selected only one, but we said so we have picked up all the 7 ingredients, active ingredients of Haritaki, made a wave for each one of them, merged the 7 waves together, and ended up with a wave which we call a sound wave for Haritaki, which is around 352 hertz and something like that. But this integrates the powerful effect of all the 7 components, all the 7 active ingredients of Haritaki. Now, so Ayurveda, we are not stopping, we are continuing. Apart from the sound waves for treating the organs, apart from integrating the benefit of the active ingredients of the herbs, we also looked at the Brahmanad I mentioned at the beginning, the supreme cosmic source which is permeating in the entire universe. The closest approximation to that Brahmanad is Om Dhwani. So we created a sound wave for Om Dhwani and integrated in our main sound wave. Vedic mantra, a very learned speaker told about, and how much is the power of Vedic mantra? And Vedic mantra is what except sound? These were sounds which were researched, investigated and put together in a proper sequence or a formulation or a bandhak. And like Lord Vishwamitra, he made Gayatri, Mahamrityunjay. So we had long list of these Vedic mantras, but we picked up only 2. One is the Gayatri mantra, the second is Mahamrityunjay. Now there are so many experiments have been done to check the efficacy of each of these mantras. A lot of research has been done in IITs. So we took just the guidance from that. The sound wave that we ended up integrates the sound wave for 3 organs, 4 Ayurvedic herbs, 1 spiritual sound and 2 Vedic mantras. And then it was a challenge. We compressed the whole thing, that's a sound engineering. We compressed it, and I'm happy to tell you our sound wave, the integrated sound wave for all this taken together runs only for 19.4 minutes. Let's say 20 minutes. 20 minutes is nothing. A wave running for 20 minutes, anybody who listens to that wave for 20 minutes is simultaneously getting the benefit of all the organs being treated and being restored to their normal level of frequency, the benefit of the herbs, which this audience knows very well, and the benefit of the divine sound Brahmanad, and the benefit of Vedic mantras. So the work has not stopped. There is no end to this. We are continuing with our effort to integrate more and more Ayurvedic herbs. Can you please show the slide? Yeah. Thank you. These are the 9 herbs on which we have already developed the sound wave. And with your kind permission and all humility, Berberine, Costus ignis, which is virtually a replacement for insulin plant, Vijayasar, Haritaki, Ashwagandha, Jatamasi. Now, in the wave with which I was mentioning, we have only the first 4 included in it. Let me give you an example about the berberine. How do we develop this? Berberine is a very well recognized herb for treating diabetes. Let me simplify it. We look at the active ingredient of berberine. Then we go for the molecular weight of that active ingredient. Now, a lot of research is required. Once we have the molecular weight of the active ingredient in berberine, then we develop a sound wave for that. If you go in the first exercise, the sound wave that came up for berberine was 30,000 hertz. It's all experimental work. 30,000 hertz. Now, I can't introduce 30,000 hertz in the human body because it is not safe audible range. So our challenge was to bring down 30,000 hertz to an audible range which is acceptable for the human beings. And this is very interesting again. We bring down by applying the harmonic principle of octave. I've been a student of music in a small way, but let me give you just a simple example. If you take a musical instrument, there is a tar saptak, there is a madhya saptak, and there is a mandra saptak. Any piano, you have 3 different scales. Now, a swar hit on the tar saptak, let's say Gandhar. If ga is played on the tar saptak, you play the same ga on the middle saptak, then you play the same ga on the mandra saptak. They are absolutely harmonically integrated. That's a medical science. But if you bring down, if you play the Gandhar at tar saptak and then you play at the mand saptak, the frequency, vibrational frequency comes down. There's a mathematical formula for that. Then you play in the mandra saptak, the lowest saptak, it further comes down. We applied this knowledge to bring down this 30,000 hertz frequency of berberine by a step-by-step process. It requires a lot of work, and I'm happy to tell you that the berberine, the sound wave for berberine, which we have integrated into our wave for diabetics, is only 501, not 30,000. Same process is applied to the others also. So we go for the full frequency of the active ingredient, but for our purposes will be, and it doesn't compromise the quality of berberine. It only allows us to bring down the vibrational frequency to an audible level, safe for the human being. Now, the work on the herbal, can you show the next slide, please? The results. God is great. And all this that we are doing is pure seva. There's no commercial angle. I've been running this center for almost 8 years now. Acharya gave us the blessings. In December 2015, on the bank of river Ganges, I established the center, probably as an experiment to learn myself to my knowledge, not that I claimed anything. And Acharya was kind enough. He is the one who inaugurated that center in 2015. And it is his blessings, Swamiji's blessings, which are working. Not that we are very capable of doing something, but those blessings somewhere up from the heaven are blessing us. And so the results that I am telling you, there are many. We are dealing with very difficult cases in our own way, like diabetic neuropathy. I'm sure you all know how serious it can be. The feet, the legs you can't stand up, they don't work, you can't walk, and a very serious case of diabetic neuropathy may end up with amputation and things like that. So, one case which I just want to mention to illustrate the point. This was a case of a diabetic neuropathy of a patient about 55, 56 years old. I knew him a little bit. He was suffering from diabetes for 10 years. About 4 years ago, he got caught into diabetic neuropathy, not able to stand up. For 2 to 3 years, he has been totally on a wheelchair. Even if he has to go to toilet, his wife takes him on a wheelchair. The medication that was being pumped into his body by doctors with the best of intentions, I don't blame them, to give you, apart from the other oral medication, metformin. Metformin is the obvious thing. 1000 mg of metformin twice a day. So in one, the normal dose would never exceed more than 500 mg. But in this case, he was being given metformin plus other medicines, metformin 2000 mg per day. Insulin, because systems were not working, 48 units of insulin, it's all in this, 48 units of insulin were being pumped into his body. The reading for the fasting sugar was 220 to 280. They were never came below 220 and they were touching 280 quite often. So I went to him on my way and I young wife, I said, Poonam, do me a favor. I can't claim anything about the way, but here we have something, maybe God will help me on this. Now, she was a wonderful lady. She said, yes, I will do. 21 days, she gave the sound wave twice a day, according to the protocol which we have designed. Not difficult. 20 minutes in the morning, 20 minutes evening, what is there? And with so much of medical thing and diabetes and metformin and insulin, after 21 days, it's on record, we have all properly scientifically validated evidence. After 21 days of listening to that sound wave, gentlemen, I am happy to tell you, his fasting sugar readings came down from 220 to 280 range to 100 and 140. This boy, she keeps on, and you can't imagine the happiness of a young couple to get out of this problem. They had diabetic neuropathy, they had probably reconciled to that. The medicine, I said, talk to your doctor. I don't want to play with your medication, but tell him, very fine doctor, homeopathic doctor, well recognized. He was very surprised. He said, what's going on? It's a miracle. He said, no, nothing, nothing. It's only the sound wave. So we said, you request him to review the medication and the insulin dosage. To cut it short, that 2000 mg of metformin has come down to 500 a day, and the insulin dosage of 48 units is today only 12 units. From 48 to 12. Now, we can't, but this is God's grace. The other very wonderful thing which has happened, which I must mention to you, this fellow was on the wheelchair all the time. So she says, Bhaisha, please come, have a cup of tea. So I stopped by, because after all it's a seva. Then I said, look here, Poonam, I want to take him off this wheelchair. Is it possible? So we came back, made another wave, which was mentioned by Dr. Ved Priya also. I was privileged, she asked for, I gave her that wave, and you must have heard her response to the effect of that wave. 1 week, for 1 week, this fellow continued to take the renal in addition to the diabetes wave. So I said, give him twice a day. So he had 1 wave diabetes in the morning, 1 wave diabetes in the evening, 1 wave re-energized in the morning. It is all sound therapy. Forget about the wave. Gentlemen, after 1 week, he gave up the wheelchair. Today, he is walking slowly with a walking stick. Now, there are other cases that the point I'm making is very simple. Now, there is no conflict for whatever we are doing, but since this conference is for introducing technology, so in my humble submission I said, okay, this is one new technology, maybe everybody knows, but it's high time, so to integrate this. Just can you show the other slides also? Now I just want to make one point before I go to close. You see, we screen this very sincerely. The equipment that I have mentioned to you, which we have imported equipment, which is this equipment has the capability of doing the screening of your organ. This is the screening you see of pancreas. On the left side, the screening is before he started the sound waves. On the right side is the screening 21 days after the sound waves were started. The yellow dots and large dots, these are only for visual purposes to show what is the improvement, but the system is capable of calculating. It stores all this information in the database, but also calculates what is the improvement. And you, if I can see it, the improvement that my computer has recorded and shown is 63% improvement in pancreas. That's what the slide is saying. Show the second one. The liver, because that's there the improvement that the system found out was 18%. Show the third one. Kidneys. The improvement in the kidneys over is 30%. The point I'm making is very simple, that this is not metaphysical, mysterious, some mumbo-jumbo thing, but these are hardcore evidence. And many times we get it corroborated by pathological laboratories. Because like insulin reading, I said, 220 to 280 has come down to 140. Now it is ranging about 120. So I said, no, go to the doctor, HbA1c, under control. Fasting sugar, under control. But he said, this is not enough. You go and get me an external independent evidence from a pathological activity, whether it is lal or dung or whatever, and whatever our system is saying, 87% of it is corroborated by the external pathological reports. To cut it short, gentlemen, what I am saying is that this is our very humble attempt to introduce another technology which, according to my submission, has great potential. And if I also make a humble section, this could be the way to transform Ayurveda from the physical domain to the virtual domain. All this extensive exercise of developing the supply chain management, the manufacturing facilities, the extensive distribution network, or look at the huge empire that Patanjali has created with the blessings of Swamiji. If sound wave is allowed to play its role, then everything moves from physical domain to virtual domain. Just one more point. I think I mentioned the entire effort of this sound therapy that we are working on as an applied research is not to go and shoot the disease. No, nobody is going to shoot the diabetes, nor to kill a bacteria or a virus or anything. There's no sense. The entire effort is to bring the natural system back to its functioning as designed by nature and let the nature function. There's hardly anything I do for diabetes. It's the nature which works. The improvement that we are seeing is because these organs which were not working at their natural level, somehow, with God's grace, your good wishes, they are now working at the natural level. Pancreas is producing the insulin. Why should there be need for medicine and all that? This, I said, takes Ayurveda not only from the physical domain to the virtual domain, but also, with all humility, this is a very befitting response to the call given by Param Pujya Swami ji and Param Pujya Acharya ji, the call to go back to nature. That's what we are trying to do. Thank you.
साउंड वेव दिया कैसे जाता है?
Sound wave, sir, may I just say, music is a sound, mantras are a sound, but the sound wave, the real work is in formulating that sound wave of the desired frequency, of the desired magnitude, of the desired intensity. Now, we recommend that you should listen it through a headphone. The reason is that if you don't use the headphone, that there is so much external disturbance in the atmosphere which compromises the quality of the sound that we want to do. So the simple thing is, the protocol is very simple. We say, please drink a glass of water, please lie down, relax, take 3 deep breaths, and then put on a headphone and listen to that. Now, listening to the sound wave, you don't have to come to my center. Whether somebody is sitting in Ghaziabad or somebody is sitting in Bangalore or somebody is sitting in Calcutta, we say, I'll send you the sound wave. Now, the transmission of the sound wave, because it's a big file, 25 to 42 MBs file. So by transmitting it on an email, it compromises the quality. So what we have done is, we have now devised a technology to load my sound wave on Google Drive and then send the person the link to the Google Drive. You click that and it opens in your system. You can listen it on your laptop, you can listen it on your computer system or desktop or anything. So the technology has taken care. So therefore, people from Bombay, people from Bangalore, they are contacting us and we are delighted to share this knowledge and this facility. So sending the sound wave is entirely our job. To develop and to send it, that's our seva. Now the local, and I must say that 90% of the success that we are getting is because of the wives or the daughters or the ladies. Behind every healthy man, I think there is a woman who monitors, who gives the wave, who tells. So I am very grateful that this situation is. So all I want to say, we had taken no credit. We have no conflict with Ayurveda or the principles, but it's a humble effort to introduce a technology which could transform.
थैंक यू सो मच सर. अभी आप लोगों को अगर पता चला हो तो इसके ड्यूअल इफेक्ट हैं. ड्यूअल इफेक्ट कैसे हैं? क्योंकि साउंड वेव में पीछे से जो साउंड बजती है, आगे से उसमें मंत्र हैं. जैसे गायत्री मंत्र है, त्र्यंबकम यजामहे, महामृत्युंजय मंत्र है, ओम नमः शिवाय वह मंत्र है. तो मंत्र चिकित्सा के विषय में हर आयुर्वेद का विद्यार्थी जो है वो जानता है. और मंत्र चिकित्सा एक वैसे ही आप कह सकते हैं कि बहुत ही अच्छी हमारी ऋषि संस्कृति में चिकित्सा पद्धति उसको बोला जाता है. उसके साथ साथ जो ये साउंड वेव है, क्योंकि मेरा खुद का पर्सनल एक्सपीरियंस भी है, जो अभी रिसेंटली सर ने मेरे को हरिता की वेव दी है. तो शुरुआत में जब वो वेव ली, क्योंकि वो 7 कंपोनेंट्स के मिक्स वेव थी, तो शुरुआत में उस हर्ब की एक स्मेल के साथ जिह्वा पर टेस्ट भी आना शुरू हो गया, जो कि मिरेकल से कम नहीं है. मतलब अगर बिना दवाई खाए आपको जिह्वा पर टेस्ट डेवलप होना शुरू हो जाए, क्योंकि अगर बीइंग अ साइंटिस्ट, बीइंग अ रिसर्चर, हमें कभी कभी ये बातें जो हैं थोड़ी सी ऐसा लगता है कि शायद उतनी साइंटिफिक नहीं है, लेकिन हम लोगों को इस पर और रिसर्च करने की आवश्यकता है. एंड वी आर ब्लेस्ड विद आचार्य श्री, जो कि इन चीज़ों के महत्व को जानते हैं. आदरणीय डॉक्टर देविका जी यहाँ बैठी हैं, डॉक्टर उपासना अग्रवाल जी और डॉक्टर प्रशांत कटियार जी, और उनके साथ एक पूरी साइंटिस्ट की टीम इस कार्य के ऊपर आगे रिसर्च कर रही है. और क्योंकि ये सर का पहला हमारे साथ लेक्चर था, अभी बहुत सारे होने हैं. मैं नमन करती हूँ सर को, सर के डेडिकेशन को, सर के इंस्पिरेशन को. और इस एज में भी जिस ऑनेस्टी के साथ, जिस शिद्दत के साथ, और मैं कह सकती हूँ कि सुबह से आप लोग यहाँ बैठे हैं, आप सब इच्छा मना रहे हैं कब हम छूटे और जाएं, इजंट इट? लेकिन आप सर की एज देखिए और उनका डेडिकेशन देखिए और उनका हर वक्ता को उन्होंने बहुत अच्छे से सुना है। हम सर आपको प्रणाम करते हैं। हम बहुत छोटे हैं आपसे, लेकिन जरूर हम परमात्मा से प्रार्थना करते हैं कि जब हम आपकी एज में पहुंचे तो हम इतना अपने अंदर एक तारतम्य बना सके। तो मैं इस मंच से सर आपको प्रणाम करती हूं और हम आगे इस कार्य पे जैसे भी पूज्य आचार्य श्री के दिशा निर्देश होते हैं आगे हम कार्य करेंगे। अब मैं चाहूंगी।
डॉक्टर साहब एक मिनट।
जी सर।
ये साउंड वेव मिलेगी कहाँ, इसका कुछ लिंक वगैरह?
सर कुछ मेरे पास मिल जाएंगे, मैं दे दूंगी। 4 वेव मेरे पास हैं जो भी सर ने शेयर करी हैं।
अच्छा। इसमें कोई ऑर्गन डिजीज से रिलेटेड है अलग?
ये बहुत जरूरी सवाल है। साउंड वेव बना के, कंसेप्ट बना के हम घर बैठ जाएं, रिसर्च पेपर पब्लिश कर दें, नाम अपना छपवा दें, वो नहीं चाहिए। हमारा प्रयास ये है कि सेवा का काम अगर हाथ में लिया है तो कितने लोगों को, लार्जेस्ट नंबर ऑफ पीपल विद द लार्जेस्ट बेनिफिट। तो उसके लिए मैंने आपको बताया कि साउंड वेव की हमने टेक्नोलॉजी बना दी है भेजने की। आप कहीं भी हो, चाहे आप हरिद्वार में हो, चाहे देहरादून में हो, चाहे कहीं भी हो, साउंड वेव पहुंचाना हमारी जिम्मेदारी है। वो आपके तक पहुंच जाएगी। अब आपको सुने, उसको फॉलो करें, यही हमारी आशा है। और ये अभी हमने प्रयास किया है, आचार्य जी के बड़ी कृपा हमारे ऊपर है। मतलब मैं यहाँ बैठा क्यों, मुझे तो कोई, मेरा कोई स्थान ही नहीं है। तो अगर उनका आशीर्वाद हुआ और वो ये समझेंगे कि दिस इज़ ए लॉजिकल एक्सटेंशन ऑफ द आयुर्वेदा, साइंस ऑफ आयुर्वेदा, बाय इंट्रोड्यूसिंग an integrated approach for treatment of the organs as well as the herbs, as well as the spiritual sound, as well as the Vedic mantra. तो हम डॉक्टर वेद प्रिया जी से प्रार्थना करेंगे और कहेंगे ये जिम्मेदारी साउंड वेव हम लोग बना रहे हैं और बन रही हैं, बहुत सी बन रही हैं, वो छोड़िए, आप इन तक पहुंचाना हमारी जिम्मेदारी है।
डॉक्टर साहब एक सवाल हो रहा है, इसमें हर एक बॉडी की क्या साउंड वेव अलग नहीं होगी?
क्या
हर एक ह्यूमन बीइंग्स में हर एक बॉडी की एक साउंड अलग होगी?
ये देखिए सवाल,
तो उसमें
आप लोग इतने नॉलेजेबल हैं, इतने ज्ञाता हैं, मैं आपके सवाल
सर इसमें क्या है, अभी प्रिंसिपल सर मैं बताती हूँ, इसमें अभी हमें शोध करना है। ये बहुत प्राइमरी स्टेज पर हम कह सकते हैं। और हम लोगों ने पूज्य आचार्य श्री के निर्देशन में कुछ केस स्टडीज प्लान की हैं जिसके अनुसार अभी कार्य करेंगे और रिसेंटली शेयर करेंगे। अभी क्योंकि पूज्य आचार्य श्री आदरणीय सर का वेट कर रहे हैं तो हम चाहेंगे कि एक बार एक कॉमन जो फेलिसिटेशन है हमारी तरफ से।
सर का बहुत बहुत धन्यवाद।
तो आदरणीय डॉक्टर कुणाल भट्टाचार्य जी और अनुपम सर से मैं रिक्वेस्ट करूंगी। मैम आप भी ज्वाइन कीजिए प्लीज। तो जैसे कहते हैं कि हर सफल व्यक्ति के पीछे महिला का हाथ होता है, तो हमारी मैम है जो सर के इस उम्र में भी इतने डेडिकेशन से कार्य करने के पीछे उत्तरदायी हैं। उनके लिए भी बिग राउंड ऑफ अप्लॉज। बहुत बहुत धन्यवाद सर। अब मेरी रिक्वेस्ट हमारे क्रू मेंबर्स से, जो जया एंड चिराग जी, कि आप सर को नीचे तक छोड़ के आए। मैम आप भी, आचार्य जी वेट कर रहे हैं। तो बहुत सारे इस प्रकार के कार्य अभी पतंजलि में चल रहे हैं। इस कॉन्फ्रेंस में उसको लाने का या उसके आपके सामने प्रेजेंट करने का एक उद्देश्य है कि आयुर्वेदा विद मॉडर्न टेक्नोलॉजी, और ये वह टेक्नोलॉजी कहें या वह साइंस कहें जिसके ऊपर अभी हमें और कार्य करने की आवश्यकता है, और वो हमारी सक्षम जो टीम है वो कर रही है केस स्टडीज के रूप में। नाउ आई इनवाइट डॉक्टर रश्मि मित्तल जी फॉर फर्दर प्रोसीडिंग्स।
Our next speaker is Professor Balram Ji Umar. Sir is professor in Department of Microbiology in AIIMS Rishikesh. He has been awarded several prestigious awards. He has pursued several professional diploma, such as in clinical research, pharmacovigilance, and many more. He has published several research papers in high-impact journals and having ResearchGate interest of 280.4%. He is also leading projects as PI and co-guide of several projects. I welcome Professor Balram.
कर लेता सो ये क्या टाइमिंग से मुझे लग रहा है कि आपने इसके साथ टाइमिंग डाल दी टाइमिंग ये अपने आप से रन हो रहा है अपने आप ही रन कर रहा है बैक करके फिर से खोलो मैं वो स्लाइड बंद करता हूँ एक बार ऐसे ही खोलो बस ये तो पीडीएफ बना दूँ उसको हाँ पीडीएफ नहीं बस ये डुप्लीकेट कर सर ठीक है कोई बात रन हो रहा है ये रन क्यों हो रहा है मैं इसका पीडीएफ बना के रन करता हूँ एक सेकंड दीजिए टाइमर लगा है तो इसका पीडीएफ बना के मैं तुरंत से प्रॉब्लम हो जाएगा पीडीएफ के थ्रू रन कर लेंगे should have to be navigate using down and up key. एनिमेशन चला जाएगा मेरा सो सर प्रथम मैं तो स्वामी जी का और आचार्य जी का आभार प्रकट करता हूँ साथ में ऑर्गेनाइजिंग कमेटी का भी आभार प्रकट करता हूँ कि मुझे यहाँ बोलने का अवसर दिया और चेयर का भी अभिवादन करता हूँ मैं जो मेरा टॉपिक है आज का दैट इज रोल ऑफ हैवी मेटल्स आर्सेनिक इन एनहांसिंग एंटीबायोटिक रेजिस्टेंट जीन ट्रांसफर फ्रॉम मल्टी ड्रग रेजिस्टेंट क्लिनिकल स्ट्रेन्स टू एनवायरमेंटल स्ट्रेन्स। तो मैं बोलना चाहता हूँ मेरा जो कोर टॉपिक है मैं बेसिकली अ क्लिनिकल माइक्रोबायोलॉजिस्ट हूँ। मैं हार्ड कोर रिसर्चर नहीं हूँ, मैं क्लिनिक्स में ज़्यादा क्लिनिकल माइक्रोबायोलॉजी में ज़्यादा इन्वॉल्व हूँ। तो वाई आई चोज दिस टॉपिक बिकॉज एज एवरीबडी नोज दैट कि अभी हमारा एंटी माइक्रोबियल रेजिस्टेंस का मिनास पूरे ग्लोबल नॉट ओनली इन इंडिया पूरे ग्लोबल में इसकी बात हो रही है और हमारा वो हमको भी हॉन्ट करता है, क्लिनिकल साइड वालों को भी हॉन्ट करता है और जो इकोनॉमी है उसको भी हॉन्ट करता है। कहीं ना कहीं एंटी माइक्रोबियल रेजिस्टेंस से मरीज भी अफेक्टेड है, बहुत लंबा समय तक उसको स्टे करना पड़ता है, उसके पास इकोनॉमिकल बर्डन पड़ जाता है और इनडायरेक्टली सरकार पे भी बोझ पड़ता है. सो डेवलपिंग एंटीमाइक्रोबियल रेजिस्टेंस इज द होलिस्टिक अप्रोच, द रोड अहेड. क्या अगर हम सभी चीज़ों को समाहित कर दें, सारी विधाओं को समाहित कर दें, तो क्या हमको इसका आंसर मिल सकता है? सो दिस विल बी माई फ्लो. द प्रॉब्लम ऑफ राइजिंग एंटीमाइक्रोबियल रेजिस्टेंस, द global awakening and the concern, the commitment of government of India, some data from the AIIMS Rishikesh, and what we can do and what is the holistic approach we can take. The problem is not new. The problem Alexander Fleming ने भी 1945 में उनको समझ में आ गया था कुछ गड़बड़ है. जब उनको पता चला कि जब हम पेनिसिलिन दे रहे हैं तो अगर उसका छोटा डोज देंगे या कम डोज देंगे, राइट डोज नहीं देंगे, राइट पेशेंट चूज नहीं करेंगे, राइट डायरेक्शन नहीं यूज़ करेंगे, राइट रूट चूज नहीं करेंगे, राइट टाइम नहीं यूज़ करेंगे, कब उसको ख़त्म करना है, कब उसको वो करना है, तो ऐसे बैक्टीरिया में रेजिस्टेंस क्रिएट हो जाएगी. और जब वो रेजिस्टेंस क्रिएट हो जाएगी तो अंत में वो बाकी जो अनरेजिस्टेंट या नॉन रेजिस्टेंट ऑर्गेनिज्म है उनमें भी फैलती जाएगी और we will have a problem in the long run. So the emergence of multi-drug resistant bacteria is currently posing a significant threat to the global public health, and the plasmids which are involved for the gene transfer are a critical source of genetic diversity in bacteria, contributing to adaptation and diversification of the prokaryotes. Prokaryote मतलब bacteria और छोटे organisms, and which is the driving force behind the development of multi-drug resistant MDRs and increased virulence of the healthcare associated bacterial infection. अभी कम्युनिटी से मल्टी ड्रग रेजिस्टेंस कम आता है, पर जब हॉस्पिटल में एक बार मरीज एडमिट हो जाता है तो उस एनवायरनमेंट में बहुत सारे बैक्टीरिया अपने आप ही, जो कि एनवायरमेंटल बैक्टीरिया हैं, जो कि नॉर्मली पैथोजेनिक नहीं होते हैं, वो भी पैथोजेनिक हो जाते हैं और मल्टी ड्रग रेजिस्टेंस जीन उनके अंदर ज़्यादा होती हैं. वो बैक्टीरिया एक बार अंदर चले गए मरीज के तो बहुत परेशानी से वो बहुत परेशान करते हैं और बहुत सारी एंटीबायोटिक्स वर्क नहीं कर पाती. एंड एज ऑल वी नो दैट वी आर कंप्लीटिंग 3 years ऑफ कोविड-19 पैंडमिक. एक बहुत ही खराब स्थिति से थोड़ा बाहर निकले हैं, पर अभी भी हमको मालूम नहीं कि हम कहाँ स्टैंड करते हैं. वी हैव सीन सारे रिसोर्सेज यूज़ हो गए, हॉस्पिटल सारे भर गए, सारे डॉक्टर्स काम के बोझ से परेशान हो गए, सारे थर्ड लाइन वर्कर, सेकंड लाइन वर्कर, फर्स्ट लाइन वर्कर सब परेशान थे. एंड कुछ नहीं मालूम था किसको क्या देना है. वायरस था, वायरस के लिए कोई दवाई नहीं थी, पर इन्फेक्शन, सेकेंडरी बैक्टीरियल इन्फेक्शन हो रहे थे, सेकेंडरी फंगल इन्फेक्शन हो रहे थे, तो हमने इनडिस्क्रिमिनेटली एंटीबायोटिक्स का यूज़ किया. और अगर उस समय का डाटा बहुत ज़्यादा क्रिएट नहीं किया जा सका क्योंकि सब चीज़ बंद थी, तो एंटीमाइक्रोबियल रेजिस्टेंस का जो उत्पाद था वो और ज़्यादा इंक्रीज हो गया. सो एक डेटा है सेक्सुअली ट्रांसमिटेड डिजीजेस इन्फेक्शन का, उसमें देखा गया कि गनोरिया का जो रेजिस्टेंस है वो 80% इंक्रीज कर गया, 70 to 80%, एंड अबाउट 83 million न्यू केसेस वर देयर फॉर द रेजिस्टेंट गनोरिया. इसीलिए ग्लोबली वी सेलिब्रेट और वी ऑब्जर्व, नॉट सेलिब्रेट, इट्स नथिंग टू सेलिब्रेट, वी ऑब्जर्व द वर्ल्ड एंटीमाइक्रोबियल अवेयरनेस वीक, केवल इसलिए कि ऐसी मल्टी ड्रग रेजिस्टेंस या रेजिस्टेंस के लिए जो डेवलपमेंट हो रहा है उसके लिए एक इंफॉर्मेशन जाए पब्लिक में, सरकार में, ब्यूरोक्रेट्स में, सबको पता चले कि हो क्या रहा है और इससे आने वाली परेशानी क्या होगी. जब पेनिसिलिन की खोज हुई थी 1950 के अराउंड, तो जो निमोनिया वाले पेशेंट थे देर reduced from 25% from 80% they reduced to 25%. एंटीबायोटिक आ गई थी, उसके पहले जितने भी निमोनिया वाले पेशेंट होते थे ज़्यादातर सकम कर जाते थे, मर जाते थे, राइट. पर आज की डेट में क्या हुआ, कुछ रिपोर्ट्स हैं जैसे टंजानिया में due to antibiotic resistance, neonatal septicemia has increased to a very high level from 20% to 70%. यूरोपियन यूनियन के देशों में about 25,000 पेशेंट डाई पर मंथ, और इसका बोझ, आर्थिक बोझ कितना पड़ रहा है, इकोनॉमिकल बर्डन, you can see at least 1.5 billion euro per year. That is a huge amount of money which could have been spent on some other things. अब different years में different antibiotics खोज हुई. You can see in 1928 में फ्लेमिंग ने क्या खोजा था? Anybody? पेनिसिलिन की खोज हुई, राइट? 41 में इसका यूज बढ़ा, राइट? पर 40 में ही उसके अगेंस्ट में रेजिस्टेंस आने लगी. पेनिसिलिनेज एंजाइम ने रेजिस्टेंस क्रिएट किया. फिर इसी तरह 60s में, 58 में उस एंजाइम के अगेंस्ट में बीटा लैक्टेमेजेस एंटीबायोटिक और डिफरेंट टाइप की आविष्कार हुई. बट धीमे धीमे इन 1965 यू गॉट अ बीटा लैक्टेमेजेस ग्रुप ऑफ एंजाइम, एंटी बीटा लैक्टेमेजेस बनने लगे. वो और जो एंटीबायोटिक थे उनको डिस्ट्रॉय करने लगे. फिर और आगे खोज करी गई तो सिफेलोस्पोरिन्स डेवलप हुई. सिफेलोस्पोरिन्स के बाद थर्ड जनरेशन सिफेलोस्पोरिन भी डेवलप हुई. एंड बाय द टाइम इनको भी कॉम्बैट करने के लिए बैक्टीरिया ने सीख लिया कि भाई इनसे भी बचना है एंटीबायोटिक से. तो उसने कहा कि चलो मैं कार्बापेनेमेजेस बनाता हूँ. तो उसने मेटालो बीटा लैक्टेमेजेस का अपना वो स्टार्ट कर दिया, कार्बापेनेम्स. उसके बाद इमीपेनम आया, इसको कॉम्बैट करने के लिए इमीपेनम हायर ग्रुप ऑफ एंटीबायोटिक आई. उसने कहा चलो तुम ठीक है. फिर एज यू नो कि देयर वाज अ एंजाइम विच वाज कॉल्ड एज NDM1, न्यू दिल्ली मेटालो बीटा लैक्टेमेजेस. उसको भी खत्म कर, उससे भी परेशान हो गए. पूरे वर्ल्ड में एक भारत का नाम बदनाम करने की साजिश हुई उस समय कि भारत में ये एंजाइम डेवलप करी थी, कहीं और से, स्वीडन से थी वो. पर उसके बाद 2005 में टाइकीसाइक्लिन आई, कुछ राहत मिली. पर आज की डेट में जो परसेंटेज ऑफ जो परसेंटेज ऑफ MDRs थे वो 5-6% होते थे. अब की डेट में 25% तक MDR बैक्टीरिया मिलते हैं. सो आर बैक्टीरिया मोर स्मार्टर देन अस? हर एक को सर्वाइवल चाहिए, है ना? जहां आप किसी को परेशान करोगे तो अपने उसको परेशानी से बचने के लिए वो अपने मैकेनिज्म डेवलप कर लेगा. उस मैकेनिज्म को जानने की बहुत आवश्यकता है. सो एज दे टुक बर्थ, व्हाई? बिकॉज as we were the keen discoverer, they were more influential. They were changing their pattern of resistance against the antibiotics. The more we use the antibiotics, the more they develop the resistance. और कारण वही था, गलत डोज, कम डोज, ओवर द काउंटर प्रोडक्शंस, है ना? सही मात्रा में ना देना, सही रूट से ना देना, सही पेशेंट को ना देना, सही डिजीज की डायग्नोसिस ना करना, ये सब कारण थे. देयर वाज अ रिपोर्ट फ्रॉम IJMR फ्रॉम 2011 जिसमें हम इंडियन नेशनल लेवल की बात करते हैं कि इट सेज दैट देयर इज द हाईएस्ट बर्डन ऑफ बैक्टीरियल डिजीज इन द वर्ल्ड विच इज इन इंडिया. और एक चीज और देखिए कि सिफेलोस्पोरिन का यूज was increased by 40% to 60%. तब बर्डन भी बढ़ा और एक पर्टिकुलर टाइप की एंटीबायोटिक जो बाकी पुरानी वाली काम नहीं कर रही थी, तो इसका यूज कितना बढ़ गया. उस यूज की वजह से इकोनॉमिकल बर्डन बढ़ा, है ना? बैक्टीरिया के ऊपर सिलेक्शन प्रेशर बढ़ा कि वो अपने आप को और रेस्ट करे, राइट? बहुत सारी ऐसी डिजीजेस थी जैसे कि URTI, अपर रेस्पिरेटरी ट्रैक्ट इन्फेक्शन या डायरिया, जिसमें एंटीबायोटिक की जरूरत नहीं होती. दे वर गिवन द ड्रग्स बाय द फार्मास्यूटिकल पीपल, मे बी फार्मेसी, मे बी सम ओवर द काउंटर परचेज जो होती है उसमें वो देते रहते थे. एक अनपब्लिश्ड डाटा है SGPGI लखनऊ का, जिसमें कह रहा था कि वो हर मंथ 35 लाख रुपीस ओवर एक्सपेंडिचर कर रहे थे ऑन एंटीबायोटिक्स। पिछले डाटा से उन्होंने कंपैरिजन किया। सो एज यू सी कि जो कंडीशन है, जो मीनस है, इट्स अ टिप ऑफ आइसबर्ग, छुपी है, बहुत ज़्यादा छुपा हुआ है जो हमको मालूम नहीं है। छुपा क्यों है? क्योंकि हम तो केवल इंस्टीट्यूशनल लेवल पे देख रहे हैं, हम क्लिनिक के लेवल पे नहीं देख रहे हैं, हम दूसरे अलाइड साइंसेस के पर्सपेक्टिव से नहीं देख रहे हैं, ओवर द काउंटर प्रोडक्शन नहीं देख रहे हैं। वो वहाँ से पता ही नहीं चलता है कि कौन MDR में जी रहा है। सो हु इज रिस्पॉन्सिबल? As a clinical microbiologist, I am. As a researcher, I can be. Who is manipulating the bacteria? क्या हुआ कोविड के साथ? कहाँ से आया? लैब से। लैब ने मैनिपुलेट किया, उस पर एक्सपेरिमेंट कर रहा था, निकल गया, भाग गया। इट कैन बी, यहाँ पर यू का मतलब है प्रिस्क्राइबर, राइट? जो प्रिस्क्राइब कर रहा है, अगर उसको पूरी नॉलेज नहीं है, सही डिजीज के लिए सही ड्रग नहीं दे रहा है, तो ही इज रिस्पॉन्सिबल। एंड सम अनरजिस्टर्ड प्रिस्क्राइबर्स लाइक फार्मास्यूटिकल्स, फार्मेसी मतलब मेडिकल स्टोर्स, veterinary people, they don't know exact, they are giving for the good of their animals, पर उनको राइट डोज का एविडेंस नहीं है उनके पास, कितनी देनी है, कौन सी ड्रग देनी है। फूड इंडस्ट्री में फंगस या बैक्टीरिया से फूड आइटम्स को बचाने के लिए कुछ एडिशंस होते हैं, इट इज़ एडेड विच मे बी इन अ लो डोज, और हम उसको खा रहे हैं, तो इट विल, व्हाट विल हैपन, जो हमारे गट के बैक्टीरिया हैं या पैथोजेनिक बैक्टीरिया, उस लो डोज को लेके दे विल बिकम रेजिस्टेंस। और सबसे बड़ी बात है इंटरनेट मेडिकेशन, सेल्फ मेडिकेशन। गए और देखा कि भाई गला खराब है, ले लो एजिथ्रोमाइसिन, होता है ना, कॉमन प्रैक्टिस। सो व्हाट वी हैव टू डू, वी हैव टू फाइंड अ सोल्यूशन टू इट। व्हाई वी हैव टू फाइंड अ सोल्यूशन? बिकॉज एक लिमिटेशन है डिस्कवरर की भी, मैन्युफैक्चरर की भी एक लिमिटेशन है, कितने मॉलिक्यूल्स निकालोगे। बायोइन्फॉर्मेटिक्स वाला मॉलिक्यूल निकालेगा, उसके बाद तुम चेंज करोगे, उसके बाद इट विल टेक सेवरल ईयर्स टू प्रोड्यूस अ न्यू मॉलिक्यूल, राइट? तब तक strains are increasing, number of new antibiotics discovery is not in that proportion. So apart from the societal cost, increase in the societal cost, it becomes a global challenge. So we need to do a joint initiative. So government of India also took the lead in that. In 2011-2015 they developed the gap AMR. They developed a document. They wanted to find a solution to it कि भाई कैसे डील करें, एक कॉमन पॉलिसी बना दो कि इस पर्टिकुलर चीज़ में इस तरह का मरीज आएगा तो इसको लोअर लेवल की या लो रेंज वाली एंटीबायोटिक दोगे। अच्छा, नॉट इन इंडिया, इट वॉज फर्स्ट टाइम इन 2015 बराक ओबामा जब प्रेसिडेंट थे, उन्होंने व्हाइट हाउस से भी डॉक्यूमेंट निकाला AMR के लिए। ही डिड अ NAP प्लान, नेशनल एक्शन प्लान, जिसमें इनिशिएटिव लिया कि 2020 तक एंटी माइक्रोबियल रेजिस्टेंस पे कुछ काम हो। फाइनली देयर वाज अनदर डॉक्यूमेंट दैट वाज कॉल्ड एज NAP AMR which was released in 2017, and by 2021 they have identified some objects like improving awareness and understanding AMR through effective communication, education and training, strengthening knowledge and evidence through surveillance, reducing the incidence of infection through effective infection prevention and control, optimizing the use of antimicrobial agent in health, animals and food, promoting investment for AMR activities, research and innovation. उन्होंने एक तो रिसर्च में पैसा फंडिंग स्टार्ट करी, राइट? हेल्थ केयर सेटिंग्स में हॉस्पिटल इन्फेक्शन कंट्रोल कमिटीज़ बनाई गई, तो एक इनिशिएटिव लिया कि कैसे, किस तरीके से इसको कंट्रोल किया जाए। अच्छा, प्रॉब्लम ये बहुत स्मॉल सा डाटा है 2 मंथ्स का, जिसमें 950 आइसोलेट लिए, और व्हाट वी फाउंड कि अगर हम 950 सैंपल्स लेते हैं तो हमारे बैक्टीरिया उसमें से आधे 50% बैक्टीरिया आइसोलेट्स होता है, राइट, और 50% में भी करीब 10% से लेकर 12% आर रेजिस्टेंट बैक्टीरिया, ठीक है। तो मतलब कि ऑलमोस्ट 20% ऑफ द आइसोलेटेड स्पीशीज वर MDR, तो इट इज अ ह्यूज अमाउंट। और सब तरह के बैक्टीरिया, नॉट ओनली इन वन टाइप ऑफ स्पीशीज, वन टाइप ऑफ बैक्टीरिया, वन टाइप ऑफ जेनरे, all of the bacteria were showing some or other type of resistant. So this is a bookish slide, theoretical slide, कि resistance होती कैसे है. It may be due to enzymatic inactivation, altered receptor or altered antibiotic transport. So again, what we have to do as a clinical microbiologist, मेरा फर्ज क्या है कि मैं अपनी रिपोर्ट, पहले तो हम जब एक बैक्टीरिया, किसी सैंपल से बैक्टीरिया आइसोलेट कर रहे हैं तो हमें ये क्षमता हो कि ये पैथोजेनिक है कि कमेंसल है. बहुत सारे ऐसे बैक्टीरिया जो ऑलरेडी उस सैंपल में जहाँ से साइट है, जैसे हम गले में स्ट्रेप्टोकोकस निमोनी एक कमेंसल होता है, पर अगर वो वही लंग से आइसोलेट होता है या स्पूटम से आइसोलेट होता है तो वो पैथोजेनिक होता है. अगर हम थ्रोट स्वाब ले रहे हैं और उसमें स्ट्रेप्टोकोकस निमोनी निकाल के दे रहे हैं तो उसमें एंटीबायोटिक की कोई ज़रूरत नहीं होगी. अगर स्टूल कल्चर है उसमें हम इश्रीचिया कोलाई निकाल के दे रहे हैं तो उसकी कोई वैल्यू नहीं है, क्योंकि वो ऑलरेडी एग्जिस्ट करते हैं वहाँ पर. स्किन में अगर स्टेफलोकोकस ऑरियस की जगह एपिडर्मिडिस या कमेंसल्स वाले आइसोलेट करके हम उसकी रिपोर्ट बना कर दे दें तो इट्स अ यूजलेस थिंग, क्योंकि वो पैथोजेनिक है ही नहीं वहाँ पर. तो व्हाट वी हैव टू we have to correctly isolate and identify the organisms. उसकी जो सेंसिटिविटी लगाते हैं हम लोग, एंटीबायोटिक सेंसिटिविटी, वो कौन सी ड्रग देनी है या नहीं देनी है, बतानी है कि नहीं बतानी है. अगर हमने एक वुंड इन्फेक्शन से स्टेफ ऑरियस को आइसोलेट किया, ठीक है, और हमने उससे कहा कि तुम इसको हमने रिपोर्ट में n नंबर ऑफ ड्रग दे दी, उसमें वैंकोमाइसिन भी दी, तो क्लिनिशियन क्या होगा, वो वैंकोमाइसिन चढ़ाना चालू कर देगा. ठीक है, तो हमको कास्केड रिपोर्टिंग करनी पड़ती है कि हम बताते हैं कि भाई नहीं, पहले आप एम्पेसिलिन, अमोक्सिसिलिन या लोअर रेंज ऑफ ड्रग्स दो पहले. तो हम उन जो हायर ग्रुप ऑफ रेंज ऑफ ड्रग्स होते हैं उनको हटा देते हैं, छुपा देते हैं. तो दिस इज कॉल्ड कास्केड रिपोर्टिंग. दिस इज द वर्क ऑफ क्लिनिकल माइक्रोबायोलॉजिस्ट. कुछ ऐसे बैक्टीरिया होते हैं जो पर्टिकुलर एंटीबायोटिक के लिए इंट्रिंसिकली रेजिस्टेंट होता है, ठीक है, उसमें काम ही नहीं करेगा, राइट. तो वो भी बताने होते हैं कि भाई तुम इसमें अमाइनो ग्लाइकोसाइड मत दे देना, माइकासिन मत दे देना, कि उसमें काम नहीं करेगी. सो वी हैव टू राइटली इंटरप्रेट एंड इम्प्लीमेंट द थॉट्स ऑल्सो. एंड फाइनली वी डू हैव टू डू अ रिसर्च फॉर एवरीथिंग. So now I will call Mr. Ashish, my research scholar, to elaborate various studies which are going on and which have been done at AIIMS Rishikesh. Ashish.
Thank you, sir. Good evening, everybody. This is Ashish. As sir told, the antimicrobial resistant. So what is the antimicrobial resistant? So what is antibiotic resistant? How it created by the bacteria? So as we all know, the antibiotic resistant, created by the different mechanism by the bacteria. One of the mechanism is enzyme production. So by the various type of enzyme are there in bacterial cell wall, and sometimes they produce a diffusible, like sometimes they produce from the cell, sometimes they produce inside the cells. So once we giving the antibiotic to the bloodstream, so those enzyme bind to the antibiotic and destroy that antibiotic molecules. So one of the important factors to create such kind of generate such kind of enzymes, like diffusible, non-diffusible enzymes, the plasmid play a major role in this. So in one of our study, what we did, we selected the environmental samples from the different environmental location according to the human interference, human interference on those particular locations. Like we took roadside samples where the human interference was very high. We took agricultural land where the human interference was, compared less than roadside, like interaction of the humans. We took core area of jungles where the human interference is not restricted at that place. We took samples from where the heavy metal concentration is very high, like Sukratal, like Gangotri. In Sukratal there was arsenic level was very high. In Gangotri there was mercury level were very high. So we isolated those bacteria from the different geographical locations as we shows in diagrammatic representation. So what we did, we performed the antibiotic susceptibility pattern with those environmental bacteria, which was recipient bacteria, and those clinical bacteria, which was donor bacteria. The donor bacteria have a plasmid, a circular DNA molecule, which have a gene to transmit through the through the environmental through the clinical bacteria to the environment bacteria. So as you can see in the heat map, those are antibiotic given in this heat map. Those all are resistant to that clinical bacteria. But in environmental samples, as you can see, some green color coded antibiotic only resisted. Rest of was sensitive to those environmental bacteria. So what we did, we again checked the plasmid stability inside that bacterial colonies, bacterial colonies. So what we find, those in clinical stain having lot number of plasmid mediated gene encoded in that cell wall inside the cell. As we seen that in the environmental bacteria, there was no plasmid, plasmid circular DNA molecule was seen in those environmental bacteria. So what we did, we perform, what we perform horizontal gene transfer in presence of antibiotics, in presence of heavy metals, and in presence of without antibiotic. What we seen when we perform the experiment without the antibiotics. So we seen there was horizontal gene transfer. The plasmid mediated gene was transferred to the from the clinical stains to the environmental stain was very high. The frequency was very low as compared to the experiment we perform in presence of heavy metals, different concentration of heavy metals and different concentration of antibiotic. As you can see, you can see in this diagrammatic representation that X number of denoted that X number of concentration is the minimal inhibitory concentration of bacteria from the zero to the high to the minimal. That minimal concentration is called in which the bacteria is died at the surface level of the culture media. So we perform that horizontal gene transfer with the different antibiotic concentration, with different arsenic concentration, and in this scenario, what we find that in the minimal inhibitory concentration below the concentration of some minimal inhibitory concentration, the horizontal gene transfer was very high as compared to without antibiotics. So again, what we did, we accepted selected the patient who were consuming the water samples from the high arsenic level of area, high arsenic level water source areas, and we tested the heavy metal from the blood samples of those patients. We find the concentration of heavy metal was very high in the Sukratal as compared to other locations. So what we did, we selected those patients who were the infected patients admitted the nearby hospitals, and we isolated the bacteria from those patient samples. Like sample was the blood and sputum and so on, and we perform the antibiotic susceptibility pattern compare with other location bacteria. So what we find, the Sukratal patient who directly consuming those water sources which contain high levels of arsenic as compared to other locations, the antibiotic resistant pattern was very high to those patients as compared to the location having less amount of heavy metal in the drinking water samples. So as you know, the pigmentations also, the pigment production, some bacteria produce different type of pigment, like Pseudomonas aeruginosa produce 4 different type of pigment, like pyocyanin, pyoverdine, pyomiranin, that green pigment, yellow pigment, blue pigment, red pigment. So these pigments are very crucial in virulence factor till now. Till now the literature was suggested that these pigments play a very important role to create virulency in the bacteria. But one of the research
I'm
what
sorry,
we find...
we are almost reaching the time. We are at the other time actually.
So what we find that the different pigmented bacteria play a very important role in antibiotic resistant mechanism. Like as you can see, the yellow pigmented bacteria, which called pyoverdine, is very important in clinical stain Pseudomonas aeruginosa was very highly resistant to the antibiotic and to the different mechanism of antibiotic resistant, like beta lactamase producing. These are the enzymes which produced by bacteria, and these enzymes destroy the antibiotic inside the cellular surface, outside the cells. And the antibiotic is no more longer to work in that particular bacteria. So recently we selected some extremely drug resistant, where we don't have any drug of choice. Like all the discovered drug are resistant towards those bacteria, and that period we don't have any selected antibiotic to give the patient. What we did at that time,
I will take over. The time is limited actually. So what we wanted to show you actually, I wanted to show कि रेजिस्टेंस हो कैसे रहा है और उसके लिए कारक क्या है, फैक्टर्स क्या है, और हमने किया क्या, हमने न्यू एंटीबायोटिक कॉम्बिनेशंस के लिए रिसर्च करी, कौन से ऐसे मॉलिक्यूल है जिनको हम सिनर्जिस्टिकली जोड़ सकते हैं और जो एमडीआर्स हैं उनके लिए हम यूज़ कर सकते हैं. तो जब हमने सिनर्जिस्टिकली कॉम्बिनेशन किया तो देखा कि वो इन विट्रो रिड्यूस हो जाता है. साथ में क्या किया, वी वर इन कोलैबोरेशन विद आईआईटी रुड़की, वी आइडेंटिफाइड अ न्यू मॉलिक्यूल विच वाज नेम्ड एज IITR 00693. दिस वाज combined with polymyxin B to it was used against the MRSA and the Pseudomonas aeruginosa MDR strains. तो इट वॉज वेरी इफेक्टिव. सो एक तो हम सिनर्जिस्टिकली जोड़ सकते हैं और नए मॉलिक्यूल की खोज कर सकते हैं, राइट. अब आगे क्या करना है, एज आई एम इन द हब ऑफ आयुर्वेदा, सो दिस वॉज अ स्मॉल रिसर्च अबाउट द phytochemicals which was done in 2012, which was published. And as you know, all the phytochemicals or maybe the essential oils which are containing the phytoconstituents, which can be used in a different way, like inhibiting the efflux pump inhibition, or maybe plasmid curing, maybe enzyme inhibition, maybe increasing the membrane permeability for combinations. We can manipulate this science to combat the MDRs. Data shows that many plants have these antibacterial or antiparasitic, antifungal properties, antiviral properties also. So these are the chemical constituents, you are well aware of about that. So it's recommended that what we have to do, we have to do a surveillance, we have to increase the use of diagnostic test, the correct reporting, quality reporting. We have to strengthen the infection control committees. We have to regulate the veterinary use of antibiotics, the disposal of pollutant in the river water or the water bodies. We have to regulate the over-the-counter sale of the unnecessarily antibiotics. And we will request the government of India to fund more for the multicentric research so that we can fulfill the One Health approach system. And there should be a well-validated quality check of the medicines which are being supplied. And finally, I will cut the word allied sciences. ठीक है, हम allied नहीं हैं, हम लोग भाई भाई हैं, भाई बहन हैं. ठीक है, अगर हम आपस में हाथ मिला के काम नहीं करेंगे तो हम किसी भी जगह पहुंच नहीं पाएंगे. ठीक है, हर एक साइंस की कुछ लिमिटेशंस हैं और ये हमको एक्सेप्ट करनी पड़ेगी. So, as Swami Vivekananda said, they alone live who live for others. Rest are more dead than alive. So, with that, thank you for patient listening.
Thank you.
Thank you, Professor Balram ji. Thank you very much.
Thank you, Dr. Balram sir, for such an informative session. Please give a huge round of applause to him. My request to our honorable session chairs to present a memento to Dr. Balram sir as a piece of memory from our institution. Please give a huge round of applause to such an eminent speaker. Please keep applauding for him. Thank you once again, Dr. Balram sir. Now, my request to our session chairs to please conclude the session.
इस सेशन में सभी वक्ता अपने अपने क्षेत्र के विशेषज्ञ थे, तो और उसमें हमें लगता है कि ज़्यादा कुछ कहने की आवश्यकता नहीं है। आप सब ने उनके सभी के लेक्चर्स को बहुत ध्यान से सुना और जो क्वेश्चंस थे वो भी लोगों ने पूछे हैं। मैं चूँकि आज ये इस कॉन्फ्रेंस का अंतिम दिन है, तो मैंने करीब 20 वर्ष पहले कुछ पंक्तियाँ लिखी थी। उन पंक्तियों को आपके सामने मैं प्रस्तुत करना चाहूँगा।
Please.
पिछले 3 दिनों में आयुर्वेद आगे है या मॉडर्न सिस्टम आगे है या एक साथ चलें या अलग अलग चलें, इस पर बहुत बहस हुई। हम विकास करते करते लगभग विनाश के पथ पर आ चुके हैं। तो इसी कुछ भाव को लेकर मैंने कुछ पंक्तियां लिखी हैं। इंसान पेड़ से बन तो गया आदमी। कहते हैं कि जो जीव का विकास हुआ वो एक कोश की क्लोरेला से हुआ और उसी से पेड़ पौधों का विकास भी हुआ और जीव जंतुओं का विकास भी हुआ। तो उसी भाव पे ये लिखा है, इंसान पेड़ से बन तो गया आदमी, पर जड़ से कटते ही हुआ आदमी अपने पूर्वज पेड़ का दुश्मन हो गया। और अपने पूर्वज पेड़ का दुश्मन हो गया। सीखा था चलना जब आदमी ने, तब उसने अपने पहले कदम तले। सीखा था चलना जब आदमी ने, तब उसने अपने पहले कदम तले। कुचला था किसी मासूम पेड़ को, और तब से आज तक ये आदमी तथाकथित रूप से आगे बढ़ता गया, तथाकथित रूप से विकसित होता गया, पेड़ों की लाशों पर आगे बढ़ता गया। तो हम विकास तो कर रहे हैं, लेकिन कल जैसे सर कह रहे थे कि हमें नई नई वैरायटी ढूंढनी है, तो जब पेड़ पौधे ही नहीं बचेंगे तो हम वैरायटी किसकी ढूंढेंगे, और फिर आयुर्वेद का अस्तित्व भी खत्म हो जाएगा। क्योंकि आयुर्वेद जो है 90% जड़ी बूटियों पर आधारित है, बाकी मिनरल्स और मेटल्स पर आधारित है। तो पेड़ पौधों को हमें बचाना है। घायल पेड़ों का लहू हरा बहते बहते, जंगल पर आदमी की क्रूरता सहते सहते, धरती का दामन प्यासा प्यासा ही रह गया। हरे लहू का प्यासा ये जालिम आदमी, नदियों की मांग लाल लहू से भर रहा। अब ये केवल जड़ें ही नहीं काटता पेड़ की, आदमी को भी तराशता है बड़े करीने से। यूक्रेन में पिछले 1 वर्ष से युद्ध चल रहा है और कोई उसको रोक नहीं रहा है। कोई पर्यावरण विरुद्ध कितनी सभाएं होती हैं, लेकिन कोई कुछ नहीं बोल रहा है। और दूसरे नाटो के देश उसमें और हवन में घी डालते जा रहे हैं। अब ये केवल जड़ें ही नहीं काटता पेड़ों की, आदमी को भी तराशता है बड़े करीने से। जड़ें अपनी जमाने की कोशिश में, रास्ते में आए पेड़, जंगल और आदमी को विकास के नाम पर ये निगलता गया, धरती को और बंजर कर बढ़ता गया। अब देखिए चारों ओर क्या सुंदर समा है, एक भी फूल पौधे का बाकी नहीं निशा है। कहीं भूल से भी एक पत्ता नहीं खड़कता, हरियाली पर प्रतिबंध है पूर्ण रूप से। अधखिली किसी कली पर भंवरा नहीं मचलता, यह केवल ईंट चूने पत्थर का नगर बन गया। जरा देखो तो सही आदमी कितना आगे बढ़ गया। जरा देखो तो सही आदमी कितना आगे बढ़ गया। तो हमें इस पर भी विचार करना होगा और अपने पर्यावरण को, अपने पेड़ पौधों को बचाना होगा। धन्यवाद।
Beautiful one. Thank you once again, sir. Now my request to Kunal sir also to please give a huge round of applause to our wonderful speaker, session chair of the event.
So, coming to the session, after this beautiful poem by Dr. Anurag sir, being the students of Ayurveda doesn't mean that you stay at a single stage, at a single level. आपको जितना अपने रूट्स के बारे में जानना है उतना ही आगे एडवांस भी करना है। तो ये दो तरफा एक कह सकते हैं कि आपको दो तरफ से चलना पड़ेगा। पहला चीज जो ओरिजिन है हमारे इस कल्चर की, इस मेडिसिनल सिस्टम की, उसको बहुत डीप से आपको समझना जरूरी है, जो आपने पूरे इस 3 दिन के कॉन्फ्रेंस में सुना। पर उसके अलावा जो आज इस सेशन में आपको पता चला वो ये है कि हम इसको जो हम ये नॉलेज गेन करेंगे, जो हमारे ट्रेडिशनल नॉलेज है, हम उसको स्टोर कैसे करें। और वो पार्ट है with the advancement of technology, that is with the information technology, with the advancement of nanotechnology, understanding it with the help of molecular biology, और दूसरे जो sound therapies भी एक advanced technique ही कह सकते हैं, because now we have the understanding of the quantum physics, and using that technology, using that knowledge, we can apply that to the sound and we can understand how the things can work, जो जैसे कि हमारा ओम वर्ड है। Now there are a lot of information that you can see in the NASA website where this Om word sound is actually coming from the universe. Actually, sun makes that type of sound. So it is not that a lot of people think कि हमारे जो ट्रेडिशनल साधु थे और जो हमारे आचार्य थे, आचार्य चरक जैसे उस समय के, उन लोग सिर्फ एक्सीडेंटल डिस्कवरीज करते थे। ऐसा कुछ नहीं है। They had very intense knowledge. The only reason why that went off and another Western traditional medicines could evolve was that at some point it was coming through verbal. So things changed, whereas the other ones like the Chinese medicines या फिर आप अगर Greek medicine की बात करें तो उन्होंने उसको लिखा था. So now it is our responsibility to bring that old traditional medicine, understand it with the modern ideologies, with the modern knowledge, with the modern scientific see-through, and gain that and write it in the right way. That was missing, and advance the technology at par with what Chinese medicines, what Japanese medicines, what Korean medicines, or whatever the Western medicine is doing. We are also capable of doing that. So I think I can conclude from this whole conference as well as this session that we have to move in both the ways. We have to understand the technology, the modern technology, and we have to understand the traditional medicine, and finally we have to merge both of them together so that we get the best of them and we can treat the patients using these traditional medicines. Thank you.
Thank you once again. A huge round of applause for our dynamic session chairs. Now, my request to Professor C.B. Dhanraj sir and Professor Anil sir to please come on the stage to felicitate our dynamic session chairs. A huge round of applause for our dignitaries. I again request C.B. Dhanraj sir and Professor Anil sir to please come on the stage. Please welcome our respected dignitaries with a huge round of applause. I again request you all to please keep applauding for our wonderful session chairs. Thank you once again, sir. Now, my request to Dr. Vedpriya Madam to please come on the stage for valedictory and concluding session. Please welcome her with a huge round of applause.
With the permission of the chair and the organization, I request Dr. Balram ji to come over the dash for the future activities between Patanjali and AIMS.
I forgot to say something to the students and to the organizers and the Patanjali researchers. Ki, you are most welcome to AIMS Rishikesh for any collaboration, for any learning, and I will be most happy to do that. And I think our administration will be also happy to do that.
Thank you so much, Doctor, for this great extension of collaboration.
Yes,
Yeah.
and the PG students should collaborate. They will learn new things. We have got a lot of machines, and we will be happy to join you.
Big clap. Give a big applause, please. Thank you so much, madam.
So thank you so much. प्रिंसिपल सर से और धनराज सर से मेरा निवेदन है अभी मंच पे ही रहें, अभी हम आपको जाने नहीं देंगे। क्यों जाने देना है? अभी सारे बच्चे बैठे हैं। नहीं तो सबसे रिक्वेस्ट आई है, नहीं तो शिक्षकों को तो यहीं अभी रहना है, क्योंकि अब घड़ी आ गई है विनर्स की। तो आप सब प्रतीक्षा रहते हैं। So first of all, I am going to announce winners of oral presentation. सबसे पहले तो हम लोग ये बात कहेंगे कि जितने भी हमारे oral presenters थे, उन्होंने बहुत ही उत्तम प्रयास किया, और आदरणीय रविंद्र रैना जी के लिए हम एक बार क्लैप करेंगे, क्योंकि उन्होंने बहुत अच्छी जजमेंट दी पूरे सेशन चेयर्स जो हमारे थे, और उनको ड्यूली रिगार्ड करते हुए, क्योंकि क्वेश्चंस भी ज़बरदस्त थे और आंसर भी ज़बरदस्त थे और बहुत अच्छा परिणाम आया है। मैं सबसे पहले थर्ड प्राइज अनाउंस करती हूँ। नाउ द थर्ड प्राइज गोज टू डॉक्टर अन्विता सिंह, हिमालयन स्कूल ऑफ योगा साइंसेज, SRHU, देहरादून। आई रिक्वेस्ट डॉक्टर अन्विता सिंह. Let's have a big round of applause. So her topic was impact of lifestyle based yogic intervention on blood pressure and anxiety level in hypertension patients at yoga clinic at Himalayan Hospital, a pilot study. Many congratulations, ma'am. तालियां रुकनी नहीं चाहिए। Can you guess second prize? So second prize goes to आदरणीय डॉक्टर आशीष भारती गोस्वामी जी। फ्रॉम आयुर्विज्ञान एवं अनुसंधान संस्थान पतंजलि। So his topic is Ayurvedic management of a severe case of erythrodermic psoriasis, a case report. So विद्यार्थियों का जोश बता रहा है कि सर बच्चों में कितने फेमस हैं। सर के लिए एक बार दोबारा से क्लैप। अब फर्स्ट प्राइज तो गेस करिए प्लीज। सो फर्स्ट प्राइज गोज टू डॉक्टर विभू सुधाकर पवार जी। So his topic is Ayurvedic management of his topic is multimodal approach in the management of compressive Many congratulations to all winners and best of luck for the rest of the oral participants. अभी छोड़ना है नहीं, तो अभी विनर्स ऑफ पोस्टर प्रेजेंटेशंस का भी अनाउंसमेंट है, तो सर सबको आशीर्वाद दे के जाएंगे ना. सो थर्ड प्राइज गोज टू रितु त्यागी, research scholar, University of Patanjali. Her topic was a cross-sectional study to assess anemia-related knowledge and nutritional behavior among the general population. Many congratulations. Now the second prize goes to Dr. Dushyant Sharma, scientist, Patanjali Research Institute, and his topic is potential mechanistic approach towards diagnostics, delivery and treatment of common illness using nanoparticles. पीछे वाले शांत क्यों हो गए? जबरदस्त तालियां होनी चाहिए. Many congratulations. Now the first prize goes to Kavita Bakshi, research scholar, University of Patanjali, and her topic was patient reported outcomes of adverse drug effects by NSAID therapeutics and views on Ayurvedic regime in chronic pain condition. Let's have a big round of applause. Many congratulations to all of the winners, and dear participants, don't lose your heart. Next time you can also try for this. Sir, please, up. You can take seat. Please, 2 minutes last. Now it's time to conclude the conference, which is perhaps a difficult task. Now the conference, which is named as Achieving Holistic Health Through Ayurveda along with Advanced Technologies, during first day with the blessings of Param Pujya Swami Ji and Param Pujya Acharya Shri, in the presence of our chief guest Adarniya Vaidya Shri Jayant Dev Pujari Ji, Dr. Parmeshwar Arora Ji, and many of the distinguished guests, after the inauguration and several sessions, we now came across to the concluding of this particular conference. In nutshell, we are able to bring Ayurveda and modern technology at a common platform, and we believe that together, like I would like to refer here the word which is used by Adarni Dr. Balram Ji, bhai bhai. Okay, so in together approach, like together we have to grow, we will achieve new heights in an integrated manner. And not only this, केवल रिसर्च की बात नहीं है, केवल आयुर्वेद की बात नहीं है, क्योंकि पतंजलि ने तो पूरे मेडिसिनल सिस्टम ऑफ द वर्ल्ड पे काम किया है. उस पर देखने के बाद भी, सब कुछ देखने के बाद भी, आज इस मंच से प्रामाणिकता से हम ये कह सकते हैं कि आयुर्वेद पूरे विश्व में सबसे पुरातन और सबसे ऑथेंटिक पद्धति है. आयुर्वेद केवल एक पुस्तक ना होकर, आयुर्वेद केवल ग्रंथ ना होकर बेसिकली अ लाइफस्टाइल है. और उसके अलावा उससे भी अतिरिक्त अगर बात कहें, जिसको मॉडर्न प्रैक्टिसेस के साथ, एविडेंस बेस्ड मेडिसिनल प्रैक्टिसेस के साथ पतंजलि उस पर बहुत ही बड़ा और आगे बढ़कर कार्य कर रहा है. Well, I would like to take an opportunity to thank all of our honorable speakers and our distinguished guests. I would like to thank Param Pujya Swami Ji Maharaj and Param Pujya Acharya Shri for their unconditional blessing and always supporting us in all manners. I would like to thank patrons of this particular conference, आदरणीय डॉक्टर महावीर अग्रवाल जी, आदरणीय डॉक्टर साध्वी देवप्रिया जी, आदरणीय डॉक्टर अनुराग वार्ष्णेय जी, आदरणीय डॉक्टर अनुपम श्रीवास्तव जी, आदरणीय डॉक्टर CB धनराज जी एंड आदरणीय डॉक्टर अनिल कुमार जी। आप जोरदार करतल ध्वनि से सहयोग करें, क्योंकि इनके सहयोग के बिना यह कार्य होना बड़ा मुश्किल था। I would also like to thank conference chairman of this 3 days conference, डॉक्टर अनुराग श्रीवास्तव जी, और जिन्होंने बहुत डेडिकेशन से इस कार्य में हमें गाइड किया। आज अनुपम सर बैठे हैं, तो हम चाहेंगे कि हमारे रिगार्ड्स और हमारे थैंक्स उनकी तरफ से पहुंचे। आपकी जानकारी के लिए बता दूं, आदरणीय डॉक्टर अनुराग श्रीवास्तव जी अनुपम सर के बड़े भाई हैं, तो हम उनसे भी रिक्वेस्ट करेंगे कि हमारे डीप रिगार्ड्स उन तक आप पहुंचाएं। My deep gratitude for the PRI faculty members, Patanjali University of Patanjali faculty members, Patanjali Ayurveda Hospital staff members, as well as very honest and जो हम ये तो क्या कहें कि पूरी उपस्थिति दी, तो हम लोगों को आप लोगों पे गर्व है। हमारे प्रिय विद्यार्थी जनों को हम आप सबका हार्दिक धन्यवाद करते हैं। इस पूरी कॉन्फ्रेंस को सक्सेसफुल बनाने में बहुत सारे लोगों की भूमिका है। आई वुड लाइक टू थैंक आस्था टीम मेंबर्स। आप लोग तो बैठ के कार्य कर रहे हैं, ये लोग खड़े हैं, हम आपका हार्दिक धन्यवाद करते हैं और आपके सभी सपोर्ट स्टाफ का धन्यवाद करते हैं। मैं यूनिवर्सिटी ऑफ पतंजलि की फैसिलिटी मैनेजमेंट टीम, दुबे जी और उनके साथ हमारे जो भाई इधर उधर भागते हैं, कहाँ है अभी? आकाश जी ये रहे, लेकिन मेरे टाइम में माइक खराब था। मैं आपका हार्दिक धन्यवाद करती हूं फॉर द बेस्ट मैनेजमेंट ऑफ फैसिलिटी। मैं धन्यवाद करती हूं फूड कमेटी ऑफ यूनिवर्सिटी ऑफ पतंजलि। आपके लिए मेन्यू हमने स्पेशल प्लान किया था। हम लोगों को लगता है कि हमने अच्छा प्लान किया था, कुछ अगर उसमें आपको कमी लगी तो हम क्षमा प्रार्थी हैं। लेकिन फूड कमेटी टीम ऑफ यूनिवर्सिटी ऑफ पतंजलि, मीडिया पीपल आदरणीय कपिल वालिया जी सहित बहुत प्रिंट और सोशल मीडिया की हमारी टीम, ईरा के नेतृत्व में जो सोशल मीडिया की टीम अभी कार्य कर रही है, तो हम उन सब का धन्यवाद करते हैं फॉर द बेटर प्रमोशनल एक्टिविटीज ऑफ दिस पर्टिकुलर इंटरनेशनल कॉन्फ्रेंस। Lastly, I would like to thank all of my team members of Patanjali Research Institute who are very dedicatedly and honestly working days and nights to make this event most successful. आज मैं आपके सामने ये बता नहीं सकती कि कितनी रात्रियों से, क्योंकि कार्य तो चलता रहता है, कितनी रात्रियों से इन लोगों ने अथक परिश्रम किया है हमारे जो डेलीगेट्स के साथ, कोऑर्डिनेशन के साथ, उनकी फ्लाइट्स बुकिंग से लेकर हर चीज को मैनेज करने का, रात को 11-11, 12-12 बजे तक घर जाने का। मैं सभी पतंजलि रिसर्च इंस्टीट्यूट की ऑर्गेनाइजिंग कमेटी मेंबर्स का तहे दिल से धन्यवाद करती हूं। और ये तो आरंभ है, अंत नहीं है। अंत कभी नहीं होता, आरंभ है। आज अभी मेरी बात हो रही थी, क्योंकि कल हमने एक और सेशन रख लिया है, मैंने ग्रुप में मैसेज भी डाल दिया है। आरंभ है ये अंत नहीं है। अंत में मैं केवल इतना ही कहूंगी, अभी तो और संभलना है, अभी तो और संभलना है, अभी तो और संभलना है। आयुर्वेद के सपनों को, आयुर्वेद के सपनों को हमें पूरा करना है, अभी तो और संभलना है, अभी तो और संभलना है, अभी तो और संभलना है। थैंक यू सो मच वन एंड ऑल, थैंक यू सो मच।
आप सभी की ओर से मैं डॉक्टर वेदप्रिया जी का भी बहुत बहुत धन्यवाद करना चाहूँगा उनके नेतृत्व के लिए। आप सब एक बार जोरदार खड़े होकर तालियां बजा दीजिए।